Okay, hi everybody, and welcome to this session of Neuromatch 3. My name's Elaine, and I'll be introducing the speakers today. So I'll just start by reminding you that if you would like to share any thoughts or comments on Twitter, to use the hashtag NMC3. So we have a great lineup of speakers today, and we're going to start with two very different modeling talks and then finish up with an experimental test of cortical plasticity. So I'm really excited about all of the talks. So please use the chat to send messages to everyone and share any interesting resources. And use the Q&A tool if you want to ask any questions of the panelists. So don't be shy, and you can also vote for the questions that you find most interesting in the Q&A. So please take advantage of that as well. So our first speaker today is Joe Pemberton. He's a second-year PhD student in the Neural and Machine Learning group, working with Rui Pontecosta at the University of Bristol. And today he's going to talk about cortico-cerebellar networks as decoupled neural interfaces.
Thanks, Elaine. I'll just share my screen. Cool. Yeah, so today I'm going to be talking about some work I've been doing over the last year of my PhD. And I'd like to thank my colleague Ellen, who's done a lot of work with me, and our supervisors, Richard Happs and Rui Pontecosta. So if you open up someone's head, you'll see this large and very recognizable structure, which we call the neocortex, the most famous part really of the central nervous system. But connected to it at the bottom, you'll also find this thing called the cerebellum, which is more modest, but we also think that there's a big role it has to play within the central nervous system. And in particular, we think that its loop that it forms with the neocortex is very important for learning adaptation, both of motor control, but also more recently, other cognitive tasks, language, even maths, and really any cognitive process is thought is now being shown to be linked maybe to cerebellar processes. So in the motor control case, we have this nice model where we think that the cerebellum, for example, if you want to move your hand, the cerebellum would predict, would make some internal model about what it means to move your hand. And if you send some motor command, for example, to move your hand, the cerebellum would know in advance what to expect and help the motor cortex, in this case, to prepare its activity and to react faster than could be processed by the normal sensory processing pathways. So yes, so the cerebellum wants to replace this feedback, somehow makes an internal model of this feedback, which is inherently slow. When you see something and it gets to your neocortex, this process takes in the hundreds of milliseconds. So we want to ask, what exactly does the cerebellum do to help the neocortex in motor learning and perhaps other forms of learning? Right. So we make the model of the brain or the nervous system as a forward network of control, starting from sensory input we receive from, for example, something that you see. As an example, maybe you see a basketball hoop that you want to throw a ball in. That will send a motor command, which sends to your arm so that you can move your arm such that the ball can go into the basketball hoop. And so we have this forward processing, which are these H's from visual stimuli onto the motor areas, onto your hand. But we also get this feedback signals, which go back a similar route back down the network. And we think that this feedback activity is very important for learning and synaptic plasticity. And in our case, we're going to interpret this feedback activity as some kind of encoding of gradients. So we're going to introduce another separate network, which we call the cerebellum. So if we view this on our left as the neocortex, we have also the cerebellum. And we're going to make the claim that the cerebellum is actually predicting these gradients given the activity of a certain brain area. So in this case, for example, the cerebellum receives an efferent copy of some motor command and will make some prediction based on that motor command in the form of gradients. And how do these predictions make sense? Well, the cerebellum itself will learn to mimic these true gradients, this true feedback activity via the inferior olive, where it will compare its prediction with eventual what we call back-propagated signal or feedback signal given from the hand or leg or whatever. And this setup is exactly what was proposed a few years ago in the deep learning community by Jederberg et al. And it's called decoupled neural interfaces. And the idea is that we have now multiple brain regions or parts of the central nervous system which can work asynchronously without having to wait each other. It just has and has this kind of special relationship and loop with the cerebellar module. So this is the motor case, but this can actually easily be extended to any kind of forward process within the brain. So we can actually think about this more in the cognitive domain, where perhaps we see some visual stimuli, or maybe we hear a certain bit of language, make some associations with that language, and perhaps make a response based on that. And we think that the cerebellum is, even in this case, making constant predictions about what to expect in the future. So it's important to note that this predictions works in the spatial case, where it's predicting activity coming back from other brain regions, but also in the temporal case. So we tested this model on a simple target reaching task, where there's a temporal constraint where it receives input at time step 0, and then 10 time steps later we'll have to associate that input with some target that it wants to get to. And we compare 3 modules, one normal recurrent neural network, in this case an LSTM, without a cerebellum component, and then we compare both the cerebellum component joined to that, as well as a sparsely connected cerebellum component. So the cerebellum is kind of unique, 2 unique qualities of the cerebellum is this massive divergence onto the granule cell layer. I'll go back a slide. There's this massive divergence onto the granule cell layer, but there's also notably the sparse connectivity from the input, which is the mossy fibers, onto these granule cells. So we compare 3 modules, one without the cerebellum, one densely connected cerebellum module, but one sparsely connected cerebellum module. And so for this task, we see that the cerebellum, without the cerebellum, it's actually incredibly difficult for the current neural network to associate some original input with a target and hardly learns at all. Whereas with the cerebellum components, it manages to do pretty well. And if you actually extend that with sparse connections, it performs even better. And the learning curves look something like this, where, as we see, it's a non-cerebellum brain hardly learns at all. Whereas with this cerebellum component being able to transport signals from the future onto the present inputs, we see good learning takes place. And this can be extended to harder sensory motor tasks. So we actually tested this also on the sequential MNIST task, where each time step, the model would receive a row of a number from the MNIST data sets, and then either has to classify that number, so a classification task, or has to associate that number with some 2D coordinate around a circle. And in both cases, we see, again, the advantages of the cerebellum components. The LSTM doesn't do so great by itself, but when you add these cerebellum components, which make these predicted gradients, we see, again, reduced error and better learning. So, okay, so something we kind of were surprised about were why the sparse connection seems to help learning for both these tasks that we've seen so far. So we looked at whether there are any decorrelating effects that happen in the sparsely connected cerebellum. So this was noted by Keiko Gage now a few years ago, that with sparse connections inherently comes decorrelation within the cerebellum hidden layer, so our granule cells. So here we tested a variety of input correlations. So this is our correlations of the LSTM. And with higher correlations with a densely connected cerebellum, which is an orange, we see that we actually get worse performance. And this is because it's a harder job, right? We've got more correlated input, it's harder to differentiate them. But if we were to, if we introduce sparsity on our network, then it actually seems to counteract these effects of initial correlations, and the error remains relatively stable. And here we show explicitly how the synthetic gradients that are produced within the network, i.e. the activity of the cerebellum module, the correlation is higher for a densely connected cerebellum compared to a sparse. And this is true both for the target region task, and we also saw it in the sequential eminence tasks. Okay, so the nice thing about our network is that it can be applied not just to sensory motor tasks, but any kind of task. So we also tested this on a language modeling task, where we asked the network to predict the next word of a sentence, and we can, we asked the network to make a caption of given an image, and we can generate our own sentences given an image with our models. And we see here in gray our non-cerebellum model, and again our densely connected model in orange, and sparsely connected in green. And we saw some qualitative difference in the types of sentences being produced. So this would be akin to cerebellar patients showing some kind of language deficits, which actually has been shown experimentally. And again, if you look at the learning curves, then we see advantages with the cerebellum components. They are, I think, somewhat slighter, but they are still significant compared to the sensory motor tasks. Right, so conclusions. Well, we've introduced a new model of the cerebellum, in particular its link with its interaction with the neocortex, and how we could perhaps view the cerebellum as enabling this kind of decoupled neural interface, as proposed a few years ago in the deep learning community, and allow that different cortical areas to compute forward and feedback activity asynchronously. We've shown the advantages of the sparse connections within the cerebellum. So this notable quality that only each granule cell in the cerebellum receives 4 different inputs has been demonstrated to show computational advantages in this case, in our learning tasks. And these tasks, both in the motor case and the cognitive case, the results of our model and the way it learns is consistent with experimental observations. Cerebellar patients have been shown to not only perform tasks worse, but also learn more slowly. And we think that our model is consistent with this. And finally, our model predicts, or would have shown, that it is especially valuable for tasks in which there's, for temporally challenging tasks in which there's only a loss signal or a reward signal coming later in time, which needs to be transferred, which needs to be transported into some original action or activity. Okay, that's it. So thank you.
Thank you very much, Joe. That was a really clear and really interesting talk. So while we wait for questions from the audience, maybe could you elaborate a bit on why you chose the particular tasks that you chose, and maybe how it links to the deficits in cerebellar patients a little bit more?
Yeah. Okay. So first of all, we wanted to make a task that resembled something of a motor task, so with some kind of initial stimulus and some kind of action that we would relate to that stimulus. We know that the cerebellum's important for time, and we wanted to capture that with this temporal task. And that's why we chose the sensory motor tasks. But we also wanted to look at how this model generalizes into perhaps harder cognitive tasks, to see how maybe the thing with the cerebellum, it's kind of interesting. You think that it's actually, I think originally it was created for the motor purpose, but I think the brain has a good way of generalizing things, and it's been shown now to actually be involved in much more. The cerebellum actually grew at a similar rate as the neocortex, even though the neocortex was doing far more than motor stuff. So I think it's just interesting to see how our model can capture how the cerebellum might generalize into different tasks from sensory motor and into the cognitive domain.
Yeah, thank you. It's a beautiful demonstration of such a wide range of tasks. Okay. I guess if there's no questions, from the audience, or I don't know if any of our panelists want to ask a question, you can unmute. We might move on to the next speaker. Thank you so much, Joseph. That was
sure,
an
no
excellent
worries.
talk.
Thank you.
Okay. So our next speaker today is Akshara Gopiswaminathan. Akshara is a master's student in optometry and vision science at Manipal University, and she works in the lab of Ashwin and Bradley. Akshara comes from an optometry and vision science background, and she's interested in visual neuroscience. And today she'll be talking about overcompensation of lens accommodation in case of convergence insufficiency, a theoretical model. So thanks very much.
Thank you, Aline, for the introduction. So I'm Akshara, and I'm currently a master's student at Manipal University, and this is one of my is not my core project as such. It's something deviated from what I usually do in the clinic. So greetings, I'm Akshara, and my topic will be about the theoretical model of two aspects of binocular vision, that is accommodation and convergence. This is the that is the overcompensation of lens accommodation in case of convergence insufficiency. So the main idea of the paper was put in by Ashwin and Bradley Alicia as well as myself. So before going into the topic, let me give you a short introduction on some of these basics. So convergence is the simultaneous inward movement of both the eyes. The main purpose of convergence is to maintain a binocular fusion. You converge in order to view objects that are at different distances. Constantly viewing targets at closer distances like reading and viewing digital screens leads to fatigue of the muscles involved in convergence. That is the medial and the lateral rectus, which leads to an inability of the eye to move to maintain an optimal binocular function. So this is called as convergence insufficiency. Accommodation is the ability of the lens of the eye to change its dioptric power by changing the curvature in order to make the target clear. Accommodation and convergence are related by two pathways, the accommodation triad and the convergence accommodation dual interaction control system. Both of this will be explained in this upcoming slides. So the accommodation triad revolves around the change in the accommodation of the lens, the pupil constriction, and the change in the vergence. This is sequelae happening in the stimulation of retina with change in the targets that you view. Say you are watching TV that is 3 meters away, and you want to change the program with a remote control. So when you are looking at the buttons in the remote, in the remote control in your hand, your retina gets stimulated to the defocus. This is because your lens was well adapted previously for the 3 meters distance. So this goes all the way through the optic nerve and the optic chiasm, and then from there to the optic tract. From there it goes to the lateral geniculate body, and then to the V1, that is the primary visual cortex at the occipital lobe. The perception of blur is processed by V1 through V4, and these process signals are then transferred into the midbrain and then further into the third nerve complex for further motor and parasympathetic processing. This fires the oculomotor nerve complex, creating the neural stimulation for the accommodation triad. The motor branch innervates the medial rectus. The parasympathetic branches synapses at the ciliary ganglion, giving rise to the short ciliary nerve. The short ciliary nerve further reaches into the eyeball and innervates the ciliary muscles and the pupil sphincter. So this image describes the relay process well. So through the brain through the cortices and the midbrain. So the oculomotor complex, as you can see here, is it's well detailed described here. So it has the collection of both parasympathetic as well as the motor nucleus here, and motor roots. So a positive feedback from the prefrontal cortex excites the Edingle vespal nucleus, which gives rise to the oculomotor nerve. So along with the positive feedback from the prefrontal cortex, it excites the edingal-vespal nucleus. And negative feedback is simultaneously given to the abducens nerve or the sixth nerve, which inhibits the action of divergence, that is the outward movement of the eye. So now coming on to the core topic, because of the prolonged extraocular motility, extraocular muscles excitation, as well as the ciliary muscles contraction due to increased continuous near work, the muscles are fatigued, causing an inability to accommodate as well as converge. This results in blurred vision and diplopia or double vision. So, as the muscles fail to accommodate and converge, the subject or the patient feels asthenopic symptoms such as headache, nausea, and periorbital pain. So, this usually persists as a couple, that is the convergence insufficiency as well as the accommodative insufficiency. But in this presentation, we would be looking into the eccentric clinical presentation of convergence insufficiency with accommodative excess. So let's look into it. So this is the model of the accommodation and vergence. So it is self-existent basically. The target distance is taken as the input for the accommodation model as such, and the target binocular substance is taken as the input for the vergence model. The models process parallelly and do not depend immediately on each other for any component as such. As you can see here, it does not any output from one of the model doesn't actually completely alters the other one. So the output of the accommodation model mechanisms involves only a change in the accommodation, but also a proportional leap of change in the vergence called as the accommodative convergence. And the output of the accommodation model influences the amount of vergence and vice versa. So there is an amount of accommodative convergence for every accommodation output, and then there is an amount of convergence accommodation for every convergence output. So this is usually represented as a ratio for the amount of influence value for the actual amount of convergence or the amount of accommodation that has happened. Although the eyes converge, disparities still exist. And when the extraocular muscles get fatigued, the vergence system is taken over by the accommodative system as a secondary feedback to the accommodation control system. So as the secondary compensatory mechanism for the reduced amount of convergence, the amount of accommodation is more. So this is diagnostically taken in by the ratio of convergence accommodation to the convergence amplitude, and in this case it is higher. The compensatory accommodation is greater than the expected accommodation for that distance while the convergence is less. So the presentation of accommodative excess with convergence insufficiency is rare. The usual presentation would be convergence insufficiency with convergence accommodative insufficiency as discussed previously. So all the therapeutic procedures involves training the vergence system to act independently without accommodative system. The targets are designed in such a way that they produce not only demands for the accommodation, for only convergence and not accommodation. So we train them clinically using vision therapies. So there are several vision therapies, but in this case, we treat them with the VTS, that is the vision therapy software. So they're given with liquid crystal glass that acts as alternate occluders with the vision therapy system software. And also the other vision therapy techniques which we could use is the heart chart. Like we'll ask the subject to read one letter here and then another letter from the chart that is placed at some distance, and then we also give them with the eccentric circles for convergence. So though this gives us a valid explanation, the actual mechanism of acquiring convergence insufficiency in this case is not known. That is, the fatigability of the ciliary muscles is not known in this case. So this could potentially be taken as the research scope. We are also planning to include the variance of range of this overcompensation across the population and among the refractive errors as the correction classes given in the form of spectacles as plus and minus lenses do have an impact on the amount of convergence and accommodation. Apart from this, even the normal subjects, those with no refractive errors, are adapted with plus and minus lenses and checked for these binocular facilities. So we are planning to look into this model to convert it into a comprehensive agent-based computational model. This will have the accommodation and vergence as two separate entities as enclosed in the was disclosed in the previous model. The chart, and that is, it is a dual interaction model, and the inputs will be in the form of accommodation in the form of distance for accommodation model and angular substance in for the vergence model. These are my references. Thank you.
Thank you, Akshar. That was a really excellent talk. So I guess, sorry for the very naive question, but I guess I was interested in whether the timing of fixation is also different in these patients, and whether, or maybe whether your model has implications for kind of saccade to fixation.
Can you repeat it again?
Oh, I was just wondering, like, are these patients, are they slower to fixate?
They are not.
Well? No, it has no, okay. Or whether there's any implications of kind of, yeah, saccades, I guess, as well.
I think that hasn't been studied yet, but yeah, I think that could also be included in as a part of this, like, even, it could be included into this as such.
Okay. Okay, well, we may, thank you very much, Akshara, again, for the great talk. If anyone has any more questions, for Akshara? Or if not, we can move on to the next speaker. Thank you. Okay. So our final speaker today is Dr. Jan Jablonka. Jan is an adjunct in the Institute of Functional Biology at the University of Warsaw, and he works in the laboratory of Piotr Bebas. Jan got his interest in neurobiology during his PhD at the Nenki Institute of Experimental Biology. And today, Jan will be talking about the interhemispheric interactions in cortical plasticity. Thanks very much. Sorry, Jan, you're still muted.
Okay. Thank you, Elaine, again. Today I would like to present the results regarding interhemispheric interactions participation in the cortical plasticity. The general idea of these experiments came from my previous works on my PhD studies on the cortical plasticity after stroke, when the interhemispheric interactions are strongly altered. The injured hemisphere has diminished activity, the spontaneous activity, and the cortical plasticity was also changed. We previously thought that it is inhibited, however, later it seems it works a little bit different. But at this moment, we just repeated the experiments with the same paradigm, where the experience-dependent plasticity was induced by partial whiskers deprivation on rats, and the 2-deoxyglucose brain mapping was performed. The size of the cortical representation of the spared, the non-deprived whiskers were measured, and later the local field potential was measured. I will say a bit more about the methodology. The unilateral whiskers were cut for a month, and rats used just one row of whiskers, row B. because there are 5 rows of whiskers which are repeatedly growing on each animal. And after and this row of whiskers we call spared row or non-deprived row of whiskers, in the contrast to all the other which were deprived on this side. And this whisker, these row B whiskers, will be called undeprived. After a month of deprivation of these whiskers, the 2-deoxyglucose brain mapping was performed. The 2-deoxyglucose is incorporated in the areas proportionally to the activity of the tissue, of the metabolic activity, and within the brain it is proportional to the activity of the neuronal activity as well. However, it incorporates to the astrocytes and neuronal cells and inhibitory and excitatory cells as well. During the 2-deoxyglucose brain mapping, 3 paradigms were performed: the unilateral stimulation of whiskers, of the spared row of whiskers, which gave has the representation in the deprived hemisphere, contralateral hemisphere; unilateral stimulation of the non-deprived side, row B, and bilateral stimulation of both side whiskers. Here you have the cytochrome oxidase stain section with the with the Barrel field represented in the layer 4 of the somatosensory primary cortex of the whiskers representation. And here you see the rows where it's strongly somatotopic, and each whisker is neighboring the same whiskers as on the rat's snout. And here you can see the, oh, I didn't say that. Later, after the 2-deoxyglucose was injected, it's the methodology, although it's quite good in the spatial activity distribution, it gives results on the lethal after postmortem, yes. So later the sections of the brain of the flattened cortex were exposed to the autoradiograms, and the artificial colors were fit to the optical density of the autoradiogram. So what we can see here is the cortical row B representation within the Barrel field, and the width of this representation was measured. After a month of the width of the optical density, and after a month of such deprivation, the bilateral stimulation of row B of whiskers, that are whiskers of just one row of whiskers on each side, result in an enlargement of the cortical representation of the spared whiskers in the contralateral hemisphere. The enlargement, the cortical representation was widened by 50%, and you can see in here the autoradiograms with from the undeprived hemisphere and from the deprived hemisphere, the widened cortical representation of the row B representation. Here is the auditory cortex. What we have found that the unilateral whiskers deprivation of the spared whiskers results, instead of doubled width of the cortical representation, resulted in just in 20% of widening when the 2-deoxyglucose brain mapping was when we use the visualization of the width by the 2-deoxyglucose brain mapping, brain activity mapping, obviously. So you can see here the bilateral stimulation results with the widened by much more widened cortical representation of the spared row of whiskers than after the unilateral whiskers deprivation. So we have the same result, the same the same way the deprivation and the cortical plasticity was performed. However, 2 types of the stimulation during the 2-deoxyglucose brain mapping resulted in different image within the cortex of the cortical representation within the cortex. And here you can see the comparison between the undeprived and deprived Rossby whiskers representations and the doubled width of the spared whiskers representation when the bilateral stimulation was applied. So the overriding conclusion is that when we stimulate the whiskers bilaterally, we can see some impact from the ipsilateral whisker stimulation. Because when we stimulate just the contralateral whiskers, the width of the enlargement of the row, which we can visualize with 2-deoxyglucose brain mapping, is half of that when the bilateral is applied. So in here, here we show that the bilateral whisker stimulation seems to visualize with 2-deoxyglucose brain mapping not only the real enlargement of the spared whiskers representation, but also some impact from the ipsilateral stimulated whiskers, the homotopic whiskers of the ipsilateral. However, to be able to confirm the idea that the surrounding whiskers might be that the surround of the whiskers representation, which are enlarged during the experience, after the experience dependent plasticity was induced, we made some electrophysiological recording from the cortical surface. The recordings were a bit more complicated. However, in here, I just show you some preliminary data from the cortical surface recording. And we recorded from the 16 electrodes which were positioned perpendicularly to the row B, cortical row B representation. We did our best to make it perpendicular. And in here you can see on each hemisphere, oops, sorry. No, I thought I have another slide in here. In each hemisphere, there was an electrode with 16 points recording from both sides, like in here. And you can see from 32 channels and from contralateral whisker stimulation within the response within the undeprived hemisphere, and the deprived hemisphere has a silent response. In here, you can see the spared row whisker stimulation. And in the contralateral hemisphere is a huge response. In the ipsilateral is just tiny response, and the bilateral whisker stimulation resulted in bilateral response within hemispheres. In here, I just show as an example the response within the layer 4 of the barrel field. And you can see that except the direct response to the contralateral whisker stimulation, which is the huge and the strongest response to the whisker stimulation, we also have the slight response, a bit postponed, to the ipsilateral whisker stimulation. Postponed so that probably a few more synapses we have between the ipsilateral whisker stimulation and the response we record within the ipsilateral hemisphere. Because we can't say much about the response just basing on the local field potential, we have to compare the response between electrodes to be able to say what is the source of the activity. And in here, we performed the kernel current source density analysis. It's a kernel current source density which assume that which assume not equal the electromagnetic field distribution around the electrodes, because we have the recordings from the surface, so we have an error on one side and the tissue below. In here, you can see the sinks and source of the potential changes of the current flow. And sinks are blue, sources are red. And recording from the surface, cortical surface, we see the position of the electrodes is signed with the dashed blue line. And the sink in here is most probably the response of the current flow due to the source of the activity within the layer 4, which we observed before, and within the layer 3 just after the whisker stimulation. And later we can see the source of the activity, which is result of the cortical surface, the current flow from the supragranular cell layers. And we compared the response from the bilateral and unilateral sinks and sources within the within defined time points. In here, you can see the exceeding source of the cortical activity after within the deprived hemisphere, which exceeds the huge sink after that. And the comparison result with the difference between the sources within the undeprived hemisphere, although the similar.
Come close to wrapping it up, Jan. We're close to time.
We had to time. Okay, so the differences between hemispheres. Between the bilateral and unilateral stimulation allows us to conclude that which we show within the to the oxyglucose brain mapping and the source and sinks and source analysis allows us to conclude that the changes within the wiring in response to the new experience in one hemisphere is partially modulated by the ipsilateral sensory input. And although when we assume, as it is with, in the case of the cortical viscous representation, as we assume the complete cross of the representations, the controlateral hemisphere has to participate in the experience-dependent plasticity rearrangement within the cortex. Okay, thank you. So to assure that we also performed some deeper layers recordings of the local field potential. And whoever would be interested in analyzing and participation in the analyzing of the data, because it exceeds a bit my computational abilities, it's cordially welcome. And you can contact me to this address. So thank you.
Thanks very much, Jan. We have a question from the audience from Avital Hahami. He says, thanks for the talk. I wonder if this plasticity is really the result of interhemispheric stimulation rather than the result of simultaneous stimulations of more than one whisker. Have you tried depriving all but two whiskers on the same side and then stimulating them both? This would still be unilateral, but would evoke more inputs on the deprived side. I wonder if this would also increase plasticity, like interhemispheric stimulation.
Wait a second, I lost the last few sentences.
Oh, I'm sorry. Can you see the question, or shall I?
Yes, I see. This would be still be the unilateral stimulation. While but to accept what would be the difference between in this case?
Well, I think the
stimulation. To be honest, I'm probably too confused to understand the idea. Because I don't see the difference between the stimulation of deprived of this but you want to leave just 2 whiskers, or you want to deprive just 2 whiskers?
It says leave just 2 whiskers, I think.
Okay,
yeah.
so we would leave just 2 whiskers. And what would be the difference between leaving 2 whiskers instead of leaving the row of whiskers? I don't see the difference. I mean, there is a difference because there will be different cooperation between the barrels. However, if we compare, because
And so he says, because then you can stimulate 2 whiskers on the same side.
yeah, but in here I stimulate entire row of the whiskers. So I stimulate 4 whiskers, for 5 whiskers, yes, from the row B. And I see the representation of entire row of whiskers. If I just will choose 2 whiskers, it's always a question which we should choose, if the longest one or straddler or the more frontal, but I don't catch the difference. I would stimulate. Yeah, the idea that it's the interhemispheric interaction based mainly on the concept that the whiskers within the the cortical representation of the whiskers is completely crossed. So if it would be not, although it's assumed that it is, but if it would be not, we still can say that the ipsilateral stimulation of whiskers have some influence on the changes, experience-dependent plasticity changes induced within the cortical representation, even though it is not necessarily contralateral then. But so far we can, based on the data, we can assume that it is the interhemispheric interaction per se. And if you want to know something more, please ask me on the email, because I might not catch the idea of this question. Sorry.
Well, thanks very much for the answer, and thanks for a really interesting talk. So I guess we'll finish up the session now. So I'd just like to thank all of our speakers once again. I really enjoyed it.
All right, welcome everyone. And let's perhaps wait for a few more minutes to see if there are new people coming in. Okay, I'm counting down 20 seconds and then we can start. All right, hello everyone. Welcome back to the Neuromatch Conference 3.0. Good morning, good afternoon, and good evening, depending on where you are located in the world. My name is Lei Zhang. I'm currently working as a postdoc researcher at the University of Vienna in Austria, working on the field of social decision-making. And in fact, if you are indeed interested in what I am doing, I will have my own interactive talk in the next hour, and I will put the link into the chat in the next minutes. But today is not about me, and today me and Sophia from University of Leeds in the UK will be the hosts of this interactive session. And in case you're new here, let me just briefly recap the format of the interactive session. So as the name just suggests, it will be interactive, which was created to largely resemble the post-presentation format. So the speakers, they will have a short walkthrough for about 5 minutes to cover the topic, and then we are expected to have some interesting and ideally insightful discussion afterwards for about 10 minutes. So we are using the Zoom webinar format. If you're new here, there will be 2 functions that we will be using. One is the chat, like you can just chat to each other and pay attention to who you will be chatting to. You can select either to the panelist or panelists together with all the attendees, like the audience. And the other more important function is in fact the Q&A function. And then there you can ask your questions and for the discussion. And please do indicate yourself in the question to say if you want to show up onto the stage or not by using this on screen in the curly square bracket. And if you do not want to show up yourself, which is totally fine, and then I will just read the question for you. And if you're on Twitter, feel free to use the hashtag NMC3 to show and to share everything going on around today's topic and today's discussion. Today, the topic will be about translational neuroimaging that uses neuroscientific findings to inform pathology and brain diseases. And we have 2 exciting speakers today. And the third speaker, unfortunately, cannot make it. Joining us today are Johannes and Antonia. Both are working at the University Hospital Bonn in Germany. And in fact, they are from the same lab, the Translational Neuroimaging Group led by Dr. Theodor Huber. I will also put the link of their webpage into the chat. So the group has done some really amazing work on pathology, on the pathology of limbic encephalitis diseases. And today you will be hearing about 2 recent pieces of works from different perspectives. And today, the first speaker will be Johannes Theta, and he will be talking to us about the volumetric analysis reveals bilateral cerebellar atrophy in Rasmussen encephalitis. And Johannes, the stage is yours.
Okay, thank you for the kind introduction, Lai. I'm gonna share my screen. Moment. So now you should see my presentation, right? Okay. So thanks for the kind introduction and thanks for joining. Today I'm going to present you, at least in my opinion, quite interesting and fascinating disease. Some of the work my colleagues and me have done to get a better understanding of this condition, and then finally have a discussion with you. But let me start with a brief case presentation of a 6-year-old girl who was admitted to our hospital because of an aggravation of epileptic seizures and the new onset of unilateral signs of paralysis. And then as a clinical follow-up MRI diagnostic MRI was performed, shown here. And this MRI showed severe unilateral neuroinflammation and atrophy, which was restricted to one single hemisphere, and that gave the crucial hint towards the diagnosis, so that the diagnosis of Rasmussen encephalitis could be made. And Rasmussen encephalitis is characterized as a strictly unilateral disease with progressive neuroinflammation, as it is shown on the image on the right, and following atrophy. But the problem on this disease is that it's not very well actually understand what's actually happening, because there are so few cases of this condition. But in our hospital, we have a relatively high number of Rasmussen encephalitis patients, and therefore we've performed the cortical thickness analysis, comparing around 60 Rasmussen encephalitis patients with matched healthy controls. And the results we've got were that there was, as expected, on the ipsilesional hemisphere, widespread atrophy observable, while on the contralesional hemisphere there wasn't significant atrophy in our results. But something we've noticed while looking at individual MRI scans was that not only the cerebrum was affected, but there are also cases in which the cerebellum, in fact, seem to be affected. It is shown here. This patient, the cerebellum on the contralesional side, seems to be atrophic as well, following the neuroanatomic connection between cerebrum and cerebellum. And other cases we've seen follow the opposite pattern. So here, for example, you see that the ipsilesional cerebellum is affected. And to quantify this observation, we've performed the voxel-based morphometry study comparing Rasmussen patients and controls. And what we've seen there was that, as expected, on the contralesional hemisphere of the cerebellum, there was a vast amount of atrophy, as it is shown here on the slide, on the image. But there was also some atrophy on the ipsilesional side of the cerebellum. And another observation we've made was that also the individual patients sometimes differ very vastly in the regional affection of atrophy within the brain. So, for example, here you can see a patient who is affected very much on the occipital side of the brain, while this patient, for example, is affected on the frontal lobe. And therefore, we have subdivided our patient group into 6 subgroups, and repeated the cortical thickness analysis. And what we've get was that was shown here. So, for example, there were cases that very much a frontal focus of cerebral atrophy, while there were some others who had an insular one, and so on. And then we ask ourselves whether these different groups would also show distinct atrophy patterns within the cerebellum. And therefore, we also repeated our analysis of the cerebellum. And what we've seen there was, at least, for example, for the frontal group, surprising to us. So it seems that the frontal group did not show significant atrophy on the cerebellum, while, for example, the occipital, the temporal, and the insular group show relevant atrophy in the cerebellum. Some of them on the ipsilesional side, and for example, the occipital group, the temporal group on the contralesional side. And for example, group of diffuse atrophy pattern on the cerebrum did not show significant atrophy on the cerebellum. And to sum up, I would like to say that we found that the cerebral atrophy patterns are seeming to be differing between different patients in in Rasmussen encephalitis, and that depending on this subgroups, also the cerebellar atrophy pattern are quite different in these groups. Yeah, now I want to thank my my lab colleagues, other way around, and the Department of Epileptology, as well as the Institute of Experimental Epileptology and Cognition Research in Bonn. And I'm looking forward to your question. I think that was very fast, so I could imagine that there are some questions regarding disease or the presentation. Thank you very much.
Great, fantastic. And we'll maybe do this knocking table way. Very great. Let's see if we have any questions from the QA. Anyone who's joining, feel free to ask questions. And perhaps, if not, I will just start. So you mentioned one of that is you subdivided the group into 6 small groups, subgroups, and how or in which criteria you are doing this division, or classification analysis, or just some, how do you do that?
Yeah, thanks you for the question. So for the first, we did it just kind of visually. So as I showed on this slide, there are some patients who did really show a kind of clear atrophy pattern. So then we so therefore we have chosen them visually in these groups, and we've chosen just all lobes as a group, and all those patients did not fit in any of the lobular groups we put into the diffuse group. But of course, we could do this classification also in a automatical manner. But the problem here is a little bit that sometimes the pathology is so severe that, for example, the free surfer analysis we did is difficult to, or at least it's difficult to get information in which exact region is atrophy, because the labeling of the regions in free surfer is kind of wrong because of the severe pathology, and therefore it's difficult to use, for example, the free surfer oscillations as classification criteria.
Yeah, great. That's very helpful. Let's see if we have any questions. Feel free to ask questions in the Q&A session or also in the chat. And also let us know if you would like to show up on the stage, open your webcam and web camera, and then to speak directly to the speakers. Okay.
Maybe I have again one question. Did you classify the patients by atrophy on the affected side or the unaffected side?
No, yes, thank you for the question. So we classified them regarding the atrophy on the affected side, because on the unaffected side there was not really much clear atrophy measurable. So at least not very significant atrophy. So we definitely classified them according to the atrophy on the ipsilesional hemisphere.
Okay, great. Maybe I have a follow-up question regarding still the subgroups. Maybe let's look at question from the Q&A first. So the question from Essay, and he said, or she, I'm really sorry about that. So I might missed it. How was the patient's cognitive states? I mean, cerebellum has strong relation with language and working memory. So they are cognitive states.
Yeah, that's also an interesting question. To the best, what I have known is that, so this phenomenon of cerebellar involvement in when you have a severe cerebral disease is already known, for example, from traumas or from big tumors, and it's called cross-cerebellar atrophy. But both in our cases and in these studies, there was never a clear cognitive sign, for example, for the cerebellar involvement shown. So, for example, they did not show any cerebellar symptoms like intention tremor or anything like that. So it seems like that it's difficult to imagine that, for example, something like that wouldn't have a clinical presentation. But until now, what I've known is that there are no clear signs of cerebellar symptoms.
Yeah, great. That's an interesting question, an interesting answer. Yeah, yeah, great. Are there any other questions? Maybe I can start my follow-up question. Sorry. So this subgroup division analysis, can that also be generalized? Let's say if there are new patients and then using the current analysis of the sample, can these results also be generalized somehow? If you maybe analyze the voxel-based cortical thickness, and then can you classify the new patients into one of the 6, or how to do respond to this kind of question? Yeah.
Yeah, I think so. I mean, based on the patient just need an MRI, structural T1 sequence, but based on that, it should be in fact possible to classify them then in one of the 6 groups. Yes, and we are currently into diving deeper in this and seeing whether there are maybe clinical implications of this. So, for example, we have seen, as I mentioned, that for example, the patients affected more frontally did not show atrophy in cerebellum, the group study, but the one, for example, that insular involvement did show atrophy. So we are currently looking into whether that could have a clinical meaning. So, for example, it's for this patient, it's very important to decide what treatment they should get, because there's a kind of a dilemma between medical treatments like anti-epileptic drugs and immunosuppressive therapy and surgery. And the problem is that often you do not know who patients have a so severe disease that a surgery will be necessary. And it could be that, for example, the assignment to one of the groups could predict whether surgery makes sense or not. Yeah.
Yeah, great. Thank you. It's very interesting. There is another interesting question from the QA. And Johannes, can you also see the Q&A? And if you can first read it loud and then also answer that.
Okay, so contralesional cerebellar. Okay, so I read it now. Contralesional cerebellar atrophy can be explained by diaschesis, I guess. How would you explain ipsilesional cerebellar atrophy? Yeah, that's also quite interesting question. The thing is, I've shown you the 2 examples, and in this case seems to be quite clear. So this is probably due to this phenomenon called cross-cerebellar atrophy, but this isn't. But one suggestion is that primary disease affecting the cerebrum could have spread here to the cerebellum, so that Rasmussen encephalitis actually affected the cerebellum in this case. And some hints towards this suggestion is, for example, that the white matter of the cerebellum on the ipsilesional side seems to be also very much involved. So there are hypointensities here, and the cerebellum looks not very symmetrical and is quite locally affected. So therefore, one explanation could be that the primary disease affects the cerebellum in this case, but it's still not understood actually what's happening when the cerebellum in Rasmussen encephalitis is on the ipsilesional side affected.
Yeah, great. It's a very interesting question. Are there any more questions from the audience, or also from Antonia as another speaker? No, she doesn't have. Maybe the last one before we move on to her talk. What is the current stage of this research? I'm asking because do you have any follow-up analysis plans, or how do you want to move forward?
Yeah, so to examine what's happening in the cerebellum, our plan is to also examine the white matter in the cerebellum, and especially also the white matter within the brainstem, because we want to see if there are signs, or is it possible to see also atrophy in the white matter, indicating to how much extent cross-cerebellar atrophy as a secondary atrophy is occurring here. And on the one hand, we already have performed a voxel-based morphometry of the white matter, but one other thing we want to do is to look at DTI data of those patients. And we are very much interested whether we maybe seeing their changed FA values in the brainstem, or something which hints towards a disconnection between the contralesional cerebrum and the cerebellum.
Yeah, perfect. It sounds really like a plan. Oh, great. So let's thank Johannes again. And I hope the discussion is useful and helpful in some way. So let's move on to the next talk. The next talk will be from Antonia Harms, and her talk will be about brain plants capture stage-specific lateral shape asymmetry in autoimmune limbic encephalitis. The stage is yours. I tried to practice already beforehand. Still didn't really work out very well. You will have to unmute yourself, Antonia. Yes, great.
Okay, now can you all hear me?
Perfect.
Perfect, okay. Yes, so I will talk about limbic encephalitis, as you have already said, thank you. Yes, and I brought you the case of, let's call her Mrs. Burns, and she came to our clinic with sudden amnesia, and she suffered from epileptic seizures. And when we examined her blood, we found LGI1 antibodies, which are antibodies against the structure in the brain, in the limbic system. So then we performed MR imaging, and we found some increased signal intensity in the temporal lobe area. Then what we did then was to treat her with high doses of corticosteroids, and her condition got better really quick. The problem with limbic encephalitis, however, is that not all limbic encephalitis subgroups can be treated that easily with corticosteroids. And a second problem is that not all groups show the same clear image picture of the MR imaging. So what we're trying to do is to develop imaging biomarkers that can classify each limbic encephalitis group into their corresponding antibodies. And we do this by looking at the volume and the structure of subcortical structures. And since the volume analysis has already been done in several cases, we are now focusing on the description of the shape of subcortical structures. And we use a tool that is based on the segmentation of Resurfer, and it classifies the structure as a vector of eigenvalues. And this is really handy because now we can calculate the distance of each vector of eigenvalues for each structure from the left hemisphere to the right hemisphere, and by that get a within subject asymmetry measure. And we applied this tool to 9 different subcortical structures to our different patients who have antibodies against the glutamate acid decarboxylase and LGI1 and a protein called Caspr2. And we also subdivided our patient groups into an early and a chronic disease stage. And then we matched healthy controls by sex and age. And this is some first results we found so far. This actually depicts that the shape asymmetry captures the different serotypes of limbic encephalitis and also the different stage of disease the patients are in. And then as the next questions, we ask ourselves, which structures actually differ significantly from the healthy controls in their shape. And we found the amygdala, the thalamus, and the cerebellum white matter to differ significantly from the healthy controls. So now the final question is, does shape information about the shape actually gives us additional information, or is it just a repetition of the volumetric analysis? So we repeated the same study with volume measurements, and we found different subcortical structures to be significantly different from our matched healthy controls. So as a summary, we could say that shape actually adds new and complementary information to volume analysis, and it captures stage and serospecific lateral asymmetry in this specific pathology. So now our goal is to combine all this information about the morphometry so that we can actually classify our patients correctly and then treat them with the according therapy. Yes, again, thanks to my lab and to the Department of Epileptology, the Department of Neuropathology, and the Institute of Experimental Epileptology and Cognition Research, and the DZNE in Bonn. We can do this research, and yeah, maybe now we can start into discussion. I brought some questions. I don't know if you first want to ask your own questions.
Yeah, either way. So brilliant talk again. Thank you very much. And to this, and also this. Let me try to ask Johannes, even though you are lab mates, do you have questions to your colleague?
I do have a question regarding the different subgroups of limbic encephalitis. Is it always the case that this amygdala swelling is observed in limbic encephalitis patients, or does it differ between the different serotype groups, or are they in general more similar, or are there differences?
In the volumetry of the amygdala, there are differences between the different serotypes and the different stage the patients are in. So first, the amygdala is swelling, and of course, in the late stage of the pathology, the volume of the amygdala decreases. I don't know if that's answering your questions.
Yeah, thank you. And I meant clinically, are they more similar? So are there differences between LGA1 or Caspr2 or gut patients, or do they behave in the same way you, for example, showed at the beginning?
No, and also in volumetry, they behave differently. So the volumetry captures in the amygdala swelling, it captures differences between the serogroups.
Yeah, great. I guess that's the questions from your colleague, Johannes. Perhaps that's the most challenging one you will have from today's session. And let's see if we have questions from the chat or the QA. Yes, Bastian or David, the question, provocative question, what use would an imaging biomarker be in the end? As in, do you think that neuroimaging biomarkers for LE will ever replace the serum markers, for example? I think the question is perhaps about the precision or accuracy, if you want to use the biomarker or the marker to try to classify and then eventually to treat patients.
Yeah, so
it's my interpretation. If I'm wrong, then Bastian, feel free to come onto the stage to really speak about yourself, for yourself.
Yeah,
sorry, Antonia.
There's several reasons actually. So one reason is if we actually find a good classification for those subgroups, and we don't need to investigate the liquor of the patients anymore. So we don't have to get their cerebral spine fluid, which is always a little risk for the patients. So that would be ideal if we actually can classify them that good that that doesn't have to be done anymore. And the second reason would be that not all patients show antibodies. So there are patients that cannot be classified by their antibodies, but they clearly by clinical symptoms have a limbic encephalitis. So we are hoping to find a correlate in the shape and volume analysis that can classify our patient groups according to the therapy, so that we can apply the correct therapy in the end.
Yeah, great. I guess this is the idea of personalized healthcare or personalized treatment, right? So using one method is not really enough, and then there will be the physicians, the doctors, and at the same time, the complementary, there will be also like machine learning techniques. For example, as you talk about here, like the classification, and then all of them, if they really reach some kind of agreement, then that's perhaps an easy treatment because there is no disagreement. But if there are some disagreement, I always wonder about this question. So if the doctor says one thing, if your statistics or data says another, so how do you use your expertise or domain knowledge to resolve this challenge or difficulty or discrepancies?
Yeah, that's a really interesting question. I would say it's a little bit philosophical, nearly. So
Johannes, also feel free to join in this question.
So I cannot answer the question, but I can maybe give my opinion on that, because until now, since I would say that the experience of physicians weighs really heavily, and especially in this case of limbic encephalitis, the classifier that we have not yet developed. Of course, we cannot apply it. So until now, we are dependent completely on what the physicians decide and say. However, there's other classifiers that are really good and that are sometimes maybe even better than the decision of physicians. So my opinion is that that data-based decisions, or the data maybe in future actually beats physician knowledge, but that's just my opinion. So I'm open for critique.
This is indeed a challenging question, and we are happy to hear about different opinions, I believe. So Johannes, what's your opinion?
I can also just share my opinion, but I think that the combination of both will make it, because I think there should always be a person behind deciding important questions. But on the other hand, I would expect that an informed doctor who have seen, for example, the results of a morphometric study will make the best decision. So I think the combination of both is maybe the best model for the future. But of course, to reach this, I think most have to be done in getting better tools. So I think we should focus on that the most.
Yeah, that's great. Also, thank you for sharing your opinion. And just specific to this specific analysis that Antonia just presented. So this type of analysis, how easy that can be implemented, for example. So if you are not, if the patient doesn't go to University Hospital Bonn, instead she goes to Hamburg, for example, and then there's another doctor, and the doctor wants to use this analysis, and that's the researcher there or the physician there has to contact you to use your expertise, or they can simply use some package or toolboxes to see to simply implement the analysis that you presented.
Well, so the toolbox we're using is by the lab of Martin Reuter. I had him here in my.
Yeah, feel free to share the link, of course.
Yeah. Yeah, so on the right side, and his group developed this toolbox, and I think it's really easy to you just have to sign up for the toolbox, and then you can kind of put in any T1 weighted image and for the segmentation of free surfer, and then do the analysis of the surface. So you don't have to be in Bonn to do that. You can just sign up at the webpage of Brainprint to.
Okay, that's encouraging. Yeah. Okay. Great. Thanks. Are there any more questions from the audience or from Johannes? Okay.
I have one more question. Regarding those patients who are not classified into one zero group, is there a data set in our clinic, and have you already looked on them in your analysis, Antonia?
I haven't included patients that were not classified in one zero group yet. I think that would be a next step when we find similarities in the groups of the antibody groups that we know. And as a next step, you could look into patients where no antibodies have been found and where limbic encephalitis is just an idea, but has not been verified completely, because there are no antibodies, and without the antibodies, it's really hard to say that the patient suffers from limbic encephalitis.
Great, thank you. And then perhaps one last question from my side. I asked this already to Johannes, the same question. So what's your follow-up, or how do you want to proceed with your project, Antonia?
Yeah, so we are looking at more data in the future, because of some patients we have not just 2 scans, but like 5 or 7. So what we are doing next is to look at the longitudinal development of the volumetry of the structures, as Johannes already asked about, for example, the amygdala that is first swelling and then the volume is decreasing. And by looking at that, we want to know a little bit more about the pathology, and also, of course, go on with our classifier so that we can actually try to develop that.
Yeah, great, thank you. Perfect. So let's just thank Antonia again for this amazing talk and also for the discussion. I hope that's helpful and insightful. So if you're new here, our third speaker, and she canceled her event, and we should just thank the speakers, both of the speakers, Johannes and Antonia, one more time, and also thank all those audience for this actively involved in the discussion. And I hope to see you soon in some later sessions during the Neuromatch conference 3.0 today and also tomorrow. So then bye-bye.
Also, thanks to you.
Yeah, thanks a lot.
Perfect.
Buenos Aires, Argentina.
Yeah, that's right. Okay, so would you okay if I say you are from University of Buenos Aires?
Yeah, it's okay.
just
Perfect.
to know, because I tried to Google, you know, the speakers, and I just couldn't find and say, oh my God, I need to ask that before we started. But yeah, it's.
I think we had to write it down on the form when we submit it for the presentation, but that's okay.
Yeah, the thing is they didn't inform us, so we don't have that information. So
Oh.
like, yeah, you know, it's a huge number of people participating, so it's not really. Hi. I know. Yeah.
Hi, sorry, I didn't turn on my video. I forgot about it.
Yeah. You are two, right?
Yes, I'm Yang.
Yeah.
Yang Chu, yes.
Yang Chu. Okay,
yes.
Great.
I think it's 5 minutes before we start. I can start the meeting now so people can get into.
Done. Okay.
Yeah. Cool. Try to start it. Hi.
Hi.
So I have to share my screen like in a regular Zoom meeting and share my presentation. That's okay.
Yeah. Yeah.
Yeah.
Give me just a second. I can just. Okay, so we have.
Yeah, so I started the broadcast. Everyone should be able to join.
Okay. So I have in the schedule that Huawei is gonna start, then Jin Ting Ling, and later Soledad. Is that right?
Okay.
You agree? Oh, you are gonna start, Soledad?
No, I think I was on the first grid, but I don't care the order. The real
I
order.
mean, here are just the names, but I mean, I don't know.
Oh, that's perfect. I can be last. It's okay.
Yeah, it's.
Yeah, that's perfect.
Yeah, I mean, actually, let's see whether Huawei is coming early. If she's not, maybe I just move to Jin Tingling. Am I saying right, Jin?
Yeah, I'm fine. Yeah, that's
Okay.
fine.
Can you tell me where are you working from?
New York University, Abu Dhabi.
Okay, can you write in the chat, please? I just want to introduce you.
Okay, so you're Haiti, right?
Yeah. Yeah, yes.
Let me see if you can type. Yeah. Okay.
That's great. This is good. Okay, that's New York University, or what is?
Yes. Yeah.
Abu Dhabi. Okay, great. New York University, Abu Dhabi.
Okay.
So, and you are doing your PhD, postdoc, or?
No, I am a research assistant here.
Okay, great. Research assistant. Okay. Perfect.
So Soledad, would you please test if you can share your screen and if everything is going well?
I share my screen, so you test
Yeah,
it or what?
yeah, to see if it is okay.
Yes.
I think we got 5 attendees for now already.
Okay.
I just share it. You can see it, or
yeah, cool.
And if I move, it's just
Great.
Yes.
Huawei, hi. Can you? See. Hi, Huawei. Can you hear me? I don't know whether he can. Yes. Great. Should we start now? Yeah, okay. I think it's time. So, hi everyone, welcome for being here. Today is the 29th of October, 2020. This is the session 13. This is the room number 3, and we are in the Neuromatch conference. So my name is Isaide Garcia Lázaro. I'm hosting this session. But before to start, I just wanna thank our speakers. They are Huawei, Jin Ting Lin, Soledad Pico, and a special thanks to Chu Yang. He is our co-host. He's in the back. He's making sure that everything here works perfect. Okay? So before to start, I also wanted to remind you that we have our Q&A question in the below of your screen. There you can pose your questions. And remember, there are no dumb questions. So Neuromatch is a safe place. So the more you ask, the more you are gonna get, okay? You can also use the chat right below. There you can also ask for help if you have any technical issue, you want just to leave a comment, or you want just, you can use for talking to other attendees. And don't forget, you can use the hashtag NMC3 in Twitter just to continue the conversation and, you know, make more public our work. So we have to start with Huawei. He's working in Southwest University, and he is gonna talk about attentional control of inhibition in test anxiety. Please, you can start, Huawei. Oh, could you share your screen, please? Great. Okay, I think we have some technical issues now. Just give us a second to continue.
Okay. Can you hear me?
Great. Sorry for the technical issues.
Oh.
Yeah, could you share your screen? Thank you.
Okay, let's go.
So you can share your screen. Perfect.
Yeah.
Sorry, guys. Go ahead.
Okay. And I will present my work about anxiety, tested anxiety and inhibition. And the topic is enhanced or improved the recruitment of top-down attentional control of inhibition in tacit anxiety. And my name is Huawei from Southeast University, China. As we all know, tacit anxiety refers to a situation specific of anxiety with intrusive anxiety-related cognitive behavior and affective characteristics elicited by testing stimulus in academic or evaluation setting. Recent estimates have suggested that between 50 and 22% of students admit high levels of tacit anxiety. An attention control theory posed that tacit anxiety impairs inhibition and a key element of the central executive function. Meanwhile, a large number of experimental studies have supported this hypothesis. But presently, the literature is divided as it is unclear to whether tacit anxiety is associated with enhanced or improved recruitment of top-down attentional control of individual inhibition. The first account is proposed by the processing efficiency theory and the attention control theory. They both conclude that recruitment of top-down attentional control of individual inhibition. They focused on treated anxiety, use go-no-go, or scrub tasks. They found that compared to the low anxiety individuals, high anxiety individuals will have a large enter amplified and strong ALPF activity. However, the sector account, which is not really tied to a space-lived theory, they provide experimental support for the idea of improved recruitment of top-down attention control of individual inhibition. One should realize that a set count does not entail just one, but a multitude. They have two wheels. The first wheel is presented by the base shape. They use a task and use fMRI to find that anxiety is correlated with and improves the LPF activity in experimental trials when provided tasks improve low demand on individual attention. Experimental support was found for the first wheel, but experimental support should be considered limited in the sense that support is only provided setting characterized by low but not high demand on attention control. And just you see the task is very simple. But our study context will deal with tacit anxiety and focus on education or everyday setting is characterized by high demand on attention control. A fundamentally different and least the key element, namely the extent of the demand on attention control. Additionally, the second will support idea with improved control of top-down attention control individual inhibition, furnished in experimental settings characterized by high demand on attention control. In this view, it is affected that high individuals failed to compensate for impaired efficiency through enhanced effort or use of attention control resource as all task demand are in overload. High tacit anxiety individuals tend to motivate to recruit greater attention control resource to compensate for the impaired efficiency. However, when the demand on attention control are in overload, it will become impossible for the high individual tacit anxiety individual to compensate for the impaired efficiency through enhanced effort or use of attention control resource. So, how enhanced or improved the recruitment of top-down attention control inhibition in anxiety? How can we solve the problem? In our study, we use a flag task to measure the inhibition and to confront individuals with either a threatening or no threatening conditions, and quarterly control the amount of attention control results that can be used to perform a given inhibition task. Thus, in a flag task, the individual performs either a low or high working memory task. In our study, the low working memory task have 1 liter, and the high working memory load task have 6 liters. And the ERP converter N2 was used to measure the processing efficiency of cognitive effect in the study. Our hypothesis is that in the technical level, we expect the following data pattern to emerge. Unlike low test anxiety individuals, the high test anxiety individual record greater control resources and make more effort to meeting task performance. A conclusion is supported by high test anxiety individual large into and imply in the following experimental no test resistant threatening and in congruent trials both combined, but in contrast to low test anxiety individuals, high test anxiety individuals will display improved requirement of top-down alternative control results. A conclusion is supported by high test anxiety individuals have smaller N2 especially in the following experimental conditions: tester, retail threatening, high working memory load, in congruent trials, all combined. The logic is that in a high working memory load and there less attention control resource can be used for the individual to perform the flag task, and from the low threatening condition to a high threatening condition, the attention control resource can be used is small. So, and the 100 participants, 50 high test anxiety individuals and 50 low test anxiety individuals were used in this study, and the dual task design were used. And the data collection and analysis. Data was
sorry, we have 3 minutes left.
Used the NeuroScan amplifiers and processing using each lag, and the N2 amplifiers and FCD actually was used for further analysis. And we just see the figure in a no threat and low working memory load, high test anxiety individuals have a high large N2 amplifiers than the low test anxiety individuals. And in the no threat and high working memory load, they show the similar results, but in the threat and the low working memory load, there was no difference for high and low test anxiety difference for the N2 amplifiers. In the threat and the high working memory load, high test anxiety individuals have a smaller N2 amplifier than the low test anxiety individuals for the especially for the incongruent trial.
Okay.
And conclusion, in some of the present our study demonstrated that high test anxiety individuals can inhibit top-down attention control resource when the available resources for attention control are not limited. However, when the available attention control resources are limited and the experimental task is in overload, high test anxiety individuals typically fail to efficiently enhance the.
Okay, sorry to interrupt you. I think it's we have to move on to the next speaker. So please keep your questions to Huawei. Thank you very much. So our next speaker is Dr. Jin-Ting Lin. She's from New York University. Please.
Yeah, you're welcome. Could you stop sharing your screen?
Okay, wait a second.
Perfect.
Can you see the screen right now?
Yeah, we can see perfect. So
okay, I'll just start. Is it showing up fine?
Yeah. Thanks. Yeah, go ahead, please. Thank you.
So thank you for the introduction. I am Jin-Ting Lin. I am a research assistant at New York University, Abu Dhabi. And today I'm going to talk about a study which looks at whether we can reallocate resources within working memory. So in working memory, we can hold a few items for a few seconds. And in a typical working memory task, we often ask people to remember a few items. And after a delay, we often ask people to report one of the colors from their memory. Importantly, during this delay period, if we show a retro cue telling participants which item will be relevant to the memory task, this will improve memory for that item. And this shows that we can prioritize information in working memory based on our current goals. So why is cueing an item beneficial to memory? So there are several possible explanations for this. One of them is that this is due to reallocation of memory resources. So according to this account, retrocueing an item will allow for removal of other items from memory, and this will free up memory resources that can be transferred to the relevant items. So most studies have tested this by asking whether there are costs in testing the non-cued items, because the reallocation account would predict very poor memory for these items. However, there is currently mixed evidence to whether such costs actually exist. So is this benefit really due to reallocation of memory resources? So another way of testing this is to ask whether the size of this retrocue benefit will depend on the amount of resources that can be freed up from the uncued items. So the way that we test that is to have people remember few items of varying reward values, so they can either be high, median, or low. And so this is in order to create an unequal distribution of resources among these items. So the basic idea is that more resources will be devoted to the items with higher reward. And we're interested in looking at what happens when one of these items becomes irrelevant. So if we assume that resources can be reallocated from the uncued items to the cued items, we would predict that dropping a higher reward item from memory would free up more memory resources, thus leading to a larger benefit in memory. So we tested this in our experiments by having people to remember 3 colors. Then we cued them to remember 2 of these items. And then we showed a memory probe by, so basically we asked people to report the color by selecting the value on the continuous wheel. So we have 2 key manipulations for this study. The first is that we manipulated the reward values of items. So basically we offer bonus payment to participants, and the amount of bonus people earn will depend both on people's performance and the reward values of these items. So these values could either be high, median, or low, and it's based on the location. And these reward location associations are kept constant throughout the entire experiment. So the high reward item is 100 times more valuable than the low reward item, and the median item is 10 times more valuable than the low item. So there's a huge difference in the size of reward because we wanted to encourage people to use these reward values as much as possible. Second, we also vary the type of cue presented to participants. So the cue could either be neutral, meaning that people have to hold all 3 items in memory. However, the valid queue condition will have 2 green arrows pointing towards 2 relevant items. So people will only be tested on the queued items and never on the unqueued items. So this means that people can hold only 2 items and drop the unqueued item from memory. And this unqueued item can either be high, median, or low reward value. And this will allow us to assess whether the size of the queue and benefit depend on the amount of resources that can be freed up. But before we look at that, we first check if our manipulations of reward values as well as queues are effective. So here we show a plot of response error. So basically, lower errors mean better memory. And on the x-axis, we show the reward values of items. So we see a linear relationship. There's better memory for higher reward items. And do people also use the queues? So we found that in general, there's better memory when people can drop one of these items from memory versus when they need to remember all 3 items. So this suggests that people do use both the queue and reward. So the more interesting question is whether the benefit of the valid queue comes from reallocation of resources. So remember, in the valid condition, people can drop an unqueued item, but does it matter if the one people have to drop is the higher or lower reward item? So if we assume that this benefit comes from reallocation of resources, then we would predict that dropping a higher reward item would lead to a larger benefit in memory. So, but on contrary to these predictions, we did not find a difference between these conditions. So dropping a higher reward item does not benefit performance more. And Bayesian analysis also showed evidence in favor of the null effect. So these results are actually a little bit surprising and are inconsistent with a dominant view that the queue of the benefit from the valid queue is due to transfer of resources. But before we trust these findings, we further follow up to see if there are other potential causes for this pattern of data. So one of them is that perhaps we found these results, the null effects, because there are differences in strategy in how people use the reward and queues. So it's possible that there's a differential effect in dropping the higher versus the lower reward item, but maybe we can only observe this difference when people are actually using the queues and reward. So to test this, we try to single out participants who used the queues and rewards by using bootstrapped confidence intervals. So basically, we resampled all available trials in a given condition numerous times, and we used the distribution of resampled means to estimate which participants actually use the queues and reward. So this leaves us with 29 participants who used both queues and rewards. And however, as we can see from the plot also showing the response error, when we performed the same analysis, there is no difference in dropping the higher and lower reward. So this suggests that our result is not due to participants not using the queues and reward. So another potential explanation for these results is that—
Sorry, we have 3 minutes more.
Okay, right. Perhaps this is due to a very tiny effect of this reflects a very tiny effect of reallocation. So we did our study based on the assumption that there's a difference in the amount of resources for high versus low reward items. But what is the difference between the amount of resources that can be dedicated to high versus low reward items? So the difference can either be small or large. So in the case of small difference, we would predict a slight increase in benefit when dropping the higher reward item, even if reallocation of resources were to occur. On the other hand, if there is a large difference, the reallocation would predict a sizable benefit. So which of these would be the case in our study? So one way to test that is to see if the benefit scales with the amount of resources that could be freed up. So to do that, we try to estimate what is the difference, what is the proportion of resources that is allocated to higher versus lower reward items in our study. And then we try to take from our data, was it the benefit of dropping the lower reward items? And then we can make predictions from our data to see what is the benefit of dropping the higher reward item. So the basic idea is that we can take from our data, the green bar, what is the benefit of dropping the lower reward item, and multiply by how much resources are we allocating to the higher reward item, and predict, according to the reallocation account, what would be the size of the benefit from dropping the higher reward item. Then we can compare these predictions to what we actually found to assess whether our findings actually reflect a tiny effect of reallocation or null results. So to estimate the resources allocated to these conditions, we fit data on an individual basis to standard mixture model. So basically, this is trying to compare, so we use this parameters to try to compare how much more likely are we to retain the high versus low reward item, as well as to try to test, find from these parameters what's the precision ratio of the high versus lower reward item. So this is done in order to approximate how much more resources do we allocate to the higher reward item. And now we can compare this benefit to what we find in their study. So first we took the retro Q benefit when dropping the lower reward item. And so to predict from the reallocation account, what would be the benefit of dropping the higher reward item, we can just multiply by the reward resources ratio we estimate from the model. And then we can compare to our actual data. So basically we found that the predicted benefit is actually marginally higher than the actual benefit of dropping the higher reward item. So this suggests that the null results are unlikely to reflect a tiny effect. Just to conclude, so we found no evidence that the size of the benefit depends on the amount of resources that is being freed up. So this raises question about the dominant view that the retro Q benefit comes from reallocation of resources. So this suggests that this is a benefit that can be independent from the memory strength. So how much resources we dedicate to the unqueued items. So this might be consistent with other explanations that doesn't involve weakening of the unqueued item, such as to strengthen or to protect the queued items from interference.
Great. Thank you, Dr. Jean. So we have a question in the Q&A section.
Let me see it.
Okay, you wanna go on, please? So try to keep it brief. We have just 1 minute for the question. Sorry for.
Okay.
Okay. Is it all right?
So where can I see it?
Okay. Would you okay if we move on and you just give an answer in the Q&A section? Is it okay?
Okay.
Thank you very much. So our next speaker is Soledad Pico. She is from University of Buenos Aires.
Okay.
And she's gonna talk about emotional memories. Please go on.
Hi, how are you? I'm gonna share my screen.
Yeah, that's perfect.
Wait a second.
Yeah, right below. There is the.
So you're gonna see my share screen?
Yes.
So I'm gonna take about reconsolidation and just a little quick introduction, so we don't have much time. So a brief introduction for those who are not familiar with these terms. When you acquire a new memory, it is unstable or labile, and after a period of time, this memory is stabilized through the process called consolidation. By a presentation of a reminder, this memory can be reactivated
and go back to an unstable or labile stage. And after a period of time, this memory can restabilize through a process known reconsolidation, which allows the memory to update in strength or content. And amnesic agents can act over the unstable memory and interfere with this process. So in laboratory, threat conditioning has been taken as the paradigm to assess fear memory and anxiety disorder related memories. And so this process of reconsolidation has been proposed to allow that would allow modify this maladaptive memories. So in previous works in our lab, we designed a threat conditioning in humans and some tasks to assess the cognitive system associated with that memory. So in the study I'm going to show you today, we want to step forward and we want to see the effect of interfering the restabilization of this type of memory. So we designed this 3-day, 3 groups protocol, where on day 1, subjects perform a threat conditioning training session. On day 2, they had a reminder or not, and they perform a high working memory task or low working memory task. And on day 3, participants did an extinction and restatement session, that is the classical testing the retention of this threat conditioning memory. And also we added some of the tasks that we used to assess the cognitive bias of that memory. So going deeper on the protocol, our threat conditioning consists on 3 male faces. 2 are angry expression, and 1 has neutral expression. And one of the angry faces, and this is counterbalance between participants, is paired with an unpleasant tone. We call the face that is paired with the tone as CS+, and the angry face that has no tone as CS-. And the measures we take are skin conductance response and US expectancy in an external keypad, like a yes-no response, whether the sound is coming or not. So on day 2, for the reminder session, they only see 1 presentation of the CS+ in absence of the tone, and the task get interrupted by an interruption sign displayed on the screen. On the day 3, it's just a classical extinction and followed by a restatement. And for the interference task, in order to overload the transient memory system that has been reactivated by the reminder on day 2, we perform this spaced auditory serial addition test called PASAD that consists in a pre-recorded audio that has a number series from 1 to 9. And participants are asked to add systematically the last 2 numbers they hear. For example, if they hear 3, 2, they have to say 5, then 9, so 11, then 8, so 17. And we measure skin conductance level and proportion of correct responses. We call this task high demanding working memory task or just high working memory task. And there's another group that perform the low demanding working memory task, or as we like to call it, fake PASAD. And here, this group here, the same pre-recorded audio, the one before, but instead of adding, they just have to say whether the numbers are even or odd when they hear. So what we get as results in this study, 67 participants were included. We assess and find that there was no any previous psychopathology. And we also assess working memory capacity, and we found no difference in between groups. And when we go to analyze the interference task, this auditory addition task, we confirm the cognitive demanding nature of the task, as we see both of the high working memories groups, the one with the reactivation and the no reactivation session, both exhibit higher arousal on the skin conductance level
and worse performance when we see the proportional occurrence response in respect of the low working memory task. And when we go to see the implicit memory for all the three groups, the reactivation high working memory, no reactivated high working memory, and reactivated low working memory, through the 3 days, we have the trials of acquisitions, the reminder on second day, and on the third day, the extinction and restatement for all the 3 stimulus. We see that the 3 groups acquire the conditioning, the threat conditioning to the face and the tone. But what we found that the group that we have reactivated, so we labelized that memory, and after that, they perform the high working memory task. We see that that memory retention has faded. Here you can see it on the first trial of extinction. So we interfere that memory restabilization on the implicit memory. When we see the declarative memory, as we expected, there was no difference found. And then when we did the cognitive task, I'm just going to show you 2, then you can see our publications to read more about it. I'm just going to be sure about this. One of the tasks is stimuli representation, when there are shown 12 faces, one after the other, some aversive, some neutral, and we ask the participant just to rank them from 0 to 8, how unpleasant or aversive are these faces to them. And the 3 faces that we use for the conditioning are mixed up in these faces. And they rank it on day 1 before doing the threat conditioning and in day 3. So we can take the difference between those 2 days. And the other task they perform is an evaluation task consisting in a probability and cost section that include the faces from the CS plus and the CS minus, and they are involved in positive and negative scenarios, just hypothetical scenarios from every day, and they have to rank these scenarios from 0 to 8. And they have different questions, like for example, how likely and unlikely it will be, for example, having problems at work with him, and they have to rank from 0 to 8. And in cost, they have how good, bad it will be, and an hypothetical scenario. So when we go to see these results, we see that the reactivation high working memory group, the one that we have interfered the implicit memory, show no difference between the CS plus and the CS minus, but in the other 2 groups that we did not interfere, we see that they rank the CS plus as more aversive or unpleasant. And we see a similar effect when we analyze the evaluation results. There's no difference for the interference group between the CS plus and minus for positive and negative scenarios. And for non-reactivated high working memory and reactivated low working memory, we see that the CS plus is estimated higher probability and higher cost for negative scenarios. So in conclusion, in this work, we analyzed what happened when we disrupt a threat conditioning memory reconsolidation in the implicit, declarative, and cognitive bias using a working memory task. We demonstrate that the highly working memory task interfere the memory restabilization. And we also saw that the effect depends on the cognitive load or cognitive demand, because we could interfere with a high working memory, but not with the low working memory task. Also, we see that some of the cognitive processes were faded. And we hope our results open new venues to develop reconsolidation-based interventions in clinical settings, such as anxiety disorders. And I want to thank you all for listening, and the host and my supervisions, María Eugenia Pedreira and Rodrigo Fernández, and all my lab mates from Laboratorio de Neurociencias de la Memoria. You can contact us for more questions over here. and just additional information if you want to know a little bit more of what I've been talking about today.
Great. Thank you very much, Soledad.
You're welcome.
Yeah, is there any question for her? We have also one open
Sorry,
question.
I make it quick. I thought we didn't have any time.
We have some time. Okay. Okay, it was answer the question to Jorge. Is there any question for any of our panelists? Okay, if this is not the case, I just wanna ask you, Soledad, did you include women and men faces, or were more like
No, we only include men faces.
Okay.
Because on previous studies that all the labs did using these faces that are from the Karolinska database of faces, and they show that men faces were like presented similar skin conductance response, whether it was women or men. And there are several studies that women present a differential skin conductance in women and men, and it depends in a lot of physiological aspect related to that woman face. So we just keep the standard protocol that it is used for Fred conditioning. But we could use just square, triangles, and circles, and it will be the same. It's just the face allows you to ask a lot of these cognitive questions, like you cannot ask somebody if they like more or not a square or circle. Just that's why we choose faces.
Okay, I see. Yeah. So are you considering using another type of stimuli, or you want to continue with the faces?
We're gonna continue on this because this opened to us a lot of new questions.
I see.
So we're gonna to keep on that track.
Okay. Thank you. Amazing.
Thank you very much.
Yeah, so we are almost finished this session. I just want to thank to the panelists. Sorry for the technical issues we have today, but yeah, please feel free to contact our speakers, and thank you for your time. So see you next time. Bye.
Thank you.
I'm actually share my screen.
All right. Hi, everyone. So I think it's time to start. My name is Katya, and I'm a postdoctoral researcher in Yves Marder Lab at Brandeis University. So I will be moderating this session today together with Gori. And our session today is devoted to the research related to neuroscience of sleep. And hopefully we're going to learn more about how sleep is related to memory consolidation and cognitive functions and a lot of interesting things. So all the talks are going to be interactive talks. So I encourage everyone to please ask questions so that we have a discussion at the end of each speaker's talk. So let me stop sharing the screen. And let me introduce to you our first speaker, Yuri Pavlov. He is a postdoctoral researcher at Boris's Kochubey lab at University of Tübingen. What Yuri is going to present is, I think, very fortunate to us because he's going to provide some evidence that sleep does not facilitate human fear memory consolidation. Also, Yuri shared a preprint with us of a recently accepted paper on the analysis of neural activity during sleep in individuals with severe consciousness disorders. So if you want to learn more, the link is going to be available in the chat. And with that, Yuri, you can share your screen and you can start.
Okay, thank you for the nice introduction. So I'm going to talk about study recently completed where we looked at the central mechanisms of fear memory consolidation and how it relates to sleep. I think most of us know that acquisition of fear memories can lead to chronic mental disorders such as anxiety disorder or post-traumatic stress disorder. And also there is a vast amount of literature showing that sleep actually promotes memory consolidation. But what we don't know, we haven't looked much at the central mechanisms of human fear memory consolidation. I mean, there are some studies using skin conductance and other peripheral measures, but what's happening in the brain is kind of mystery. And we try to address this gap in the following study. The participants came to the lab 3 times, and on one of the day 2 or 3, they participated in a session of fear conditioning, then slept for about 2 hours, and then there was another session of fear conditioning where we paired neutral sounds with an unconditioned stimulus, which was a loud noise. And on the other day, everything was the same except for the intervention part, where the participants were watching silent videos and we controlled that they didn't fall asleep. While all of that was happening, and after each block of fear conditioning, we asked participants to assess the valence and arousal of the sounds they just heard. We also recorded EEG continuously throughout the whole session, and we also recorded finger photoplethysmography during the conditioning sessions before and after the intervention. Let's look at the behavioral results. Here's the valence scores before and after the intervention. Here's the sleep and the control condition, the period of wakefulness, 0, 50, and 100% reinforcement rate, meaning that in the 0% reinforcement rate block, there was no pairing of the conditioned stimulus with any unconditioned stimulus. There was a sequence of neutral sounds and nothing else. We see that the valence scores at the beginning are the same, they're not different. And in the 50% reinforcement rate block, the conditioned stimulus started being paired with unconditioned stimulus, which was the loud noise, or in the case of CS minus, it was just another sound, mildly pleasant sound. We see that here is a significant difference in this 50% reinforcement rate block, and the difference maintained in the last block where the CS and US similar were paired in 100% of cases. After the intervention, the picture was pretty similar, and we didn't see any difference between the sleep and wakefulness condition. But what is more important is EEG that we recorded throughout the session, and we looked at the event-related potentials in response to the neutral, the beginning sounds. CS plus here is the sound, there's 0.0 when it was presented, and CS minus is another sound which wasn't paired with unpleasant event. What we found when we averaged all the trials, and there were quite plenty, we found that there is no specific ERP component that can differentiate CS plus and CS minus. There is rather a slow wave occupying all this time window starting from about 220 milliseconds. There is basically a slow wave similar to contingency negative variance CNV slow wave starting at this time point. And we looked at this slow wave, how it develops and how it changes in all our conditions. What we found first, that at the beginning there was no difference between the CS plus and CS minus. Good, but then the difference became significant, and if we average all these conditions, we see quite strong effect of contingency, meaning that the slow wave to CS plus and CS minus was significantly different, and the slow wave to CS plus was larger or more negative, if you like. Here's the same data, but in bar plots. You can see there's no difference at the beginning, and actually there was no difference after the intervention and no significant interaction between the type of intervention and the pre-post comparison. It means that there is no difference between the conditions, between the condition when you spend 2 hours asleep or being awake. But what we found in both of these conditions, that the slow wave became stronger after a period of time. So this reconsolidation of fear, or relearning of fear rather, makes the response stronger. Even though in this first block, it was completely extinguished. And last, we looked at the cardiovascular responses. Here I'm presenting just one of them, HRV and heart rate variability. We couldn't actually look at the event-related changes in heart rate variability because it would require much longer interstimulus intervals, but we could look at the heart rate variability in all these 3 blocks with different reinforcement rate. Surprisingly, there was no difference between them because, yeah, we would expect it that heart rate or heart rate variability would change when you present unconditioned stimulus, this unpleasant stimuli, but it didn't happen. But what happened instead, after the intervention, there was a significant increase in heart rate variability, especially after the wakefulness condition. There was a significant interaction between this pre-post comparison in type of intervention. Why it matters? I think it is an interesting observation. I'm not sure whether we will replicate it, but it may be interested in the context of this kind of works where people look at the heart rate variability and relationship of heart rhythmability with emotions, emotional response. And fear, learning fear is also an emotion. We saw, basically what we saw, that if you increase somehow heart rate variability, these changes in heart rate variability doesn't affect fear learning. Okay, let's go to the conclusions. What we found that this long-term conditioning is equally efficient after a period of sleep or wakefulness, and relearning affair increase the slow wave amplitude, but actually we didn't see it on the behavioral level. Conditioning with 50 and 100% reinforcement rate is equally efficient. There's no difference on all three levels. And increase in heart rate variability does not affect peer learning. Thanks for your attention. Let's discuss it.
Okay, thank you very much, Yuri, for a very insightful talk. And it was very interesting to learn about this selective memory consolidation during sleep. So while people are coming up with questions in Q&A panel, I actually have a question myself. So as far as I understood, you presented either neutral stimulus to the participants or unpleasant and negative stimulus and observed the increase in the slow wave amplitude. Have you tried presenting them with some kind of positive or some kind of pleasant stimulus to see how the EEG activity changes during sleep?
What do you mean? We looked at the changes in EEG in this associative learning paradigm in response to the stimulus, which was at the beginning neutral, but became significant. So it's like the definition of classical conditioning. Right? No, the neutral sound was always neutral, but it was paired with something unpleasant. If you find a stimulus which is pleasant and we can use in our experiments, I will thank you for that. So, but it is extremely difficult in human classical conditioning. You cannot give a person like 60 pieces of chocolate, or because in order to record EEG and get this earpiece, you need many trials, and that's the problem. Mm-hmm. So now we are looking at the ways how we can actually try to approach this problem, and the only approach we have now is to try to use smells, some pleasant smells, mm-hmm. which is also technically very challenging, but I hope it is possible. Anything else is not either habituates really quickly, like this chocolate or something like that, or mm-hmm. It's just not pleasant enough. No. There is also a way, like a relief, pain relief is also pleasant, but yeah, we don't gonna.
And you don't want to
go in this direction.
Right.
Right. I have a brief question.
Mm-hmm.
Like your participants, they slept during 2 hours, right?
Yeah, they had a sleep opportunity. So
yeah. Okay. Yes, I was about to ask if the onset of the different sleep stages was similar because like all of them went through conditioning, right? You don't have participants that didn't get to be okay. Yeah, right. Yeah, nice to see those results.
Okay. I think it's actually about the time to move on. So thank you very much, Yuri, again. And
thank you.
Find Yuri's Twitter account in the chat if you want to learn more about his research. And okay, so our next speaker is Viviana Greco. She is a PhD student in Penelope Lewis Lab in Cardiff's University Brain Research and Imaging Center. and she will tell us why it is important to wear a light blocking eye mask during sleep. So please, Viviana, go ahead.
Yes. Can you see the slides?
Yes.
Okay. So hello, everyone, and thank you for the presentation. So I really hope that at the end of this talk, all of you might decide to wear an eye mask during sleep. So first of all, why we decided to carry on this study? Because there are several papers, and in particular the one that I have highlighted below, that look at the effects of the early light exposure on sleep architecture. They actually show that the early light exposure is going to affect the time spent on REM sleep, is also going to affect the amount of wake that people have during sleep, and also the slow wave sleep time. There are also studies that look at the beneficial effects of wearing an eye mask during the night, but all of that have been done in patients in the intensive care units. But what they've shown overall is that all the aspects of sleep quality were significantly improved. So what we hypothesize is that based on the fact that in Cardiff area, the sunrise time between the end of June and the end of September is between 5 a.m. and 7 a.m. And based on the fact that most of the flats have no shutters, we decided to see if the use of an eye mask during sleep might have improved sleep quality, memory consolidation, and alertness, and might also have been beneficial in terms of sleep architecture. So overall, we tested 95 participants between 18 and 35 years old, and we tested them over the summer from the end of June till the end of September in a within-subject design, in which, in a counterbalanced order, the ambient light was blocked or not blocked during sleep. So, as I said before, in a counterbalance order, we asked people to wear an eye mask or a no mask for 2 weeks. And we had 5 habituation nights, followed by 2 consecutive testing days, in which we tested people in the morning on 3 different cognitive tasks: the paired associate learning, the psychomotor vigilance task, and the motor skill learning. And at the end of day 7, so at the end of the second testing day, we asked people to wear a dream headband during the night. So what about our behavioral results? Related to the paired associate learning, in which we ask people to learn 80 different word pairs, we have surprisingly seen that the learning performance related to all the corrected word pairs was significantly increased in the eye mask compared to the no mask. So in this case, when wearing the mask, the encoding performance was higher when wearing the mask. Related to the psychomotor vigilance task, looking at the first testing days, we have seen no difference between the mask or the no mask in terms of reaction times. Instead, when looking at the second testing days, we have seen that when wearing the mask, the reaction times were significantly faster compared to the no mask. At the end, for the motor skill learning, in which we ask people to reproduce with their non-dominant hand a 5 fixed digit sequence, we've seen that the use of the mask didn't have any kind of impact on this kind of task, but anyway, the overnight consolidation was preserved for both the mask and the no mask, as previously demonstrated. About the sleep architecture, in order to sleep architecture, we use the dream headband as the one that is reported below, that has 4 electrodes on the front and 2 electrodes at the back. And I want to say that these results are just preliminary results and are still under analysis. So, but anyway, what we have seen is that when wearing the eye mask, the amount of time spent in stage non-REM 2 and in REM sleep was higher compared to the no mask. And we didn't find any correlation with the behavioral results. For the sleep quality, we assessed the Stanford Sleepiness Scale that is going to look at the morning alertness. And for the Stanford Deepness Scale, the higher number are related to people that feel less alert and vital in the morning. And in this case, we've seen that when wearing the mask, people felt more vital and more alert in the morning. So what we can say overall is that blocking the ambient light with the use of an eye mask is going to have a positive impact in terms of encoding on declarative memory, as demonstrated by the paired associate learning. It's also going to have a beneficial effects in terms of vigilance, even if the PVT is not supposed to have any learning effect. So actually it would be good to receive some input from you. And the eye mask doesn't impact on procedural memories. And there is a beneficial effect in terms of alertness. And at the end, it might have an impact on sleep architecture. I want to highlight might again. And at the end, thank you to all my lab.
Okay, thank you so much, Viviana. I think after your talk, I'm pretty convinced that it's very beneficial to wear an eye mask, especially during the summertime. So let's see. We actually have a question from Ksenia Kirillova. Does having blinds give similar effects to wearing a mask?
Well, I don't know actually. I don't think so, but yeah, I don't really know. I'm sorry.
I think the question is.
Oh, sorry, having blinds. Oh, yeah. Sorry. Okay.
The effect of blocking the light. It is.
Yeah. So the point of the study was really to see what is going to happen without blinds, in particular because in Cardiff people don't have curtains. So I think that the results will be different in this case. Yeah. So it does matter. Yeah.
Okay. Thank you.
I have one question as well.
Yes, please.
Okay, because at some point you mentioned like how much time they spent on REM and non-REM sleep. So I was wondering if there's a difference on the onset time of REM sleep, let's say, or did you check for that? Like if they get to REM faster or not, depending on the mask.
Yeah, so with the dream headband, we can only see the difference in the sleep stages. And so I only have the overall time spent in each sleep stages.
You cannot know when it started. Yeah. Okay.
Yeah.
Okay. Right. Yeah. Thanks.
You're welcome.
Okay. And I have actually a few questions myself. So I was wondering if you have any hypotheses, because it seems like you observe some differences in the memory-related task, right, but not motor-related tasks. So do you have any hypotheses when you think you see the improvement in the memory-related task, but not motor-related tasks?
Yeah. So based on the fact that we have seen, at least with the dream headband, that there is an increased time spent in stage N2 that usually has been related with motor performance. It was a bit weird at the beginning because we thought that we would have had some kind of impact of the mask. But at the same time, the overnight consolidation was preserved, even if there was no particular effect of the mask. So yeah, we are currently replicating these results to see why this might be the case.
Okay, very interesting. And I was wondering if you observed any gender differences between males and females, the effects of wearing an eye mask, or it's uniform?
Yeah, no, it's the same. We didn't see many kind of differences.
Okay. Very interesting. Okay, we don't have any more questions. So I guess last question from me is, what is the future directions? What are you planning with the next steps for your research?
Yeah. So at the moment, this study has already been replicated and we are analyzing the results. So we replicated it over the summer. And so we're waiting to finish and to see if, what are the results of the replication and if we can confirm what we have seen before.
Okay. Well, if there are no further questions, I think we will move on to the next speaker. And so our last speaker of the session is João Patriota. He is a PhD candidate in Cyril Pinard's lab at the University of Amsterdam. And besides research, he is also enthusiastic about film photography and vinyl records. So you can reach out to him on Twitter and not only discuss research, but also some analog things if you also share the same passion. And today, João is going to talk to us about how to precisely detect and target theta phases for closed loop simulations. So with that, please, João, go ahead.
Yes, sorry. Thanks a lot for the introduction. So good evening for my time zone, but yes, you adapt to yours. All right, so basically my talk's called closed loop acoustic neurosimulation targeting theta waves. All right, so on the literature, we have been talking about since 2006, kind of about target memory activation. What does that mean? That during sleep, you can interact with whatever oscillation is happening there, and this can bring you benefits on tasks, mostly in the next day, right? So there's a lot of literature about that, but mainly on non-REM sleep. So this is based on the assumption that whatever is happening during non-REM sleep, this can be associated to memory enhancement, so to say. But then this raises the question, okay, then what happens during REM sleep, right? And then we want to do, if non-REM sleep is able to enhance or diminish your ability on tasks, this can make you do memory manipulation. And then how does that happen with REM sleep, right? So there's some literature pointing towards the relation of REM sleep and emotions. There is also literature pointing towards the importance of theta oscillations that go from 4 to 8 hertz on REM sleep. And our main question is, okay, if we are able to interact with those waves happening during REM sleep, how can this translate to whatever the person is feeling or performing tasks on the next day? Yeah, so I'm going to talk about a few results of what we got in developing this method. So we got data from 9 participants that just went there to sleep, doing high HD EEG, so 64 electrodes, but mainly I was analyzing FZ, the frontal one. Yes, and then after I got the recordings, I used that to run several simulations as if it was being recorded in that moment, right? Because I was developing this method, so I had to play around with different parameters and think, okay, what are the characteristics of theta waves that are more prominent in this or that? So this was not done with the people there. So I got the data from their sleep, and then I did the simulations that got me to these results, and then I applied the release test for circular uniformity just to assure that I wasn't gathering phases on the waves randomly. So pretty much what happens is that if you look at the orange traces, this is the EEG unfiltered, how we get it real time. We do a nonlinear sinusoidal model on top of it on 6 hertz. And then every time this has a good fit, we think, okay, so probably this is going to be true for the next period. And then with that, it's possible for us to select a certain moment of this incoming signal, right? Because if it's as periodic as the sine wave we generated, we should be able to target it. And then, yeah, and then sometimes this happens, sometimes not, but then this is a good example of when it happens. So as you can see, so the blue line is just duplicated, right? It's a perfect sine wave, and then it kind of matches, but then it's the green line means the zero phase, which is our phase of interest. And like in this case, this shows this is true, right? It works. So if I look at it over all my participants, this is an example of just one. Red line on the polar plot means the average of targeted phases. So pretty much we are able, yes, to detect stuff that happens on the range of 4 to 8 hertz. And like just a small remark I didn't do in the beginning, so when people talk about non-REM sleep, they're looking at things that go from like 0.5 to 4 hertz. So they're really slow. And like if you want to predict what's going to happen in the next cycle, it was a technical background for us. And then, yeah, so with those results, it shows that yes, we can do that. But then, which raises the question is, okay, so what now, right? So like, what can we do? So there are some results from our lab that are in the way to be published. I mean, unfortunately, I cannot share the paper with you yet, but there are some relations, for instance, between how a person relates to have so a person sleeps, next morning it says, okay, my sleep was good, it was bad. And like usually when they have a fulfilling night of sleep, this is attached to the theta power that was collected during the night. So if we are able to interact with the theta waves, there are many, many questions that can be asked. And like pretty much I'm going to do that during my PhD. So yes, that's it. And then I'm not sure how much time I still have, but yes, I would like to thank Lucia Talamini and the Sleep Memory Group and Neuromatch for the opportunity.
All right, thank you so much, Joao. We actually have still a lot of time for the discussion.
Okay.
Yeah, so let's see. So while people are coming up with the questions, hopefully in the Q&A box. So I have a question regarding how long typically the theta episodes are. So in order for you to be able to, detect them and kind of then predict the theta phase and stimulate still within the theta phase, like theta rhythm episodes.
Yeah, so this is okay. So, I mean, usually theta happens like in bursts, right? It's not very constant during the whole sleep. So you can have, let's say, so it's from 4 to 8 hertz, right? So if you consider one cycle, 0.125 milliseconds. And then usually this happens like across 4 or 5 sleep epochs. So it's one epoch being 30 seconds. So it's 2 minutes, let's say. So this happens during REM sleep several times. So it's kind of tricky. It's kind of tricky to target it. And then like, we also don't know how it's going to interact with the stimulation per se, right? Because a closed loop auditory stimulation. So the idea is that every time we reach the zero phase, we play a sound. And like there, for non-REM sleep, there's some results that show that the slow oscillations, they get boosted. So they are more present during time. So we expect to see something like that for theta as well.
Okay, thank you.
Yep.
So now we actually have several questions from the Q&A chat. So Miguel is asking, have you assessed how the ERP of the stimulation can affect the model for predicting the next theta phases?
Okay.
I'm not sure if you said all of it correctly.
Right. So for that, I can have a control where I am just identifying the phases without stimulating. So I can have throughout the same session of sleep moments where I did a stimulation, and then I can do the ERP with all that. Then I can see how it looks like, and then I can compare that with when I detected in the same way, in the same person, but without any stimulation. So this would be the comparison.
Okay, thank you for the answer. We have another question from Karen. Do you think it would be interesting to compare behavioral results of REM stimulation locked to eye movements and presumably PGO waves versus stimulating theta in REM? given the different things to stimulate, I wonder if one will be the gold standard for enhancing REM, like we have for slow wave sleep. And for long one.
Yes, like this is like a wonderful question. Like, yes, yeah, I think it's interesting. I think it's interesting for sure. Like how those things are tied. It's there are some reports in the literature already that shows that, yeah, PGO waves, etc. So it's like, to be fair, yes, if you happen to do that, please let me know. Like, I'm really curious how that works. But yes, I do agree that it's a bit tough finding the gold standard, because for slow sleep's kind of given already. But yeah, that would be nice.
Okay, thank you. So far that's all the questions we have from the audience. I encourage you guys to ask more questions because we still have 10 minutes till the end of the session. Personally, I was wondering, why did you choose acoustic stimulation for your closed loop feedback, and or you're planning to use maybe for the electrical stimulations or some other types of simulations?
Like, no, the best thing about acoustic stimulation is that it's like everyone can perceive it, you know, like we do electrical stuff, it's like it's a bit harder. Yes, and also depending on the valence of your stimulation, you can expect some more interruption on the ongoing sleep, right? So like a sound is something that we all hear when we are sleeping, and it does not interrupt it as other stimulation might. So this is one of the reasons, is to make it more natural, so to say.
So has it been shown before that particularly acoustic stimulation during the REM sleep, you know, enhances the, or elongates the REM?
Yeah, there are a couple of papers on that, yes.
Okay.
Yeah.
I was wondering if there are going to be some delays induced by, you know, processing of the acoustic information versus, and there is going to be a delay in phase.
Yeah, exactly. No, this is something that it's kind of hard to disentangle, especially because we consider there's time for processing, right? And then like in this way, you cannot be sure. But like, yeah, I think it's more about if you target, let's say, the zero phase, and then we can assume how much time this can be processed, and then the sound's going to be processed, let's say, on the 90 degrees phase. So we can phrase our question like that.
Mm-hmm. Yeah, so it's beneficial to be able to stimulate at different phases and compare, I guess.
Yeah.
We have another question from Karen. Do you think it will influence dreams? If so, how?
Yes, I think this is a very good question. I mean, I'm far away from being able to completely answer it, but if we assume there's gonna be a boosting effect on REM, and this makes it last more, we can expect, yes, more dreaming episodes. Just basing ourselves on the fact that REM is the moment of sleep that there are more dreams. If it's going to affect it in terms of valence, like if it is going to induce pleasant dreams or unpleasant dreams, it's you know, I cannot tell you that. Like, I don't know. I simply don't know. I would expect something if we would have some indication that that the sound that I'll be playing on the up phases of theta waves could encode something for the person. But like, it's far away from me being able to tell you that because I didn't test for it yet. But like, yes, it's a possibility, I would say. So I just mentioned right in my conclusion that this paves ways for several questions. One of the questions is, how does this relate to dream reports? So we can just, yeah.
Yeah, that would be very interesting application also for this particular technique, if you could actually elongate the phase in the dreams.
Yeah.
Yeah. Okay, and we have one more question from Miguel. Any thoughts why the zero phase is better to enhance theta activity, or is this assumed from previous experiments for slow waves during NREM sleep?
Yes, this is a bit in line with what you asked before. So the fact that we choose zero phase is that, so there's a wave coming, right? And then it reaches zero phase, now it's going on an up phase, right? And then if I play the sound, this sound is gonna be played during the up phase. So the idea is not, it's not, not a dot, right? It's a continuous, a sound that contains some duration, and this is thought to comprise the up phase of the oscillation. And yes, this comes from previous experiments in ORM. People have shown that if you perform acoustic stimulation on the up phases, meaning you get the zero ascending, of course, and then you play the sound, your slow oscillations increase in amplitude. And if you do the opposite, if you play it on the down phases, they diminish, right? So the first question, of course, is, yes, is it the same thing? But yeah, also something to be tested. Many, many questions to be asked.
Mm-hmm. Okay. Yeah, very interesting. A lot of applications you have for closed-loop simulation. In general, it's a very popular technique nowadays. So
yeah, that's true.
Yeah. Okay. I think that's all questions that we have from audience. So we have, I guess, few more minutes before we finish. So if you guys have, we can just now have some more discussion and ask questions to any of the panelists if we didn't have time to ask questions for any of the panelists before. I actually have one more question for Yuri, which I didn't have time to ask, but maybe I missed what part of the brain was this slow waves recorded from that were amplified during conditioning?
It was a CZ channel. I show it, but it is. Yeah, normally if you look at the EEG, you can see this slow wave is generated somewhere around vertex.
Okay, so it's a common. I don't know much, but it's a common practice to record from this particular brain.
Yeah, we recorded 64 channels, but it is common practice to at least start your analysis with this single channel, because it is far from the sources of artifacts from muscles, and it is yeah, just a tradition, I'd say, because the first time when people recorded CNV, this slow wave was let's say 60 years ago. Yeah, so since then people just doing this. It is basically a sign of expectation, like you're expecting something pleasant to happen.
Okay. And did you by any chance observe any gender differences in the EEG recordings?
Gender?
Yeah.
Yeah. No, of course we didn't look at it, because the study was powered to look at the within subject differences. Not, yeah.
Oh,
it
okay.
would not make any sense to compare like 9 versus 9 participants. Yeah.
But within subjects it was uniform.
What do you mean?
I guess the changes in the amplitude.
So it depended on the conditions. I mean, yes. What do you mean by uniform? So it's changing in time and depending on the conditions, depending on the stimulus represented.
Right. No, what I meant is within the condition. The change in the same direction.
You mean if we compare the trials, yeah, it is not really easy thing to do, because we need to average lots of trials in order to see anything. So it is how we do the P analysis. Yeah, because we averaged like 60 trials. It is possible to look at like 30 and see something. Yeah, but the internal consistency is quite high, I should say.
Okay. Okay, well, thank you very much, everyone, all the participants. So we actually exactly at the time to end our session, and I learned a lot about sleep and about EEG. So hopefully, as everyone of our participants. So thanks again, and thanks.
It's 5 p.m. UTC, I guess we can start. Good afternoon, everyone. I'm Matteo Mancini from the University of Sussex, and today I'm sharing this very special session with very interesting talks. And the first one will be given by Tanya Brown from the Rothman Research Institute at Baycrest. And she will provide actually a different lens to view science through, and I really look forward to it. So Tanya?
Share my screen. Just sharing my screen. Can you let me know if can you see that okay? Mm-hmm. Yeah. Great. Great. So thank you everyone for joining us today. I'm Tanya Brown in Toronto, Canada, working at the Rotman Research Institute at Baycrest with Dr. Randy McIntosh as the program manager for the virtual brain. For those who are not familiar with TVB, it's an open source neuroinformatics platform for simulating large-scale brain dynamics. And TVB was built by a large international consortium of a multidisciplinary team, first released in 2012. And to date, we have more than 30,000 downloads. And as much as I'd love to tell you more about the impressive features and outcomes of TVB, I'm actually here today, or tonight for some of you perhaps, to talk to you about science management, specifically within the domain of neuroscience. And if you're not already convinced of the necessity for management in science, then certainly I aim to convert you into a believer. I'll also share with you some useful resources, some standards and best practices that could help guide you through your own research programs. So I'll begin with a more general focus on project management and then transition into more subdomains, including data management, knowledge translation, science communication, and people and performance issues. So I don't think you need me to tell you that science has changed a lot over the last 50 years. In fact, I'd say a lot over the last 5 years with the increase in the number of different hats that scientists need to wear. And there's an increasing amount of pressure on researchers in terms of funding and publications and collaborations, often not just within our own labs or institutions, but rather on a global scale. Perhaps some KT activities, also battling with data-related issues, always gaining knowledge from other areas that were previously not really even on our radar and beyond. And in 2018, Nature came out with a paper that reported that almost half the PIs that they surveyed declared that they did not have, but would like formal management training. About a quarter of them had some training and thought it was useful. And then there was another remaining proportion that didn't particularly have an interest or shared value in management training. But in fact, I've even received comments along the lines of management stifles science, or it kills the creative spirit of science. And to that, I'd say that then you're probably not doing it right, because science management really does have the power to elevate our practice to a new level when done correctly and in an intentional way. And here in Toronto, I consider myself very fortunate to be working with the team, albeit with Randy and the rest of the TVB team, as regarding this type of approach to science as not only valuable, but it treats it as a requisite to how we conduct our lab and just kind of the greater TVB initiative. And I'm also grateful for the NeuroMatch organizers who saw this topic as value for this community, and to those who decided to join us today. So I like this quote because it touches on the point that is often used as a reason for avoiding any type of planning, in that why make a plan if I'm not going to use it? But the nature of project management is not that anything is concrete, but rather it provides a structure to the way that we do our science that imparts a balance between exploration and production, with the built-in capacity to modify it as you go. So some examples of project management in science: I would typically be breaking down projects up into such things such as a study or experiment, or a grant application, or a presentation of some sort, a KT project, kind of your day-to-day administrative tasks, that counts too, managing a collaboration or organizing an event. And central to the formal project management is this project life cycle, and understanding how to carry out each stage here that's listed on the right with the greatest amount of efficacy can determine really the success of your project. And in looking at this list, I'd like to invite you to reflect on each and assess whether this is something that you always or often or sometimes or never do as you approach your work. My guess is that it's unlikely that you've typically ever really closed out a project or prepared a comprehensive project plan, as these are not typically expected or required, although in practice these things are critical for developing an effective workflow. But when we step back and consider how we measure success in science, I'd say that we converge on kind of three pillars, that being time, such as the turnaround in which, say, your study, in the time that it's done, and by the time you get to the end, is it still relevant? The cost, we do have a restricted amount of funding dollars, so we're expected to make deliveries on tight budgets, and scope of whether your study, your project, or your event achieved what you set out to do, or did you lose track somewhere along the way? And kind of with this trifecta of factors is when it's delivered at an appreciable level of quality, in turn, we can really witness the creation of scientific merit. So next, I could make an entire workshop about the various tools available for project management, but here are just a few of my absolute go-tos. The first being a project charter, and this is a master document that contains all the various pieces of planning and managing your documents that will support the project. And this can be tailored to the needs of each project. For instance, if you're working more independently, it's not likely that you're going to need a team management document, but almost always you'll need to have a work breakdown structure or a budget or a resource index. Next up is a Gantt chart. This is probably both your enemy and your friend, as it does require some upfront effort, but the ROI can be exceptional if executed in the right way. And because it's a great way to visualize and track your project, as well as identify problem areas and make adjustments as needed. And when it comes to project managed software, there's a plethora. Oh, sorry, I'll go back. There is lots of options out there, but my recommendation is twofold. It's to pick something that serves the problem you're trying to solve. Don't just use the next hot thing. For example, when Slack came out, everybody wanted to use it for something, and it can be great for team collaboration or communication or getting some sorts of work done, but it performs less well as kind of a project tracking method. I'm quite partial to a software platform called Trello, which does have a freemium version, which is typically sufficient, but it does have an extensive and useful plugin array that allows you to really customize each project board appropriate for the need of a particular project. The other is Git or GitHub for the more technical projects, because there's version control systems that allow you to manage or track your source code. And there's also a niche community within Git that has the potential to open up doors to even potential collaborations. And lastly, my word of advice on this is that there is no tool, at least not yet, that's not going to require your effort and attention to be effective. So pick a platform that A, you will use, B, your team will use if you're working with a team, and C, that it serves the appropriate function that you need it to. And maybe if we get to the Q&A, if anyone has some specific challenges where they're looking for specific resources, I'll be happy to deal with them then. But maybe moving into the next topic or subdomain would be data management, which is a major area of importance and consequence, but so often disregarded. More often than not, I think many researchers are in the mode of kind of making it up as they go and kind of like building a bridge. I like to say of like putting down the board as you're crossing the bridge, which is both an ineffective way to build a bridge, but similarly, it's hazardous when it comes to big, expensive, and time-consuming data. So research data is rapidly increasing in size, variety, and complexity. Big data does provide lots of opportunities and value, but it does not come without challenges, especially in health and medical research due to some of the regulatory requirements, which adds additional layers of complexity. Research data management is broken up into 3 phases, that being pre, during, and post-research. And with each section, there's a list of factors that you should become familiar with. So just to go into some of the more practical issues to be aware of when it comes to managing your data. First up is standards. So we have several in neuroscience, first being the FAIR principles, and leading some of this is INCF, where we find data that's findable, accessible, interoperable, and usable. There's also CARE, which is a global indigenous data alliance, as well as OCAP, which is something that we have here in Canada. And this is a set of standards that's established by the First Nations in terms of how data is collected, protected, used, and shared. Of course, we all know BIDs, hopefully, for neuroimaging data, and there are more. But a data management plan, the objective is to address the issues related to data management prior to starting your research project. It's a formal document which clearly articulates the strategies and tools you'll implement to effectively manage your data, and speaks to the management of data both during the active phase of your research as well as at the completion and beyond. And sometimes a data management is required even by funding bodies. This is an increasing trend that we're starting to see. And if it's not required, noting that you have a data management plan in your application can signal to reviewers that you have considered the details for your proposal. Data storage, this is a big one. I won't go into too many of the details, but just knowing that it's something that you should always be mindful of. Knowing the risks related to transferring data and making sure that you prepare accordingly before making any moves. Documentation, which is an absolute must, especially when it comes to code. And to be aware that various publication requirements are out there. For example, PLOS requires data to be open. Moving on to knowledge translation. I think for the purposes of today, I'm only going to be brief, touch on this topic, as it should be understood that even though as researchers that we may not explicitly be conducting KT activities, it's important to acknowledge the bigger system in which science is situated in. And with science being the vehicle to understanding our world better, more specifically in our instance, understanding the brain, it's commonly perceived that with each new finding, we are slowly chipping away, or rather adding to kind of this greater knowledge bubble. And with that, we must effectively capture new knowledge and ensure that it's appropriately lends to bridging this gap between what we know and what we do. So KT is effective when it's placed within an ecosystem of stakeholders. And you can kind of see some of the various players there on the left. And this is to make progress a reality, and it's our job as science managers to oversee this network of people to ensure that they all work in synergy. Having the ability to discern between the roles and incentives of this integrated network of science stakeholders is key, and who listens to who and what is convincing. So is it that we have the evidence, or do we need the expertise or stories? And all of these kind of play a role in being successful with knowledge translation, which I think also goes nicely to the next topic of science communication. And with that, I'd ask, why do I want to talk to people? So the obvious one is that you want to share your research, but also to find collaborators or funders, or perhaps recruit new lab managers. And who should you be talking to? Obviously your colleagues, those in neuroscience, but also those outside of your own discipline. a large focus of my work is to remove those barriers that keep us in our silos, whereby we can really be encouraged to have more open conversation and cultivate a dialogue in a more multidisciplinary setting. Also funders and investors, both secured and potential. And a big one also related to knowledge translation is the public. Having people care about your work not only makes it more meaningful, but it also has a downstream effect on policy and decisions. And also to be reminded that to take a step back and assess things, not just for what they are now, but a little bit more future focused. And when it comes to outreach, this is something that some of us begrudge.
Can I ask you to wrap up?
Sure. So I'll skip this one. I'll just say that this is people and performance management is also very important. And I'll skip that too. And Gantt charts, I mentioned them earlier, friend and enemy, but using them can be very useful. And ways forward, I would just like to say that developing your own science management toolkit as per your own needs is key. And if you can't do it, hire a lab manager. And if you're working on a larger research program or project, hire a project manager or program manager. And there are funding agencies that are starting to support these things. And I'm starting up a new group called the Science Management Collective, where we'll be working on such issues as this to really build out some standards and best practices within the field of science management. So thank you, and I'm happy to take any questions if we have time.
Thank you, Tanya. Let's try to have questions at the end, since we have just 2 speakers, so we keep a bit of timing here. So I will move to Lorena. Thanks again, Tanya, for the talk.
Pleasure.
So Lorena Clark is from York University, and she will talk about how art can support and encourage parents to spend more time with their babies. Lorena?
Hi. Here we go. Can you see my screen okay? Yeah? Okay. Okay, so my name is Lorena Clark. I'm really excited to have the opportunity to present my interdisciplinary MA thesis today. I'm at York University in Toronto. I'm part of Dr. Joseph D'Souza's lab. He's one of my supervisors, along with Dr. Upali Nanda and also Professor David Scadding. Whoop. So the title is I Listen to Your Voice, the Influence of Art and Design on Parental Stress, Experience, and Engagement in the Neonatal Intensive Care Unit. So can large-scale environmental graphics installed as art on walls in the NICU be used to lower stress, educate, and encourage parents to spend more time with their infants? So my thesis work focuses on 3 disciplines considered in interior design: art and architectural history and aesthetics, environmental psychology, and then graphic design and visual communication. And then these 3 disciplines led me to the why, and that's where we look to science to explain what's happening in our bodies and brains. And that's the link to neuroscience and Joe and his work. So again, just my 3 disciplines, I'm hoping to find better experiences and health outcomes where we collide. So given that I'm new to the field of neuroscience, I thought I'd provide some context on where I fit into this conversation. So I'm a part-time interdisciplinary MA student. I'm also an interior designer at Stantec in Toronto, and I specialize in healthcare design. And I've done a lot of work in pediatrics, and I've been doing it for about 15 years. So my undergrad was art and architectural history, and I minored in psych. I knew I wanted to be interior designer, so that was my foundation. Then I spent 3 years studying interior design after that. So then 20 years later, I'm returning to investigate just one idea to try to better understand the impact that we can have. So the photo on the left is our office in Toronto, and then the 2 photos in the middle are my most recent completed work at Sick Kids, a play area there. So the next few slides, I'm going to focus on the inspiration for this thesis idea. So back in 2012, we were doing a full renovation of the neonatal intensive care unit at Sick Kids Hospital. It had a real impact on me. We had big ideas to make it a much better space. So a few things we were focusing on is space for family integrated care, which is obviously most important in the NICU because these infants depend even more on family involvement and parental involvement than any other. Single rooms as opposed to the current wards, thinking about kangaroo care and skin to skin, and also the importance of breastfeeding and nursing and pumping, and also acoustics and privacy. With the following goals in mind: to regulate heart rate, better weight gain, reduce length of stay, decrease hospital readmission, and less stress for parents. So looking at going beyond what we typically can do to support the design, and how we can further support mothers and parents so that they have more to give to their babies. So they had created some art to educate and encourage parents. Here are a couple of examples. So Be My Voice and I Listen for Your Voice. And they were really lovely and they had a real impact on me. But I did wonder if they could be reinvented to have even more impact. So I was thinking about scale, location, color, image, and typography, and how I could reinvent them. So when I was writing my proposal to get into the program, this was a first stab at a very bold response, a really big photo of a baby, a powerful headline, a statistic to inform, and then a link to some support for parents. At that time, this was back in 2016, it was a QR code, but. So the next few slides are a few examples that inspired the idea and the design. So thinking about the impact of image and text and how does the viewer respond? Advertisers have perfected the art of persuasion, and how can some of those methods of visual communication be used in other ways, and in this case to help patients and families? These are a few examples of messages of encouragement in unexpected places in the built environment. So thinking about innovative ways to add impact and affect people's experiences. And a couple of these are in Toronto as well, and related to the CAMH campus, Center for Addiction and Mental Health. So this is what really made me realize that there's something powerful going on here. Looking at photos of babies. So when I was pregnant with my first baby, I had these strong feelings when I saw these photos in magazines, and I just wanted to pick up the babies and cuddle them and take care of them. So as I dove into my work, I learned that there is a real science behind this. And it's important to also highlight the sensitivities here because we're dealing with parents with babies in the NICU. So thinking about what type of photo would appeal to those parents. Who am I trying to get through to? So I looked at many hundreds of photos of babies and thinking about what responses they might trigger. So this one really appealed to me at first, this lonely baby, and I just wanted to get in there and pick up the baby and cuddle and hold and take care of the baby. But maybe photos where you see parents bonding with babies might be more powerful. And then also considering male parents and caregivers and their need to bond. And then also thinking about touch as an important consideration as well. So I thought also trying to make the message very positive. These parents are very stressed. So this was a Quebec photographer, and I came across this series of art that's a little bit more hopeful, focuses on the hope. So these NICU babies, where are they now? So they're holding photos of themselves from, and I thought it would be a really lovely gallery wall in a neonatal intensive care unit with success stories of families who have come before. So a few examples of art integrated into healthcare. First at the top is Barcelona, a hospital in Barcelona, obviously more religious subjects, but more contemporary as well with bold color and flowers and some pediatric art at the bottom as well. And they also act as to help with wayfinding. They lower stress in many different ways. So now to talk a little bit more specifically about my thesis proposal. So the idea is to design and test a series of 3 installations. And this plan on the right shows the entry to the department, thank you. The family lounge is a second installation, and then the nursing lounge as well. So this is the entry to the NICU, and there's real opportunity here to support parents And kids and siblings seldom come to this area. So that was a consideration for me. And again, this was that very first idea I had. It was very bold. And then I did many, many more versions, playing with it in different ways of how that might look to get the right impact. And then the second installation, this is the current on the left, the art currently in the patient lounge. And there's not really any privacy. Seats are all just lined up with everybody staring at one another. So although my focus is on looking at large scale graphics, I'm also interested in studying the impact of different design interventions and the impact that they can have. So in this case, maybe looking at the seating layout as well and studying different layers. This was just, again, another study of what that wall might look like if maybe there were some more cozy banquette seats with a bit more privacy and color and art, how that might look. This is a nursing lounge. So again, lots of different interventions we could do here, but first phase would be looking at just the art and the impact that could have. And there's also opportunity here for a video installation as the moms are there for a longer period of time. So that was something we thought might be interesting. So just quickly in the next few slides, a few highlights from the theory in the three disciplines. The importance of aesthetics in healthcare is not a new discovery. In this quote from Notes on Nursing from 1898, Florence Nightingale is highlighting the importance of aesthetics in the healing process. Then Sarah Williams Goldhagen talking also about the importance of disciplines coming together when we're designing for humans and understanding so many different layers. And then just talking about how buildings shape our lives every day. Every space that we're in, we're affected by. So a prime is a non-consciously perceived environmental stimulus that can influence a person's subsequent thoughts, feelings, and responses by activating memories, emotions, and other kinds of cognitive associations. Our built environment is riddled with primes, and because it is, a design can be deliberately composed to nudge people to pick one action over another. And this is what I'm trying to do with these images. So then the designer must devise ways to make us shift out of our ordinary habit-driven state and attend to our surroundings. So again, I'm trying to get the attention of these stressed parents and stir something within them. So that's a perfect segue to Thaler and Sunstein. And choice architect has the responsibility for organizing the context in which people make decisions. So there's no such thing as a neutral design. A good building is not merely attractive, it works. And the insight that everything matters can be both paralyzing and empowering. And that's something I feel every day with every decision I made. And that's the reason I've come back to study more. So if choice architects want to shift their behavior and to do so with a nudge, they might simply inform people what other people are doing. So that's where some of those images with parents bonding with babies align with this idea. And then also just talking about the automatic system, how you smile when you see a cute puppy. And that is the perfect link to another area of study. And this is where the neuroscience comes in that was very new to me. So I came across this article on cuteness, unlocking the parental brain and beyond. So the article focuses on the parental brain and provides evidence suggesting that parents have a strong response to seeing images of infants. And the statement that cuteness could be useful in designing novel interventions to strengthen troubled parent-infant relations aligns perfectly with my thesis. And they describe characteristics of cuteness and that there can be a disrupted response in the caregiving behaviors with facial abnormalities. So that was very important in my work, as these very sick babies don't have those typical cute characteristics. So this was another great book, Healing Spaces, really just focuses on the foundation of evidence-based design in the Academy for Neuroscience and Architecture. And it's kind of important to mention Ulrich's landmark study, View Through Window May Influence Patient Recovery from Surgery. And that was from 1984. And that's kind of the foundation of evidence-based design. And he found that patients with a view of trees had shorter hospital stays, took fewer pain medications, and had more positive comments in nurses' notes on their condition than those with a view of a brick wall. So I was inspired to seek knowledge in an area of study that extends across disciplines where a gap in the research exists. And if we can bridge this gap, there's the potential to have significant impact on people's health and well-being. And I'm just really excited to see where that might lead. And I'll stop there. Again, I could talk forever on this, but I'll stop there. And thank you so much. And this was also interesting. There was something that came out a week or so, oh, thank you, from York about skin-to-skin contact and pain too. So that was a really interesting thing for me to see right now. Thank you.
Thank you, Lorena. So unfortunately, we don't have yet the other speakers, so I guess that we will go on with the questions for both of you. And I would like to start for an actual question for both of you. So I think that the topics you went through are things that usually scientists should be kind of good at, because a scientist should be good at design, because if he design better what he wants to do, he will get the funding. And of course, if he has project management skills, he will get through because he can prove that he's able to manage a project. But the thing is that scientists are not trained for that. So what your take on this is, should a scientist get more training on this, or do you think that it should be something specialized and they should hire a designer and hire a project manager? Whoever wants to start.
So for me, it's interesting. This is where I really find, like, I work for Stantec as a multidisciplinary firm, and I really find that there's so much to, like, just in our own disciplines to know and learn. And I just really find that collaborating with others that have different skill sets has worked the best for me. We all need to know a certain amount in order to be able to collaborate and understand. But that's where I really love collaborating. And I love the idea going forward now of having those collaborations with people who have totally different skills. Because I'm not a scientist, I'm an interior designer, but to do my work better, I need to understand the science behind it. So I think that sort of answers your one element.
I think people can be kind of not exclusively bucketed, but into generalists and specialists. And I like to think of scientists as very specialist, and they are typically focused on their science and their research programs. And with that, I think they should be surrounding themselves by other experts that know how to support them properly and effectively. And I think that can build more effective workflows for sure.
Okay, thank you. And we have a question from the participants to Lorena. So it's from, this is very hard name, Titipat Ashakulbisut. Can you use your questionnaire feedback to help shape the design based on parental feedback?
Oh, yes. So that's a really important part. I didn't, because given the time, I couldn't really get into all the details. And at this point, I'm really just coming up with the plan and the proposal for the study. But definitely, families will need to be involved along the way, especially with something as sensitive as how they're responding to imagery. So the idea was to have focus group, for me to do preliminary designs and present them, but then to have focus groups with family members to really understand what moves them. And because it's difficult for me to understand how somebody will feel as a designer. I'm constantly trying to empathize, but these people are in a very difficult place with a newborn baby and things haven't gone as planned. So definitely family involvement is really important along the way at different times.
Okay, thank you. And Tanya, if I can ask you a question. So for a scientist that is not in such a big project that involves having an actual person dedicated just to project management, what do you think should be the most fundamental skill that they need to get to be good at actual manage their own small projects? Because the Gantt chart, for example, is something that people are kind of familiar with because it's usually in grant application of things like that. But most other things, like you talked about Git, and it's actually a pleasant surprise because for people from a computational background, it's something normal to do. With people with the more physics background, they tend to just put their code aside and don't do this formal thing. What do you think? What are the fundamental skills you would advise to build to scientists?
I think leading off from the Gantt chart, particularly time management. I feel like that is one of the biggest pillars that scientists are constantly fighting against is time. And I think I touched on that a little bit in the presentation, but yeah, time management for sure, and learning and finding what helps you keep on task and on time. And that is because a lot of the cycle in which we work within. So if you have to report to funders by a certain amount of time or have certain deliverables ready to go or meet certain deadlines, it's all dependent on your ability to manage your time appropriately. And I think the other one, and I say this because I think it's the one that we avoid the most, is managing people and really fostering those relationships across science. So how do you network? How do you manage your network? How do you keep in contact with those people and they don't just fall off your radar until the next year that you see them once a year, and that's your kind of one-off. I think if scientists start talking to one another a little bit more intentionally and thoughtfully, I think that we can build larger groups of scientists that work together, and I think that will ultimately push the field forward faster.
Thank you. And I have a question in the chat that actually relates well with another open question that I wanted to ask both of you. So this question is from Priyanka Ghosh and is specifically for Tanya, and is what is the eligibility criteria for becoming a lab manager, whom to apply? But I would like to take this chance, of course, Tanya, feel free to answer this question. I would like to take the chance to ask both of you how you started your career, that I imagine that it's not that related from the start to science, and how you ended up working with scientists.
Actually, I did start in science. I started, I was studying at University of Toronto in a neuroscience program. I was working with Dr. Randy McIntosh in my undergrad. And just as the time when it came for me to enter into my PhD program, that was also coincided with when the virtual brain was just starting to come online and kind of come to fruition, at which point I was approached because I did have some sort of organizational skills, and I seemed to have just a different skill set than that was, I don't want to say lacking because Randy's very organized, and so was the rest of the team, but they just needed somebody to be dedicated to that. So I said, yes, I'll come. I'd love to come on board with the virtual brain and start that, and give it a year, and then I can always go back and do my PhD should that be the right next step. But the virtual brain really did take off. It became a very full-time focus, and as the project grew, so did the needs. So I kind of just started to fill those needs as we built, and that goes back to 2012 when we first started, and the team just grew, and with that, so did the need for me to stay there. So I never did go back and do the PhD, and with that, it's very much I'm making it up as I go. But to answer the question about how do you become a lab manager, there isn't a specific stream. I think it's about finding whether that's something that, if you have the right skill set, or if you do feel like it's something that you can contribute in a different way, to really focus on that. And I think most people become RAs and go to a lab manager. So a lot of people do finish the PhD as well, and then go back and become lab managers once they have kind of that more niche knowledge set. So there isn't really a very traditional way about going about this, and that's kind of one of the things that we want to focus on with the Science Management Collective, of really becoming, finding one of those more formalized ways of getting into roles like this that support science. Thank you.
Thanks. Lorena?
And so for me, I really never saw myself going towards science. I think it's, as a fine arts major, wanted to do interior design. I think it was for me, it was through practice, seeing how important and how we affect human beings every day. I think it was the feeling of responsibility after 15 years of practice, that I just really wanted to know more, and felt that knowing more about the science behind how people respond to buildings in space is really necessary to take my design to that next level. And yeah, that's what inspired me. And then my supervisor, Dr. Joseph D'Souza, that connection, that's how I, you know, we'd start talking like the that Ulrich study, we'd talk about that, and then he would make me consider things in different ways of why, and just really starting to consider the science behind human responses to buildings and design.
Sounds very interesting. And I don't know if it's a question or a comment. It's from, again, Titipat, and is, I totally agree with both speakers. Design and management are both really crucial in science, and we just don't discuss them that much. And I truly agree.
It's a lot of opportunities, I think, for collaborating, right, on all of our.
Yeah. But there is always this this strange feeling in science, at least when one moves from the PhD to the postdoc, and if one manages to being a PI, that basically you need to do everything yourself, because basically you need to be a people manager, you need to be able to design things in a way, and to manage the project, and to do everything.
And you shouldn't have to. I think.
No. You can lean on one another, right?
Do you think that it should be more common that funding programs and universities should provide some support in terms of having someone that is specialized in design, project management, in people management, and support the PIs?
I think the investment in something like that would actually make the research dollar go farther, because you can accomplish a lot more, I think, with having more efficient, being more efficient with our science enables kind of a better, faster outcomes in return. So I do think that there is an ROI there for funders, that they should start recognizing this, and they have started, but to have it more widely adopted would be definitely a goal to see.
Verena, do you think the same?
Yes, I agree.
And in terms of design, so you work mostly with health, right? Do you know if there are designers actually collaborating with more basic science, or with the exploratory research, or these kind of things?
There's a lot more going on than I was aware of before. I found in the past year or so, just reading there, I mean, I'd never heard of neuroaesthetics. That's something I'd never heard of until I started this. And, the Academy for Neuroscience and Architecture, there obviously are a lot of other designers, coming together and looking at the science, but a lot of it is focused, I think, on neuroscience and responses, human responses. I'm not aware of anything, elsewhere in science right now. It's more about the psychology and behavior, and yeah.
I guess it's where the bridge is kind of shorter, and
even that's quite.
Yeah, go on.
Sorry, I was just going to say, even that is, these are recent developments too. This. Coming together, I think it's quite recent, and I think it's really important. So, it's quite exciting, I think.
Yeah, and also I think that I kind of biased, because at least in brain research, I know that at least in the last years there is this brain art movement, but maybe when you go outside the neuroscience, it's even rarer than that.
Yeah.
And since we are at 43, if I can ask a last question to Tanya, would be, I heard Randy McIntosh speaking at a workshop in June, and he told all his experience with mostly the DBS part rather than the virtual brain, so the most experimental projects. And one thing that I was wondering is, since he admitted himself that he has been to the dark side, in the sense that basically he went collaborating with industry, do you think that once you start approaching industry, it's easier to think like they do, in the sense that in pure academia, in a university, it's very rare that you find project-specific project managers, while in industry, it's very common that you will have a project, you will have the research scientist, and you will have the data manager.
Yeah, it's a slippery slope, and I think it depends on the individual. I think some people are science to the core, and they would never abandon that. And I think it is very tempting because industry does function very differently than research. And it's a challenge in a way to make them work together, where it kind of feels like you have to be on one side or the other. So there is a large amount of effort that needs to be directed towards making those two faculties kind of to function as one, to really get knowledge translation and industry partners to work for the betterment of science. They work on very different timelines. They work on very different budgets. So you can imagine that things don't line up all that easily. So I think it's very, it is a dark side. I like how you said that. But it can be very prosperous, I think. So.
Okay, and with this, I would like to thank you, both of you, for the very interesting talks and for the very interesting discussion. We made almost perfect timing, so it's very good. Thank you. And thank you all the participants for coming, and thank you to my backend host that helped us in the back. And take a 15 minutes break and keep following the sessions at NeuroMatch. Bye.
Thank you very much.
Thank you. Bye. Thanks a lot.
Using optogenetic. For those that might not be aware, it's the ability we have to transduce cells with channels that respond to light. And when the channel responds to light, it opens, leading to activation or inhibition of the cell. And here on the right, you can see when the light in blue here is turning on, we have a spike happening in the cell. Same thing at faster rate. And it's not now routinely used in labs. In our lab, we can use it to stimulate here 36 neurons at random times. And then we can, at the same time, here record for 3 different neurons using standard classical electrophysiology. Now, this has 2 major drawbacks, though, that first of all, it's invasive. So doing patching kill the cell after a short time, and also having a very limited bandwidth, in the sense that you would be lucky to do 12 to 16 parallel recordings of cells in the same preparation. So to try and address that problem, scientists have been looking at imaging brain activity with light, which is optical imaging. And here on the left, you can see one of those proteins, which is quasar2, which is sensitive to voltage. And basically, when there's a change in voltage, that protein change in fluorescence. And you can measure that change in fluorescence. And as you can see on the right, quasar2 and standard voltage recording techniques are extremely similar. And you can detect action potential and EPSP that way. So if you combine the two, you have optical recording and optical stimulation. And as you can see on the left, the two, so on the red, you have the traces of optical imaging, and on black, you have traces of classical electrophysiology. And as you can see, they look perfectly similar. So you can, in that way, record with optics, record many more cells at once, and also not invasively. So in theory, you should be able to keep your cell alive much longer. That has a limitation, and among them is that optical electrophysiology requires multiple pieces of equipment to work. Indeed, you need a high frame rate camera to capture the change in voltage, at least at 500 Hertz. You need a laser for the voltage reporter visualization,
So you need to shine quite a powerful laser on the preparation. You need a digital light processor for the pattern optogenetic stimulation, a microscope with movable stage if you want to move around the preparation. And also, if you want to do that, you need classical electrophysiological equipment. So for that, we developed that software. And we developed it using Python because Python is a rich ecosystem for software development and scientific computing. It is a large and expanding user base, and it's also free and open source, allowing for the sharing and modification of that code. We took a model view control approach, which means that every component in the model, control, and view are completely independent. And by that, it means that the user interface is independent from the data storage component and also independent from the hardware component, so the low-level code. And to take an example for that, in the view level, if I take the camera as an example, a user will be able to adjust the exposition, for example, of the camera, or adjust the focal view of the camera. Then at the controller level, it means that that level is going to store the image data and also the timing of the camera recordings, for example. And then at the model view, the hardware, it's a hardware low-level interface. And basically, it means that if you want to change the view, so the user view, because you find it ugly or something else, then you will be able to do that without changing anything else. For the controller, if you want to change the data format the data is saved in, then you can do that without changing anything else. And same thing for the model view. If you want to add some different hardware, then you can do that without changing anything else. Here's a brief overview. So when running the software, you're going to have that main window. And then you can initialize an experiment that's going to generate some metadata file, the timings of the hardware, the array position of the simulation, things like that. Then you can load different modules that are all independent. And for example, microscope, laser, digital light processor, and camera. And overshadowing, or then you can also have some config file modification if you want, and some basic analysis. And then overshadowing all of that, you have an automation module, which would allow for around-the-clock whole optical electrophysiology, which is the purpose of that, the end game purpose of that software. And with that, I'd like to thank my advisor, Alex Red, and NYU, and the funding sources, DARPA. Thank you very much. If you have any questions, please go ahead. I'll stop sharing for that.
Thanks, Jeremy. Is there anything that you would like to share about your work on this project that you most?
Yeah, you know, basically the purpose of sharing that software that's not quite finished is are people interested in it, and how robust should I make it to user base, to a certain user base. So if I'm the only one using it, then I can make the code that's robust, because I know what I'm doing with it. I know where the bugs are. I can go around them. It's fine. If someone else not familiar with the code using it, then it needs to be a lot more robust. And by the basic idea is to get an idea of how large a user base would be, if there's any, and then take that into account to then go forward with how I'm coding it. And I mean, opening it up, having people code it, code some stuff, that would be great too.
I see a question from Sophia, so I'm going to relate it to you. You probably can read it yourself too. So Sophia is asking, what sort of feedback do you hope for the community if they wish to use it? What kind of feedback do you hope to get?
Well, like how easy is it to understand, to use, how, if people want to collaborate on code, adding some hardware pieces in it, how intuitive it is in coding, in using it, what's the functions that I'm missing. I haven't been doing electrophysiology for a very long time, so I'm probably missing quite a bit on that front. So things like that. Basically, yeah, user or people that would be interested in using it, having the say in whether we want to see.
So Phil joined us with his camera on because he also has a question.
Mm-hmm.
Yes, Jeremy, first of all, I loved your talk. And as a fellow optical
imaging person, I really love when people talk about what we're doing. But so I just noticed immediately looking behind you, like I see, stimulators, and I have stimulators as well that are analog, and so often like A to D conversion can often cause all kinds of problems. So like how robust is this across the board for like different kinds of data input? Because you mentioned a little bit, could you go into further detail about like how many file types it can take, and what's it called? I'm blanking on the term for this, but like backwards compatibility with like older ways of taking. Because some of there are people who are still using functioning analog stimulators that are like 15 years old, right? And that can cause all kinds of problems. So I was just wondering.
Yeah, so most of the optical things is a direct query from the camera, for example, for the images directly into Python. So I have a low-level code that creates the images directly, and then intermediate level that saves the data, and then the user data, the user interface that shows the data. For that, there's basically no link between the computer and the cameras directly through I forgot the cable format, but anyway, it's a direct link. Then we also want to compare with classical electrophysiology in quite a few cases, at least for now, because that kind of technique is quite recent. And so for that, I'm able to interface same with Python, and there's quite a bit of available script on GitHub to interface via acquisition card, mostly any acquisition card. At least that's what we have here. And so I'm going through that to then get the data when it's classical electrophysiological data. That answer your question?
Yeah, so I am clearly not an optimal imaging person. So I was wondering, because your tool is completely open source and working in the Python environment, is it the case that the optical images are also openly available, so that if someone would be familiar with your toolbox, they could analyze images that exist in the world? Or
the idea is to make it as open as possible. So there's no data per se going with the software, but I'm hoping to get the data I'm trying to acquire right now. I'm hoping that at one point I can transform it into an NWB standard format. So for those I don't know aware, it's a format that is that aims at being completely open source with a lot of metadata in it and completely reproducible. They had a couple of workshops in the past year, which was really interesting. And the idea would be to have that at one point. I'm not there yet because the format is not compatible with that new kind of data. So it needs to be expanded, which is something you can do because their format is completely open source. And there's quite a big community around it, which is great.
Just for the people that are listening that don't know what NWB is, it's Neurons Without Borders. And I've played around with that format a little bit as well with EFIS data. Especially because Sophia mentioned earlier the backwards compatibility. As NWB is developing, I sometimes get stuck into some hiccups there, but I think it's a really great and cool initiative, and it's really nice to hear that you're actively working on software tools. Unfortunately, my phone is gonna ding in 5 seconds because it's time for us to move on to Phil. But thank you so much, Delmy. That's really interesting. Sorry. Sophia also posted in the chat what the best way is for people to reach you, Delmy. So if you can on the chat and maybe let people know.
Yeah.
Okay, cool. Thanks. So Phil Mistry uses he/him pronouns. He is a second year in the William Frost lab at the Rosalind Franklin University. And he's joining us today to talk about nudie branches and about ultra-fast workflows when working with this. Phil, I see that you're sharing your presentation through Google, I think, Sheets, and I see weird.
No, it's in PowerPoint.
Okay, because I see black bars that I don't think should be there. Sorry about that.
Strange. No worries. Is it showing now?
It's still showing me the black bars. So there's on the right side of the screen and at the top, there are bigger bars that are overlaid on the PowerPoints. I think that is where the zoom controls are. So if you make the screen where you see the faces of the attendees small, then at least it will not obscure your slides. Oh, Sophia's saying to go to the next slide. So maybe if you advance to the next slide, it goes.
Yeah. Okay.
Yeah, now it's much smaller. Thank you.
Now it's everything's fine? Okay, cool. All right. All right. Hey, everybody. Yeah, I'll be talking to you about voltage imaging in nudibranchs. So just generally speaking, activity visualization in the brain is something that so many neuroscientists are interested in. It's something that I've attended a lot of talks here about. And you've probably seen some of these techniques discussed, EEG, fMRI, and 2-photon calcium imaging. And all of these techniques are valuable and they contribute a lot to the field, but they all have limitations with either temporal or spatial resolution. With specific noting, actually, calcium imaging, I've attended various talks on calcium imaging, and they always talk about how they cannot get the actual spike data from calcium imaging. They have to infer spike data using various techniques to infer spike activity. But what we do in the Frost lab is actually get the spike data using voltage-sensitive dye imaging. And that's what I'm going to be talking to you about and describing to all of you. And we specifically use mollusks in our preparations. And there's a reason why. You might have heard of some of these. Aplysia is pretty well known. Tritonia is less known. Bergia is a very new mollusk that's entered the scene that we're working on a project to turn to the first transgenic mollusk. And we're really excited to be part of that project. But just generally, we're interested in mollusks because they have large, very easily accessible, individually identifiable neurons that can be carried across from multiple preps. And they have a relatively small nervous system, about 10,000 or so neurons, which makes it really easy to study the correlates of behavior, really easy to study networks that are directly responsible for a behavior. Because in some of these preps, a single neuron is responsible for integrating a certain motor program or executing a certain motor program. And from this, we can derive a lot of network principles, and these principles generalize really well. Historically, they've generalized really well, and we have reason to believe they will continue to generalize well. So how do we actually acquire this data that we do? So we have this an imaging rig, as shown here. We have a semi-intact preparation of usually multiple ganglia or an individual ganglion, depending on the size of the animal, underneath a lens. We shine light through it after soaking it in a voltage-sensitive dye for a certain amount of time at a certain concentration. As the preparation is stimulated, as the cells depolarize or hyperpolarize, the light intensity as it shines through it changes. And that is picked up by this photodiode array, which has 464 diodes. These changes in intensity are incredibly small, like 0.1%, imperceptible to the human eye. But the photodiode array is capable of picking up those differences. We can pick that up and run it through independent component analysis and pull out individual traces from those diodes. So what does that actually look like? So we have our rig, takes an image. This is of a pedal ganglion of an aplysia. We then do our recording, and here are this is what the traces look like in the program, right? So we can select traces and take a look at what the trace pulled out, run independent component analysis, and this gives us these Gaussian distributions of where these spikes are actually located. And we can then fit that to the original image. And now what we've done is now we have mapped, in this case, 81 neurons and their activity across 20 minutes with a precise spiking activity showing bursts, et cetera, et cetera. So if I blow that image up for you, you can really appreciate what we're visualizing here. It's been stimulated right here, and this is the escape swim motor program. And you can see these beautiful patterns of bursting across 20 minutes of 81 neurons. And mind you, this is the kind of thing that with electrophysiology, you can only get a couple neurons at once that you can record from. Here we're measuring from 81 neurons, and we have not damaged the prep. The prep is still completely intact. And then we can take that data and use, in our example, use clustering algorithms. I don't know how well this will play on Zoom, but we can cluster the neurons depending on how they contribute to the program. And with this, get a better sense of how ensembles of neurons form and contribute to a specific motor program. And once again, not inferring. These are the actual spiking patterns of the neurons in the ensembles. And we've been using this technique with our project on Bergia to identify neurons that have never been identified before in this animal. So this is just spontaneous activity in the Bergia, 35 neurons in its main cerebral pleural ganglia, and mapping these neurons. And then we have colleagues who are doing other behavioral as well as immunocytochemistry work, and they can use this information to help construct a structural functional neuronal atlas of the brain of an animal that has never been characterized before. Work that previously would have taken decades of dozens of teams' work, we can now do in a very short time frame in any brain if we can record from it. So to quickly summarize all this, what I've described to you is a really fast workload. Everything from dissection to analysis takes less than 24 hours to actually visualize network dynamics at a single neuron, single spike resolution. And because the preps are not damaged, they can actually be further studied based on the activity maps that we've described to stain for various neurotransmitters and stuff like that. And the data we've generated, the spike trains that we're pulling out, has actually been used by some of our collaborators to develop new statistical modeling techniques to better understand how networks form and change and how their processes evolve over time. So just finally, I just want to acknowledge, want to thank everyone in the Frost Lab, as well as our collaborators in the U01 project, as well as some of our collaborators in the past, computational people who have helped us make sense of all of this data, to the NIH for funding us, and to Neuromatch for holding this amazing conference.
That's great, Farrell. Thank you so much. So one of the things that I'm particularly interested in is you talk about bursting, and I was wondering what your definition of a burst is, because I think, in the literature, bursting is quite often thrown at terms where sometimes there's a very strict nomenclature that what bursting identifies, and sometimes it's very loose. I was wondering what you use here and what you're referring to.
Yeah, so I guess actually the simplest way to say it is actually in this image right here. I would describe the activity of neuron 27 right here, this, as a burst, whereas I would say some of this other stuff is just spontaneous tonic firing.
I see. Okay, cool.
And some of this is tonic increase in firing, but like bursts typically have like this pattern of a repeated firing and then a clear period of returning to rest, followed by a series of firing, followed by a return to rest.
Okay. And another thing that I was, what I wanted to ask is the next step going to be to relate these patterns to behavior? I know that as a model animal, the Plesia is of course very famous for the work that Kandel, but also others have done on touching the, I don't know what this thing is called anymore, but that kind of conditioning and looking at the neural responses there.
I think you're referring to the gill siphon withdrawal reflex.
Yeah, exactly.
And a lot of other famous. Yeah, that was of course. Yeah, so actually, yes, that is exactly what we're trying to do. We're trying to make sense of foraging behaviors and other stuff. So we have collaborators who study the behavior and are trying to figure out what behaviors we should be studying in the first place. But of course, to begin with, we're just trying to establish certain parameters of spontaneous activity, because I think that's another thing that oftentimes gets left out is there's just so much native activity, even if we're not even doing anything to the prep, let alone the idea of like trying to maybe stimulate for a scent or a predator or something like that, or give it an aversive stimulus and stuff and try to elicit an escape response or defense posturing. But yeah, all of those things we are super interested in doing in Bergia and just generally. We have done that with our Tritonia and Aplysia preparations, where we are capable of eliciting escape responses, as well as like feeding responses and stuff like that. So yeah.
Okay. There's also a question on the Q&A for you from Srinivas. who is asking, is this a genetically encoded sensor? How did you get in?
Okay, that's a great question. So actually, in these examples, they are dyes. They have been stained with a dye. Now that creates some limitations, right? Because you have to get the right concentration, the right kind of dye, stained for the right period of time. There is, however, recently, I anticipated someone would ask this question. So recently, there has been Voltron has become that's like the best genetically encoded voltage indicator that exists. And here's a recent paper in Science validating it in zebrafish, mice, and a few other species. And our goal is to get Voltron into Bergia, as well as just generally, we think this technique has validity outside, not just in mollusks, but across the board. And we would like to use that. If we've been working in mice, it would be ideal to do it with something like Voltron, because we have done work in mice before with our voltage-sensitive recordings using a dye, and we got it in the end, which is actually what this paper was, but it was an incredibly difficult task. And so having something like Voltron would have made our lives a lot easier.
Jeremy also has a question. I was just asking to ask it yourself, but I will let him decide that. There's another question for you in the Q&A. Someone is asking, can you have a behaving animal while recording VSD signals? Because it wasn't clear to this person how the animal was fixed. So are they? So
you can have a semi-intact preparation in Aplysia or Tritonia where you preserve some aspect of the musculature, or like in Bacille siphon in the Aplysia example, right, with the pedal ganglion. In Bergia, we're working on trying to have a more intact preparation that includes the organs that it uses to touch and to smell. So that is a longer term goal that we're working towards. In the moment right now, we have not developed a preparation that preserves enough of that to say that, like, okay, like we have those things directly. Instead, we're stimulating nerves and using that as sort of a corollary to what would happen when it takes in that data from its periphery.
Hey, yeah, that's really cool data. I have a question about what type of pre-processing did you do after acquiring this data? Because like usually voltage imaging, even if you can get very, very good resolution, it's still noisy.
Yeah, so this is what the raw data looks like. And this is like, if it's a good, clean, solid recording, it's still very noisy. This is after you put in just a high pass filter and a low pass filter, filtering out anything less than 10 hertz, everything higher than 100 hertz. And then this is what the data looks like after ICA. So as you can see with the filtering, you can make out spikes, but there's still a lot of noise that if you didn't have ICA, you would eliminate a lot of data. Like it would become indecipherable without ICA.
Okay. And have you tried different type of pre-processing, and what kind of variation did you see on that data?
Could you clarify what you mean by different types of pre-processing?
So you used an ICI. Have you tried PCA or non-matrix local factorization?
Yes. So we've done PCA in the past, and actually Terry Sinowski did write a really phenomenal review where he did outline that actually ICA is more effective than PCA, which is why we've been historically using ICA, but we don't know if there are other techniques that might be better than ICA. But so what was that technique? I want to write that down. What you just said.
It's from Liam Paninsky at Columbia, where they did a whole, they're working on a whole pipeline for processing different type of optical imaging data, and among those voltage imaging data. And it's really good at getting rid of background signal.
Gotcha. Thank you.
Yeah, thank you. Oh, and Voltron in invertebrate, that's really cool.
Yeah, that's the goal. That is our plan. I mean, one of our collaborators, D.D. Lyons at UCSD, is working on developing transgenic animals, and along with GCamp, one of the things we've asked her to also include is Voltron.
Awesome. Thanks both. Thank you so much, Phil. We're going to move on to the next speaker. Our next speaker is Max Hoffman. He uses he, him pronouns. He is currently a PhD student with Benjamin Jurkowitz at the Humboldt HUT in Berlin. And he measures neural activity from translucent fish. Take it away, Matt.
Yeah, thanks for the introduction. Yeah, so I'm going to talk about oblique plane microscopy for whole brain imaging of Danionella translucida. Our lab works on a new model organism. It's called Danionella translucida, and it's very interesting for neuroscientific imaging studies, basically for three things. First thing is that it's transparent throughout its adulthood. And a second thing is that it has the smallest known vertebrate brain, and you can see it here. And since it's transparent and it's so small, an additional good thing about it is that we can that it's amenable to genetic modification, and we can express, for example, calcium indicators throughout the whole brain. And all these things make it very interesting for imaging studies. And of course, the first thing you ask is what would be the right scope to kind of actually image the whole brain of this fish. And of course, different techniques come to mind. First of all, to photon point scanning here on the left, which obviously would have the right field of view up to 5 millimeters to accommodate the brain. But as is known, it's relatively slow because you have to sequentially step through all the voxels in the specimen. Yeah, and an alternative to this is light sheet, where you excite a thin sheet of light throughout the brain, and then you capture a camera image, so you can capture actually a whole plane at once, and you're very fast, and the resolution is still good. But here you require access from the side to the specimen, and even in the transparent fish like ours, the access from the side is occluded by eyes, cartilage, or some bone structures. Yeah? This is why we thought that the technique called oblique plane microscopy might be the right choice, which is basically similar to a light sheet microscope, but everything works through one objective. So you excite an oblique plane in the specimen, and then you capture the emitted light. So I can quickly walk you through a normal or conventional setup of OPM. Yeah? Here we would couple a blue laser inside at the side of an objective, and this would excite the oblique plane, which gives the technique its name. And then the emitted fluorescence could be captured by the very same objective, and by scanning this laterally, you could kind of puzzle together a volume. Yeah? But in order to kind of record this oblique plane, you usually have to do a re-imaging trick where you kind of put another system back to back and create a very small one-to-one image of the specimen, and then bring a third objective, a third microscope, which basically then images the oblique plane onto a camera sensor. So the brain of our fish, Danionella translucida, is 2 millimeter by 1 millimeter by 0.6 millimeter approximately. And no conventional oblique plane microscopy with this kind of field of view exists. And this basically has to do with this re-imaging step, where, as you see in this graph, it's inevitable that you lose portions of the light because some of the light is not propagating anymore in the cone of the second objective. And in optical design, it's very well known that this angle, which we basically call the numerical aperture of the system, correlates with the field of view. So high numerical aperture, large angles usually mean small field of views, and low numerical aperture, which are shallow angles, mean large field of views. So here, and this is the problem, because here you see that basically once you go to lower angles and larger field of views, this problem with the lost light becomes a very severe one, and in the end you lose almost all the light. So for example, in a system like ours, which works at 0.5 NA, you see here that if we would build a setup in a way as you usually do with OPM, all light would be lost. So therefore, we thought of a new way of doing this, which we call image transfer oblique plane microscopy, where we basically insert a faceplate, a fiber optic faceplate, which is tapered on one side and guides the light from the oblique plane to a straight plane, which can then be imaged on, onto a camera, as in a normal OPM. So with this technique, we are able to reach a field of view of 2 millimeter by 1.4 millimeter times 1 millimeter, and then resolution, which is approximately 2 micron by 2 micron by 40 micron axially. So now with this technique at hand, we can proceed to kind of try to image Danionella translucida in its brain. And indeed, we can do this now, and we capture a volume like this at approximately 1 hertz or at 1 hertz, so 300 planes per second. And with this kind of tool right now, we can actually start to map whole sensory pathways at 1 hertz, like here during audio stimulation. Yeah, and I guess this hopefully puts us in a position to start to address some interesting questions about this brain. And yeah, so with this I conclude, and I want to thank my lab, and I'm happy to take your questions or lead it into the discussion.
Cool, thank you so much, Max. So I have a question, actually. I don't know very much about translucent fish. What is happening in that video that you showed us, where you see the fish kind of moving?
You mean in the neuroimaging video?
Mm-hmm.
Yeah, I think so. Basically, we have to. So the fish are embedded in an agarose matrix, and but they are awake and they are not paralyzed. So you sometimes during this or during this recording, the fish basically twitch, and so this movement is so strong that you even as those videos are motion corrected, but yeah, those movements are so fast that you cannot actually correct for it. And then when analyzing those recordings, we would just exclude those frames.
Cool. I see also now that Martin posted a question for you in the Q&A. He's saying, really awesome. What is your spatial resolution?
Yeah, like I say, like I said, so the optical resolution here is basically 2 by 2 by 14 micron. Yeah. And of course, you see here that even in a tiny brain like this, when you want to penetrate an image deeper, you start to see scattering effects. This, of course, I mean, lowers the resolution more. Yeah? I think maybe this question, I hope this answers the question.
Cool, thank you. And so what do you think is the next step in this project going to be?
Like I said, so one interesting thing about Danionella translucida is that they communicate acoustically with their conspecifics. So we are currently working, trying to find out how those sounds of conspecifics, those innate sounds they produce, or it's not clear whether they're innate or not, but how they might be processed in the auditory pathway, and maybe differently from other sounds. Yeah? So that's the question, or those might be the next steps. But of course, I think there are many interesting questions. Which you could ask, and the nice thing is that it's a very high. The throughput of this technique is very high. So this in like 20 minutes you can basically map out the whole brain and responses. So for example, whatever. So changes from one day to the other, you could with very little experimental effort in principle kind of track. Yeah? And yeah, I guess that's what makes it very interesting also.
I think Jeremy has another question. Jeremy, do you want to pop on and ask again?
Yeah, I just had a question of what type of analysis you plan to do after that, because you can do I think, I'm not completely familiar with working with that animal model, but you can do like olfactory related tasks or social tasks, things like that.
Yeah, so for now in our lab, like we don't have the real task that we established so far. So far we restrict ourselves, or in these early stages, to actually do replay studies where we try to map the sensory pathway without any kind of task that the fish might perform. But
it's a little work.
Yeah, no, I mean, I guess, but yeah, so people in the lab are kind of pursuing those things, and I think eventually, I guess, things will then come together.
Cool, thanks.
Yeah, thanks for the question.
Max, there's another question for you from Alina in the Q&A. They're asking, how comparable is the size of the adults from your translucent fish to a larval zebrafish? And do you think it will be possible to do recordings from larval to adult stage in this model?
Yeah, so, okay. So it's a while that I had a larval zebrafish under the scope, but I think, so I guess larval zebrafish would be, the length of a larval zebrafish brain would be maybe around 0.6 millimeter or something. Maybe at 6 days or so. So yeah, so this is the comparison. I think it's maybe 3 times or 3.5 times longer, yeah, larger. Yeah, I think it's very well possible that, or like the answer to the second question would be, yeah, I think it's possible to image the organism for development.
Throughout the developmental stages.
Yeah, yeah, throughout the developmental stages. Yeah.
Yasir also had a question for you. They're asking, can you image the fish upside down as to improve the resolution of the side that is like less, well visible now? So I think.
Yeah, okay, sorry. Is there, no, I didn't want to interrupt you.
No, I just wanted to say
okay.
About the slide that you still have on is really nice to illustrate that, because I think that is what they're referring to, that
you mean the rotating volume. Yeah, so I think no, we can't. I mean, the fish has other organs, yeah. So it has the mouth and things that are situated below, and it also has a jaw, right? So it's after all like a vertebrate which has some bones. So I don't think that it would be the right direction. So I think it's less transparent from the bottom. So I think if you want to kind of go, or I think there are certain things which you might like computational do, or you could try to think about structured illumination approaches, even in this OPM microscope. Apart from this, eventually, I think you have to, like switch to a 2-photon microscope. And obviously, you could also think about a 2-photon oblique plane microscope. So the signal, I guess the signal would be much lower, and I'm not sure how well that would work. I think those, are the kind of directions I would take.
Cool. Okay. I think we are getting close to the end of this session. I don't know if anyone has still a question, because we do have a couple of minutes left.
I can also quickly answer 2 questions that I didn't get to answer in the chat that I see. One I think is from Sophia, and the other one is from Luigi. So to Sophia's question about how I say networks evolve over time. So we have recently published over the last few years work on networks changing in an escape response over time, over the course of a 20-minute recording. And that is something that we can capture because we can watch the spiking pattern and the bursting patterns change over the course of a full 20 minutes. And that is something that through our technique we can catch. And then to Luigi's question, where was it? How can this be linked to a behavior? So in Aplysia and Tritonia, escape swim response and gill siphon withdrawal are very well studied from an intracellular electrophysiological level, to the point where we know exactly which neurons are contributing to that behavior. So we can stimulate those neurons or stimulate the nerve that directly leads into the ganglion that feeds those responses, and then just observe and just optically record from the ganglia as it's responding. And that's what we're trying to identify in Bergia, is like which neurons and which nerves are contributing to a specific behavior, so that we can stimulate that and then just observe the rest of the network as it's firing.
Cool. Okay. Yeah. Do you all want to turn on your camera and say bye?
Yeah.
Thank you so much.
Yeah, of course.
It was really fun. I had a great
time
too. So we posted your Skype, your Skype handles, your Twitter account handles, so people can reach out to you. Are you also all reachable through your internet websites, like from the lab or something like that? Yeah. Okay.
I think so.
Okay. Awesome. Well, thank you so much. Enjoy the rest of your day. Thank you for joining us. Take care.
Thanks for having me.
Thanks.
Bye.
Bye.
introduction in about 30 seconds.
Sounds great.
You have about 45 minutes in total. So yesterday I told my speaker 30 minutes and 15, but of course, you know, it's a little late to give you instructions now.
Yeah.
So whatever, if you're gonna go over 45, then we're gonna get cut off by
yeah, I doubt it'll be over 45. I think it'll be close to 30, 35.
All right, cool. Yeah, so that should give us a little time for questions. I'm sure Daniela is going to have a question. I see her in the audience.
Good. One of my favorites.
I don't know who else is in the audience from your people. So, all right, so let's start. I think it's 8 o'clock in Vienna. It's 3 o'clock in New York City and 3 o'clock in Western Massachusetts, from where we are delivering live and direct. Michael Long from NYU Medical Center giving his keynote lecture today. Michael did his PhD with Barry Connors at Brown, and then only after his PhD, I believe, fell in love with the birdsong system, and then didn't leave it alone, took it with him, started his own lab at NYU Medical School, and has been working on it ever since, using it as a model also for speech production, obviously. And I think that's what we're going to hear about tonight. So I'm excited to host Michael and excited to hear about your work. Without further ado, the floor is yours.
Thank you, Tim. This is great. So I wanted to start with what I think is a familiar scene for most of you, especially in these times of trying to communicate with other people over video conferencing. And it's really, I think, in some sense, video conferencing has been a savior. It's knocked down things like our carbon footprint, traveling from one place to another to talk to people. But a lot of it feels somewhat unnatural. And so I found this cartoon online. We're meeting to talk about call. What's call? Looks like his screen froze. Should we wait? Yes, let's wait. I was on mute. I agree. I'm sorry. What did I miss? So this kind of frustration of lacking a clear and easy back and forth, which really we're quite tuned to do. And so I wanted to give a quick image that I think is illustrative of this back and forth conversation. And that's this annotation of just a random podcast, these two people talking in a podcast setting. And it's 2 minutes of conversation. So every time that the person on the top is speaking, we give that person a white shape, and the width of that shape is the duration of that speech passage, and the person at the bottom gets a red shape for the same reasons. And you can see that there are clear, there's clear back and forth that can happen here. And this is known in speech literature as turn taking. So very fast, throwing the ball, if you like, from one speaker to the next. And people have looked at this. The Levinson group has done the most thorough analysis of this. And what's surprising to me when I saw the data is that the modal amount of time between speakers engaged in conversation is 200 milliseconds. So that's roughly the blink of an eye. And just to take you through this graph, any of the negative numbers means that one person is actually talking over the other person for a certain duration. And these positive numbers here are gaps in between speakers. And just as a demonstration, you know, if I were to wait a full second to speak, it can actually seem quite painful. We're really optimized for sharing information across people in this kind of a way. And we can do it much faster than most other speech tasks. And so if you think of other things that we do in terms of speaking, if we think about a lot of the speech literature and what's been done, there's things like picture naming that usually takes about half a second, syllable reading, and then reading sentences. This turn taking is 200 milliseconds and is really extraordinarily fast. And how does the brain actually do this in order to have rapid exchange across individuals? And the secret really is in planning. So if you think about the stages involved in conversation and in kind of vocal exchanges, there's the perceptual stage, so listening to the other person. There's speaking, but almost immediately during our perceptual stage, there's also a planning stage, right? And not in any kind of rude way. We're not sitting here thinking about what we're going to say as the other person is speaking and not listening to them. But we're constantly forming a model of what we're going to respond with. You can imagine how ridiculous it would be, for example, if we wait for the other person to stop talking to start thinking, right? So we're constantly thinking and planning. And we want to find out how this planning step works for interpersonal communication. And there are some challenges inherent in studying planning, especially in this kind of fast back and forth context. And so one challenge is, unlike listening, where you have a clear environmental stimulus, or speaking, when you're creating sounds, speech planning is an internal state with no physical correlate. And a second challenge is that turn taking, as I mentioned, is blink of an eye rapid. And this requires a high resolution method to track this. And so if you think about a number of the speech experiments that have been done, monitoring human brains with things like bold signals, these are slowly evolving and may not have the right kind of resolution to track this back and forth. So let's address each one of these challenges one at a time. So the first one is that it's an internal state. And so to try and parameterize speech planning, we took something from the literature that was actually spearheaded by Sarah Vogels when she was working in the Levinson group. And they created something called a critical information task. And I think it's just such a clever task. And so what Sarah created is the following. There's a series of questions. So you can imagine someone being asked 20 of these questions in which just this single word is varied across questions. So I can ask, the opposite of sad is what word? The opposite of big is what word? The opposite of dry is what word? And anytime I hear that golden word, I can start planning my answer. Everything else kind of falls away because, again, it's a big battery of questions. When this happens, you can see, for example, in one of our respondents, this participant can answer in less than half a second, even though, and I'll tell you later, this is a person who has electrodes in their brain, is actually in the middle of a neurosurgical procedure. So there's a dirty trick that can be done here. So you can imagine now taking that critical word and putting it all the way at the end. The opposite of what word is sad? Okay, so in that case, if we do this planning, which is this kind of irreducible time that we need in order to retrieve and answer this question, starts with the word sad, and this is reflected in the behavior. So you can see the distribution of answers when that critical information is given at the end of the question pushes off to the right, like this, right? So speech planning is, and this is behavioral evidence that contributes to rapid responses in these staged vocal interactions. So the second challenge is how to measure these fast dynamics in the brain. So turn taking is rapid, and how can we do this? So the method that I think is really uniquely posed at this point, uniquely poised to do exactly that, is electrocorticography. So the idea there is we can record local field potentials directly from the surface of the cortex. We can do so with high spatial and temporal resolution. And I should say these are patient volunteers undergoing treatment for either epilepsy or brain tumors. These are collaborations, I should say, with Jeremy Greenlee, who's an incredible collaborator, works with the University of Iowa neurosurgery team, and we've worked with them together on a number of projects. This is, I should say, the location of ECOG electrodes, these white dots up on the upper right for 2 different patients. So we can get very good coverage and record activity in these patient volunteers. Okay, so what do we see? We look at the high gamma activity, which has been shown to correlate with spiking activity near these electrode sites. And I'm going to show you for a single trial the activity of two different electrodes in a single patient during this exchange. So this is another critical exchange, a critical information question. Now instead of, what's the opposite of big? Or so this is a body part question. How many shoulders does a healthy person have? We can ask how many fingers does a healthy person have, how many heads, etc. But that shoulders is the critical question. And if we record from speech motor cortex, what we see is something that's not entirely surprising. The activity of this electrode seems to really peak here as the person talks. So as articulation happens, this specific electrode in motor cortex shows a robust response. But if we go into Broca's region, we see something very different, right? We see that the activity starts to track up with the introduction of this critical word, shoulders in this case, and if anything, comes down and dips below baseline when articulation happens. So it seems to be something is going on that at least has the temporal characteristics of speech planning. So let's look at every single one of these trials for these two electrodes. And the way I've organized this is that each line is a trial. The color code here is just a z-score of the high gamma activity. So you can see the high responses are in yellow here. And so you could say, how many shoulders does a healthy person have? Going from left to right, the experimenters shown here with the black ticks. And when the person responds, the person has 2 shoulders, okay? You can see that every time that person responds between these red ticks across every one of these trials, there's robust activity. So the speech motor cortex is active during this articulation. If we look in Broca's region, we see something a bit different, right? So we don't really see much happening during the response. That's shown again between these red ticks. And we don't really see much during most of the question, which happens between the black ticks. Instead, what we see is if we consider the critical information, or that single word that can allow for the initiation of planning, if we align to that critical information, what we see is something that looks really quite robust here. So Broca's region is initiated, right? The activity here is initiated with the critical information, with speech planning, and if anything appears to be off by the time that the response is given. Interestingly, in this case, there were 10 questions that the patient did not answer. Either they didn't know the answer to the question, or they didn't want to answer the question. And what we see here is very little activity in this Broca's region site. So what we can do is now consider all of the electrodes that are implanted here for this patient in the forebrain and the cortex, as well as a strip of electrodes here in auditory cortex, actually from a different patient. But we can now organize across every single one of the questions that were given. And I want to draw your attention to the left-hand side, upper left here. And I'm showing the average responses for a 400 millisecond period immediately before the question is asked. And what you can see is these sites, each represented with a dot, are all kind of blue. So if anything, they're at baseline or slightly below. But when the question starts, a number of things happen, right? So the other speaker starts to talk, auditory cortex lights up. You can see these are large red responses. It's just the z-score of those responses. So the question itself is activating auditory cortex. Not surprising, but at least is validation for us that this high gamma response, at least as we're recording it, gives a truthful measure of cortical activity. Then as soon as that critical information is given, again, this grayed out area here on the left, what you see is an enormous amount of activity that's happening in Broca's region. This then, during the, right before the onset of articulation, and then during articulation, you see an activity that really bubbles up in sensory motor cortex of the patient. So there seems to be a trend, and then when it's done, and this I think speaks to the speed of the electrocorticography technique, everything returns down to the baseline. So to just see that movie once again, this is before we hear the response activity in auditory cortex. This is when planning begins in Broca's region. This is transferred over during speech to speech sensory motor cortex. It goes full bloom during the actual articulation of the answer, and then done. Okay. So this was really exciting to us. We're the first group to look at this kind of speech planning in this way, using these kinds of fast techniques in order to try and parse this. And a lot of people had implicated various cortical regions in speech planning for a number of different tasks. And I just want to say that there are a lot of different types of speech planning that can happen. We're very interested in how this happens for interactive vocalizations, for interactive speech. And so, so these are just some areas that have been implicated. And what we see, these are in white, all of the areas that we've recorded, nearly 1,000 different cortical areas across 8 patient volunteers. And the areas that showed us planning responses is doing a large GLM in order to classify each electrode's response as either sensory, motor, planning, or none of the above. And we see an enormous number of these planning responses that are clustered in this region of the brain. And so that region includes, and actually really seems to be centered around, what's traditionally thought to be Broca's region. And so this was discovered back in the 1860s by Paul Broca. This is the brain from Broca's first patient called Tan. That person could only say the word Tan over and over again. And you can see there's quite a large lesion in just the area that I've shown you. Since the discovery of Broca's region, there's actually been very little agreement on what it might be doing. And so people have proposed it could be involved in working memory, cognitive control, processing linguistic information, music, math, many other functions. And we think what could be a central role for this is planning for interactive language use. And so we wanted to characterize this a bit more, and we wanted to find out, is this generic planning? Are these signals going to be relevant for anything that is grossly motor? Or is there something linguistic and specifically language relevant for the responses that we're seeing? So what we did is a series of tasks that became increasingly more linguistic as we went on. And so the first task, we simply asked the volunteer to, with their hand, click a button 2 times. So this involves none of the muscles that are involved in normal spoken language. Of course, if somebody is using sign language, it's a very different story. But for this, we wanted to engage muscles that were not articulatory. And when we did that, we saw very little response in this region. This is just the average activity of a single electrode. We then could ask, maybe it has to do with orofacial movements. So the way that we did that is we had a number of different activities that we could just capture with a camera, pucker your lips, stick out your tongue, a number of these other things, and we find that, again, there's very little response. We could then bring up speech, but speech doesn't require any kind of lexical access. So we calling this non-linguistic speech. This is just simply repeating both words and non-words. The example I'm going to give you here is say the sound ba clearly into the microphone. So I've handed you something, namely ba, and I want to get ba back. And when I do that, I see a small response, but nothing nearly as large as what we see when we tax the system with a more sophisticated linguistic operation. So say the plural of goose clearly into the microphone. Of course, the answer there is geese. And here we can see that that area is strongly engaged. So we can go back to all of these planning electrodes that I showed you on the previous slide and characterize these as being maximally active for generic premotor actions. As you can see here on the left, these pink dots. Importantly, some of these are in the finger region of motor cortex, orofacial non-speech, linguistic speech, this repetition, and finally, these linguistic operations. And we see the largest response in linguistic operations when a person is forced to pluralize a word. So this seems to be something that is not just a generic premotor planning dynamics, but instead something that's preferentially active during these linguistic tasks. Okay, so The next question is, are similar brain mechanisms involved in artificial linguistic tasks and free speech? So I've so far been analyzing a number of these really scripted tasks that we can use to pull out dynamics. And that is, for example, the opposite of sad is what word? These critical information things. But that's not really what happens during normal conversation, where people are engaged in free exchange in a very open-ended way. So let's see what happens there. So first of all, we wanted to convince ourselves that people, even in this very extraordinary setting of being in the middle of a neurosurgical procedure, could actually engage in natural conversation. So we looked at the floor transfer offset, or the time it takes for one speaker to give way to the other speaker. And what we find is that even in this very specific, very challenging situation, they can do this quite quickly. So this kind of turn taking is something that that can be done even right on the surgery table. Okay, so what do we see? And so what we see looks kind of like this. Again, going back to the podcasts that I showed you, this is the same kind of annotation we use here. So when the experimenter talks, they're given this gray shape, and when the subject talks, they're given this white shape, and we can zoom in on that and look at the activity of two representative electrodes, one that's in motor cortex and one that's in Broca's region. And let's read what this conversation is about. Were you born in Mexico? Asked the experimenter. The volunteer says, New Mexico. Have you been to Mexico? No. Never? Never. So let's piece this apart a little bit. Speech motor cortex, this blue shape, seems to be active when that person is speaking during articulation. You can see here when they say New Mexico, there's a big bump. When they say no and when they say never, again, bumps. But when we look at Broca's region, what we see is something that is somewhat predicted by these more scripted tasks. And that is that it seems to be active during planning, and specifically on while the other person is speaking. So as the experimenter asks, were you born in Mexico? You see that this signal starts to rise. The person is planning their response. Have you been to Mexico? There's a bump. Never. There's a bump here. And this seems to be really consistent across all of our patient volunteers. Okay, so in the last series of experiments I wanted to tell you about is, can we do anything to actually get towards causality? Of course, it's a human brain, it's large and complex, and these kinds of causal experiments are very difficult. But one thing we can do is pass a small amount of electrical current to see if we can disrupt dynamics, to ask if that leads to any clear behavioral deficits. So this is a site here. This is roughly in that same region of the brain for a patient volunteer that has activity that's triggered by this critical information, so planning-like activity. And what we can do is give a very small amount of electrical current at that location at different points within these kinds of interactive tasks. So let me take you through this figure. So we have half the tasks in which stimulation is delivered and another half the tasks where stimulation is not delivered. The question is schematized as a gray line with the critical information in blue. The response in this case is either red in this case, or in the stimulated case, it's white. The time of stimulation is shown in yellow, and you can see that in the stimulated cases, first of all, they can still talk, which is important. I'm not showing you that here, but it is true across all of our cases. But it does also delay the response time by 50%, and significantly so. But what I thought was even more intriguing was the errors that start to emerge during the stimulation. So here's an example. Say the plural of man clearly into the microphone. Okay, this is the question. The stimulation time is shown in yellow here. And they said man. So the correct answer here would be men, right? That's the plural of man. You can see that this was an error. In fact, the errors jumped up by an order of magnitude during the stimulation. So it suggested that some process of lexical axis was disrupted during stimulation of Broca's region. Which word is the opposite of fast is the question. The stim was given here. The person said rapid. Rapid is not the opposite of fast. Rapid and fast are synonyms. And so when the stimulation ended, apparently there was still a memory of this, and the person said the opposite would be slow then. So how all of this happens is something that we're really actively investigating now. We've seen this kind of thing in 4 out of 4 patient volunteers, and we're analyzing the data as we speak. So to take you through a summary of what I've told you, speech perception, planning, and production are separately represented in the human neocortex. Language planning network necessary for interactive speech includes Broca's region. The interactive network displays consistent dynamics during scripted tasks as well as natural conversation. And perturbation of the activity here, this language planning network, leads to slowed interaction as well as linguistic errors. So I want to say, taking this one step farther, that coordinated vocal exchanges are not unique to humans. They actually exist at several different in several different species. And if I were to show you every species that had this kind of temporally precise exchange in which overlap is avoided and silence is minimized, it would take probably hundreds of computer screens to show you that. But here I have a small smattering of them. Here's a crow and a hyrax and a bottlenose dolphin and a meerkat and a Japanese macaque, bush cricket. Just a few of the large number of animals that communicate with this kind of turn-taking vocal behavior. And so we're trying to leverage that and trying to look at different animals that could take us into a more cellular view of how this kind of interactive communication works. Put together along with Frank Gunther and Greg Castellucci, who's really the dynamo postdoc in my lab, who's really driven the story I've told you today, put together a series of algorithmic steps that we think are going to be fundamental for interactive vocal communication. The details of this are not important, but I can tell you that certain of these steps can be found in different model organisms. So we're looking at the budgie. The budgie can do really elaborate vocal imitation and cultural exchange. This is something we're excited to study, and we are studying it, beginning to study it now in our laboratory. As well as a singing mouse. So the singing mouse is something, we had a paper on this last year. It's a Costa Rican rodent that lives in the cloud forests and has these very, I think, beautiful, elaborate exchanges with conspecifics, this turn-taking behavior. And the idea of using different model systems to try and explore different algorithmic blocks within this pathway is to get down to a cellular level. And this is something that we've done in the songbird and recorded extracellularly and intracellularly, looked at cell identity and connectivity, and a number of these things. Had a recent paper on this. And I should say that the singing mouse work, which I think has really taken off because of the really excellent efforts of this very talented postdoc, Arko Banerjee. And I should also give a quick plug. He's going to give the next talk in this series as well. So if you want to hear more about this, you should really do that. And it's a fruitful collaboration that started with Steve Phelps, and Daniel Akobe was a graduate student who did a lot of the fundamental work on those singing mice. So it takes a village, and specifically here, it takes a Greg and also a Jeremy to get this project off the ground with human interaction and how different cortical regions and cortical dynamics could lead to those fast give and take that is part of conversation, but not necessarily part of Zoom calls. Thank you.
Thank you very much, Michael. I loved it. I have a million questions. I'm hoping to get them all in before someone else is going to type one up. So number one is, how can you dissociate planning from comprehension in Broca's area? So you've always, are you sure that there is no additional stage there that produces the motor signal or that cues the motor signal? And then question number 2 is whether you can do in-flight corrections. I mean, you see sort of the corrections when you stimulate, where, you know, they alter their answer post-hoc, but what happens if you make them abort mission?
Okay, so let me take the first one. I think that the way that we're able to look at planning versus comprehension is we use a number of these very simple tasks. So, whoops, I moved to Maddie. Where are we here? So the tasks really should be at a very elementary level. And so you can imagine taxing the system more and really trying to explore the boundary of the knowledge of that participant. But these are the opposite of sad, the opposite of wet, things that one could get at a child's comprehension level. And so I think it's an exciting question, but I don't think that comprehension necessarily factors into this. I should say that planning is a big, in this case, big blue box, and we've included our simple idea of just planning for interactive speech. And this has a number of components, one of which that we're excited to actually test explicitly is working memory. So once you've had the answer, you also have to hold that in some online memory cache. And we don't necessarily know if that's carried out in Broca's region as well. And so one thing that we'd like to do to really try and piece that apart, the reason here is that people are instructed to answer as quickly as they can. So in some sense, they spit out their answer before they really have to hold on to it for any prolonged period of time. So what we're going to do now is actually give a cue, and that cue could be several seconds later to then answer the question, and that could help to expose working memory cortical regions that either could share a location or actually be found in a different place. And that's something we're excited about. The in-flight correction, we would love to look at that. I think there's a lot of fun things that one could play with. And if you imagine this kind of thing, I've given you, from Sarah's wonderful paradigm here, a single word to answer the question. So imagine if I gave you 2 words that you have to hold on to both of them. How is that integrated, right? Or if I give you a word and then a cancellation, how does that actually get reflected in these cortical dynamics? And that is exactly where we're going, and a really wonderful question. And the simple answer is we don't know.
All right, thank you. Sorry, I can't unmute myself when I'm staring at the question. So question number 1 is from the audience. Joseph D'Souza wants to know, what are the roles, or what is the role of basal ganglia in your models or in the speech production chain?
It's a wonderful question, and we'd love to get at it. Right now with electrocorticography, we're really sampling just the cortex and not deep structures. But what is absolutely true is that people have been able to make wonderful recordings down in the basal ganglia, specifically, areas like subthalamic nucleus and others in deep brain stimulation cases. So there, even single neuron responses could be recorded in these regions. And so I think it's really exciting to think about, and we'd love to know. People have pointed to basal ganglia circuits as being important for chunking, for example, and that's something that we could look at as well.
I actually wanted to follow up on the single cell resolution question. Also, in your surface electrode, how far can you resolve this? I mean, you've only spoken about activity of electrodes, but could you go any more?
Right. So with this tool, no, but luckily 1 floor above me in our institute at New York University School of Medicine is Yuri Buzaki. And Yuri's done a million things in his career. He's an incredible colleague. But one thing that he's come up with recently, and this is Dion and Jen in his lab, they both moved to Columbia and started their own faculty positions there. It's something called NeuroGrid. And the idea about the neurogrid is that the spacing is equivalent to what one would use in, for example, a silicon probe, but it's laid right on the surface of the brain. And in their cases, they can get what appear to be single neuron responses using the neurogrid. We've used the neurogrid in human patients. We even had a little paper with those guys showing that if you squint your eyes, you can kind of see something that looks like spiking responses. We'd love to use that tool moving forward, and maybe others that can emerge in order to try and see what's going on at the single cell level. That would be absolutely a gold mine. It's a great question.
Someone wanted you to clarify again whether you successfully were separated speech-related signals from motor-related signals, and a follow-up question from that is also for your hypothesis 3, where you show people, you ask them to reproduce words, like say, now say ba. Could you get the activity in Broca's area further down if you first gave them the command? So in the following, I'm going to give you 10 sounds, and then you say ba, la, da, rather than always giving them the entire sentence for hypothesis 3.
Right. So I think we really sweated the first part of that question, sweated that detail. So we cared very deeply on whether this thing was generic premotor, because Broca has really been implicated in so many different things. What I can say, and I'm only showing a kind of tip of the iceberg of our evidence, but as we've quantified everything, it's really heavily linguistic. And the responses are maximal in these more involved linguistic operations. Now, one could ask the question in repetition trials, if those became much more difficult, would we tax the system enough to get a robust response? And the answer is we don't know. But importantly, when we do stimulate the system and try and perturb it with focal current, as I had mentioned at the end of the talk, we never have been able to keep the patient volunteers from correctly repeating things. What we see are really selective errors in these linguistic operations. So if we stimulate during a repetition trial, we don't see any errors in that case. And so it suggests that it's not using the system as vitally. I hate to wave my hands, but everything is pointing to the fact that it really seems to be tuned towards more linguistic operations. Yeah.
When you give them repetition trials, so you ask them the same to reproduce the same sound or any kind of these tasks, do you see exactly the same activation patterns both in Broca's area and in the motor cortex areas, or are they?
So no. So when we look in motor cortex, we see really robust activity, right? But not during planning, right? So this is, I was a little quick with the details on these specific plots, but this is timing relative to what we consider to be a kind of critical word in these questions. So for example, timing with respect to when they hear the word ba, this planning stage. But if we look at responses in motor cortex as they're saying the word ba, they're really robust, right? It seems to be context independent. They need to move their articulators around, and indeed this is just as robust saying ba as it is saying the word goose. But I think that's actually a really nice thing to dig up, analyze, and to show as a kind of counterpoint that motor cortex is similarly engaged in all of these tasks as long as articulation is happening. Yeah.
Do you think Broca's area produces just a spatially complex signal? Is it like up-ramping to steady state rates, and then when the word gets executed, it just goes back to baseline? Or do you think there's also a temporal dimension already in Broca's area?
Oh, that's such a cool question. Oh, yeah. Sorry, please. In other words.
No, go ahead.
I would love to know, right? So, I mean, I think what looks like, I feel like I'm in some sense pointing at what appears to still be a controversy in the LIP literature in primates for those who follow that, right? So are things a ramp, or is it just a series of steps? And so I think what I can tell you here is that When we record using electrocorticography, and again, this is looking at the roar of the crowd, right? I think what we see is something that looks like a ramp. We would love to know an individual cell level how that works. This is persistent activity, and a number of people from Ranulfo Romo to Karol Swoboda have looked at the, and we've looked at it too in the songbird, right? What are the circuit mechanisms that give rise to this kind of persistent activity? And the songbird, it's a clear sequence of premotor commands. We don't know what's going on, and frankly, it's consistent with any number of models that attractor-like models that have been proposed. And until we get single neuron resolution, we really can't say.
Do you have any data from people doing 2 or 3 word production where they, or longer, where they're running out of working memory in Broca's area and have to renew during the production?
It's a really great question. So one thing we have a lot of is free conversation. So I've shown you the New Mexico, this kind of thing. And that specific example I showed you was really very fast turn taking. But we also have some really beautiful data where a person is answering, and of course, not everything is a one-word answer. For example, I'm giving a multi-sentence answer for your question. And we see huge planning signals that happen in the unfilled pauses before individual sentences. So even though a person is engaged in 40 seconds of talking, you can see planning bumps before each piece of this much longer, more elaborate response. And so we're working with—
Then no planning during speech production?
It's very little. It seems to really happen in the gap right before onset or in these unfilled pauses during that speaker's turn, but it's an elongated turn. And so there are people who have spent a lot of time thinking about this. We're working with an incredible linguist at the University of Michigan, Jelena Kripa-Kopek, and she's diving boldly into these conversational exchanges and trying to piece that apart. We've also spoken to some people who use machine learning to try and see if there's any kind of facial movement at all that could maybe be a tell for planning. And so we're going to do recordings as well as really fine-grained facial analysis to see if there really could be potentially a physical manifestation of planning to have as, in some sense, a physical ground truth, if you like.
Here's another question from the audience. Would inferior temporal areas have a role in your model? I was wondering for your hypothesis 3, if you give people a sequence of non-linguistic speech, would they show an interaction between the visual word form area, as the visual word representation of that non-linguistic speech can be activated, and the auditory motor planning areas of the brain, asks Joao Arajo. I'm sorry for butchering that.
That's a wonderful question. We have far less coverage over temporal regions, and so we would love to know. We've done enough recording down there to get really beautiful auditory responses. Of course, these are where primary and secondary auditory regions are in places like Wernicke's area, which has kind of complex auditory processing. We would love to know. I think given what's known about IT, there very well could be a really exciting relationship there. And I think, in some sense, with human patient volunteers, we really respect their time, and we respect that they've taken 20 minutes out of this procedure to help us understand how the brain works. But we kind of catch as catch can, right? The electrodes go where they need to go for medically relevant reasons. And I'll keep you posted as things roll in from temporal lobe. Yeah.
All right. We have another question from the audience, and it took me a little while to parse this, but I think I understand. Can you talk about individual differences or variations between different individuals answering the same or doing the same task, specifically in Broca to motor response? And how much does it influence the turn-taking dynamics when you combine different versus similar subjects?
such a cool question. So I can tell you that one thing we did almost as a gut check is we created functional maps using a GLM to parse and identify planning regions across all 8 of our patient volunteers, and they really line up. So we see the same rules emerge across each one of them, and that gave us a good amount of relief in terms of really seeing a robust effect here. But there are, I'm glad you brought that up about individual differences. There are people that appear to have big planning bumps right before they start talking, and often during free conversation, for example, they have longer lags. Others will have big bumps while you're talking, and they have much shorter lags. So it seems to be that I think has been a pretty clear individual differences rule in terms of where we see these dynamics start to really bubble up. Some people, and it could just be in their specific situation of being during a kind of neurosurgical procedure, that the planning dynamics happen late during that gap, and also the delays are longer, significantly longer in that case.
Cool. Michael, we're out of time, so I'm gonna just swallow the rest of my questions, or maybe ask you offline sometime, maybe in person. For now, thank you very much for the lovely talk.
And please, if anyone has any questions, please email me. I'm very responsive to email, and I really appreciate everybody being here. Thank you for taking the time.
Lovely. Have a good afternoon.
Take care.
Bye. We're not being cut.
Yeah. We can finish it. You as the co-host, yeah. All right. Thanks so much.
Okay.
I will. Yep. We are now live on YouTube.
Excellent.
Here's the chat. Here's the YouTube link if you guys want to share it with anyone.
Yeah, cool.
Nice.
I'll let in people know. Or I'll wait for Catherine for that.
So you can see the waiting attendees that are waiting? Or,
No, I can't see that.
Okay.
Okay, it's the hour, so I'll just start right now. Sorry, Catherine.
She'll be back.
Yeah.
At some point. Let's wait for a second. Okay, people are trickling in. I will wait until they sort of stabilize a little bit.
Yeah, just the timing thing. We have 15 minutes per person.
Mm-hmm.
And
yeah, I think the attendees are still dropping in. All right, I'm going to start. Okay, welcome to the session, and we're in room 3, and we have a really exciting line of speakers today. And as someone who studied social dynamic coordination, I'm very excited to learn about your work. So before we get started, I want to remind everyone that there is a Q&A button at the bottom of your screen. Please ask questions during the talk, and I will relay your question to the speaker during the Q&A session. And please feel free to post stuff in the chat, and we're going to post extended information about the speaker in the chat as well, so pay attention there. And now I'm going to introduce my first speaker while he set up his slides. So our first speaker today is Oracle Banerjee. He's currently a postdoc fellow at NYU Medical Center with Michael Long. But he is starting as assistant professor at Cold Spring Harbor Lab from next Monday. So congratulations. And Oracle is interested in neurocircuit mechanism of ethologically relevant behaviors. For his PhD, he studied neurocircuits for sensory processing in a factory system.
And as a postdoc, he worked with an exotic rodent species, colloquially called the singing mouse. And he currently holds the world record for recording every action potential has ever been recorded from this species. So Orko, the floor is all yours.
Thank you very much for the kind introduction. I'm extremely excited to be sharing with you our adventures with the Alston singing mouse. They're native to cloud forests of Costa Rica. As a systems neuroscientist, I am interested in understanding how sensory information is processed by the brain to generate appropriate motor outputs. And typically, one way to study these in the lab would be to train an animal, for example, a rodent here, to associate a sensory stimulus with a motor action, for example, from the Saboda lab, where the pole position is associated with the direction in which the animal has to lick to indicate his choice. But we are in a session about vocal communication, and I would like to make the point that vocal communication is ideally suited to study sensory motor transformations. Because, for example, in conversation, you have to process the auditory information, and then after a stage of planning or cognition, the motor cortex or the motor system in general swings into action and you have to produce the motor output. And this can happen in a very dynamic setting and can go back and forth. And what's really exciting and interesting is that it takes roughly about 200 milliseconds, which is remarkably small. Now, then the question becomes that how can we study such dynamic sensory motor transformations in the lab? And this is where I get to introduce to you the protagonist of today's story, the Alston singing mouse. They are highly vocal rodents from Costa Rica and Panama, and they're remarkable in their ability to not only sing a stereotyped song that lasts over tens of seconds, but also in their ability to go back and forth, and that's how they communicate. So what I'm going to play to you now is a video that I recorded in Michael Long's lab at NYU, where you will hear the mouse on the left sing a full advertisement song and then a response from the mouse on the right. And mind you, they can't see each other or smell each other, but they can hear each other. Okay, here we go. All right, so now these mice can participate in this dynamic vocal exchanges hundreds of times a day. Here is just 1 hour of continuous recording from these two mice, mouse 1 and mouse 2. And you can see that they actually sing fairly good amount of songs over 1 hour. But if you look closely, you will realize that they're not singing randomly. So for example, here you see that mouse 1 sings a full song that evolves over maybe 7 seconds, followed by a response from mouse 2, which I'm showing in red here. And if you do the statistics and look across many such interactions over many pairs of animals, what you see is that typical latency is around 500 milliseconds, which is quite fast. Now, one hallmark of social behavior is their ability to change motor output depending upon what the conspecific animal is doing. So in order to show you that, I need to first describe to you how we can represent one song. So a typical song is a series of notes that evolve over tens of seconds with a very stereotyped progression, which is that notes that happen at the beginning of the song are shorter and they become progressively longer as the song progresses. And you can plot this as this trajectory plot where note duration increases as a function of time. And what I'm showing here on the right is that if you take 15 songs on a random basis from this animal that's singing solo songs just on its own, they're remarkably stereotyped. They are all on top of each other. However, the moment they can hear another animal and starts participating in vocal exchanges, that stereotypy is broken. And you can see that the song becomes shorter, even longer, and the rate at which the notes increase or decrease is variable. So I have shown you so far that the singing mice can participate in very fast vocal exchanges, and they can also modify their motor output based on what they're hearing. Okay, so with this background, I want to switch gears and try to understand what are the neural circuits that underlie such vocal communication in the species. okay, we need to begin somewhere. And Daniel Okobi, who was a grad student in Michael's lab before I joined, he did a very interesting experiment where he electrically stimulated large areas of the dorsal cortex, trying to record from EMG activity in the vocal musculature, and found that an anterolateral portion of the motor cortex, when stimulated, results in a robust flexion of vocal muscles. So we call this area orofacial motor cortex, or OMC, because it overlaps with the generic orofacial motor cortical areas in other rodents as well. Okay. So then one can wonder, what's the role of OMC in this behavior, which is called countersinging, this ability to go back and forth? So I pretended to be mouse 1 in this case and played back songs from a speaker, and mouse 2 was adequately fooled into responding very robustly. So you can see that mouse 2 songs are in red and they're responding quite robustly to the playback. But if I inject mucimol and inactivate OMC, their ability to respond to playback falls apart. And even in cases where they manage to respond, it has a much longer latency. I can quantify this across many animals, and it was quite a robust effect where the ability to respond back to playback was significantly hampered if OMC was inactivated with mucimol. I also noticed something else, which is that if I quantified the duration of the songs that were produced in either saline or mucimol, you could see a shift such that the song durations after inactivation were comparatively shorter. Okay. So these two observations, this ability to participate in sensory motor transformation, which got hampered, as well as the song duration distribution that got lowered, led us to make some predictions. So the predictions are pretty simple. One is that we should expect that there should be neurons in OMC that would be differentially modulated between solo songs and countersinging, if this area is actually important in countersinging. And second, that there would be neurons here in OMC whose activity should co-vary with song duration, because inactivating OMC changed the song duration. And in order to test these predictions, I collaborated with a grad student in the lab, Margot, to develop a recording system in which, with chronic electrophysiology with high density silicon probes, I could record motor cortical neurons for weeks, maybe up to a month. And what did we find? Here is prediction number 1. This is a neuron, and I'm showing you 30 trials where this animal, mouse 2, was implanted and he sang 30 songs. And this is a raster plot. And you see this neuron's firing rate tends to peak right about when mouse 2 starts singing. However, we can do a trick here. Not all trials are the same. Some trials are when the mouse sings spontaneously, so that those would be motor trials. There are some trials when the mouse responds to a song that I've played back, so those would be sensory plus motor trials. And then there are trials where I have played back a song, but the mouse did not care to respond. So those would be sensory only trials. And if we split the activity of this same neuron into these 3 contexts, I think you can see that they show context dependence. For example, during the solo condition, the neuron, again, fires just around the animal's start of song. However, during countersinging, when the animal is listening, the activity starts departing from the baseline around 10 seconds before the animal has to sing, which is this red line here. And now if you think that that reflects the auditory activity, it doesn't seem to be so, because the neuron does not seem to care about pure auditory stimulus, which is the green line here, okay? So it seems that neural activity in this region, and giving you an example, but it's true at the population level as well, is predictive of the decision to countersing. How about the second prediction? Well, the second prediction was that there should be neurons here whose activity should co-vary with song duration. And again, here I'm showing you another neuron. I'm aligning the activity of this neuron to the beginning of the mouse's song, so that's song start. And then I'm sorting all the trials such that the shortest song is on the top and the longest song is at the bottom. And perhaps you can see that the activity of this neuron tends to monotonically vary with the duration of the song. And let's make it more explicit. If you just make a PSTH of this neuron for songs of different durations, what you should see is that the PSTH seems to systematically vary with respect to the song of the overall song duration. Moreover, what really seems to be happening is that the short songs PSTH, when artificially stretched to match the song duration of the long song, seems to be very similar, right? So we have done quantifications to show this in a more quantitative regime, but the upshot is that the long song's neural activity seems to be a stretched version of the short song, okay? So then one can ask the following question. And okay, there seems to be some neural dynamics that can track this overall song duration, but how do you know if this neural activity is causal for behavior? We can address this question by actually physically cooling down OMC. And the idea is as follows: if you cool down OMC, the neural dynamics slows down. And if neural dynamics in this region has anything to do with the behavior, you should observe a concomitant slowing down of the behavior. And that's exactly what we see. So this is on the right, control song at body temperature, and it evolves around, takes around 7 seconds, but progressively with lowered temperatures, the song elongates in time, which actually shows that neural activity in OMC that compresses and stretches to match the dynamics of the overall song actually can determine song duration causally. Okay, so with that, I would like to conclude by saying that what I have shown you today is the behavior of the singing mouse and how they can participate in vocal exchanges with each other at a very fast time scale, and also their ability to modify their vocal output based on what they're hearing. So it's a legitimate sensory motor transformation. And through various perturbation experiments and electrophysiological recordings, I hope that I have convinced you that motor cortex is required for vocal interactions in this rodent, and there are socially modulated neurons in motor cortex that distinguish between solo versus duet songs. Moreover, neural activity in this brain region seems to determine overall song duration. And what all of these tend to suggest is that there is robust motor cortical control over vocalization in a rodent, and it's not restricted to the primate lineage. And before I end, I would like to sort of advertise how interesting the singing mice species is as a model system, where you can ask fundamental systems neuroscience questions of how you can go from behavior to physiology and manipulations using viruses in a sensory motor transformation, but then also ask evolutionary questions of what changes might need in neural circuits that allows for different behavior to evolve along similarly related species. So for example, on the top here is mus musculus USVs, and we have 2 fantastic talks lined up that will explore that. So stay tuned. And in closing, I would like to acknowledge Michael Long, my postdoctoral advisor, for allowing me to work on this exciting project and for his critical insights over these last many years. And none of this would be possible without Steve Phelps, who is a long-term collaborator who pioneered this singing mouse model system. And if anybody in the audience is interested in pursuing some of these questions as a student or a postdoc at Cold Spring Harbor Labs, please feel free to either email me or contact me over Twitter. Thank you very much.
Thank you very much for a very interesting talk. We have many questions, so I will read them. And the first one from Brandon is, are these mice lifelong vocal learners like budgerigars? I'm sorry, I don't know how to pronounce that. Or is it crystallized song more similar to zebra finches? If they are vocal learners, any idea if OMC affects song and response quality or learning?
That's a great question. What we know so far is that their ability to produce these stereotype songs seems to be innate. And there's some experiments that have been done by Steve Phelps that suggest that. What is not known is how much developmental or plasticity that exists in their ability to countersing. So if countersinging itself is learned is an open question at this point. However, unlike the zebra finches and the budgerigars, their ability to produce this stereotyped song does not seem to require learning like it does for birds.
Thank you. We're a little bit over time, but a quick answer to this question. Thank you for the fascinating work. Do you plan on continuing to work with singing mouse in your own lab?
The answer is yes.
All right. Thank you. I will introduce the next speaker. Our next speaker is Cesar Vargas. He's a PhD student at Rockefeller University. He's very passionate about understanding how animals behave. He uses the lens of neuroethology to think about the evolution of brains and behavior across different species. So Cesar, take it away.
Great. Is my screen shared? Great. All right, so thanks for the introduction. So I'm gonna talk to you about how these vocal systems and vocal muscles that Arco started to introduce can, like in a very fine and sort of temporal manner, be described functionally in the motor cortex of mice. So movement coordination is a super intricate behavior, and here's an example of speech which requires coordination across multiple muscles and multiple systems, and is very complicated. So for a long time, people have wondered, how does the brain actually control our movements? And this goes back to the 1870s with Fritz and Hershig, and then famously with Penfield and sort of the idea of the homunculus, where you can stimulate parts of the motor cortex that will represent specific parts of the body. This has been extended to many different species. Here I have an example from mouse. Excuse the sirens outdoors, sorry. And more contemporaneously, there's been a lot of work on what Graziano and others have described as the action maps of the brain, rather than part of the motor cortex specifically just representing a muscle or a feature. It is sort of the gross motor behavior and sort of its full execution. So what does this look like for vocalizations? As I said, people have done these sort of motor cortical stimulations for a while. And here I have an example from macaque brain in the premotor cortex. The black sites are places that were stimulated that led to vocal muscle adduction. I'm not going to say full production. I don't think they did stimulations long enough to do that. But in any case, stimulating in these areas led to vocal fold modulation. We can use these studies to kind of estimate more or less the circuitry that underlies the vocal functions. So for example, in humans, we have two pathways for vocal muscles in the larynx. One is indirect, so it goes from the motor cortex to this green area called the reticular formation, then to the nucleus ambiguous motor neurons, then to the larynx. We also have a direct projection, which goes directly from the motor cortex to this orange nucleus ambiguous, and then to the larynx. Macaques, on the other hand, don't have this direct projection, is indirect. So it is exclusively from motor cortex or premotor cortex to reticular formation, nucleus ambiguous, larynx. And these have sort of been estimated with temporal latencies from stimulation in the direct projection setting from 7 to 20 milliseconds, and in the indirect setting where you have more neurons in between, 20 to 40 milliseconds, or generally just short and long latencies. There's been a lot of work in humans trying to describe this latency, trying to understand how we have such fine control of speech. That study I just showed from macaques had a longer latency, so it makes sense, right? They only have an indirect pathway. And as Arco just showed, in an orofacial muscle used in vocalizations, there are also short latencies in mice. So here's just an example from a recent paper from Eddie Chang's lab, where they stimulated human motor cortex. And despite the differences in voltage or even amperage in other studies, the temporal latency doesn't change, only the magnitude of the effect. So in humans, we can pretty much estimate about 10 milliseconds or less. In other muscle systems, so for example, the macaque, which has direct motor neuron projections for muscles like the bicep and the fingers, you do also see these shortened latencies. The mouse whisker pad seems to have interesting situations where you have two different types of latencies, a short and a long, which is very curious. So why mus musculus? Why am I not doing this in other animals? Well, as Arco very nicely introduced, mice have very cool and intricate vocal behaviors. Here you see a bunch of different syllables. You can see they have a lot of variety and modulation. We consider them to have some sort of sequencing, and there's analogous sort of construction, not analogous, sorry, but across different contexts, these sequences can change. And I think Catherine will talk a little bit sort of that temporal regularity that's important. But to my talk and my study, there's this really interesting thing that our lab discovered, is that when you use a retrograde transsynaptic virus in the laryngeal musculature of the mouse, you actually find a pool of motor neurons in the primary motor cortex. And using anterograde and retrograde tracing from M1 and the larynx again, you see that these there's sort of a convergence of the fibers at the ambiguous motor neurons that project to the larynx. So this was a pretty cool idea that maybe there is something going on. Mice have actually pretty fine control of their vocalizations. So to sort of start to functionally test this, I'm using intercortical microstimulation with paired EMG. So here's a quick sort of cartoon of the potential vocal motor system that I just described. And I use stimulation of the cortex with EMGs in the laryngeal musculature, and inspired by ARCO study and another recent one from the Kleinfeld lab, I think it was last year, describing the orofacial motor cortex. There's pretty good reason for us to look here as well to see if maybe the larynx was also represented elsewhere. And quickly, so I'll have this stimulation pattern, and then there's going to be a muscular response, and the time between those two is what I'm considering the latency. So just to give you a quick sense of an orientation of where I stimulated, it's this orofacial motor cortex, which I'm abbreviating as OFC rather than OMC, sorry, ARCO, and LMC in green, which is where we located our laryngeal muscles. And again, 4-pole stimulation that lasts 8.5 milliseconds. Those other studies, which I showed you from humans in particular, use different stimulation parameters, which is probably what leads to some of that variation that we see in the latencies described. So let's get into it. So if we look at the cricothyroid muscle first, you see that if you stimulate in the orofacial motor cortex, you do actually get responses, which is really awesome, even though this is not where we necessarily anatomically traced some of our neurons. But when we do go to the LMC where we did trace neurons, the response is actually slower. And so sort of where I consider the onset of the EMG to be happening is marked in the red diamond, which is just 2 standard deviations above the mean for this trace. I'll just sort of clarify that these are rectified traces. So that's why they're all sort of absolute values. So this was just kind of an interesting thing. The LMC is where we found potentially direct motor neurons, not the anterior regions. What might be going on here? But I also recorded from the digastric muscle. Same pattern again, the anterior motor cortex showing earlier responses than the more posterior regions of motor cortex. And just to kind of give you a broad sense of what many of these traces look like in population, it's pretty clear that the more posterior laryngeal motor cortex has a much slower response than the anterior orofacial motor cortex. And if we quantify the population of these latencies, very clear again that there is quite a difference between the 2 regions. I'll note that the LMC does have quite some variety. There are some earlier and some later responses. We'll just need to investigate that further. And some of it does happen to be a little bit of noise, especially the cricothyroid muscle. There's sort of breathing rhythms that'll sort of get recorded, and those are hard to parse out of the signal. So just kind of an overall sort of summary is that the orofacial motor cortex has earlier responses than does the laryngeal motor cortex in 2 different muscles, both used in orofacial behaviors. So again, to kind of recap, our hypothesis was that we would have direct projections based on our anatomy, and we could measure this at short latencies. This was sort of our expected model, but based on our results, perhaps there is another set of direct projecting motor neurons that we did not capture for technical reasons in our transsynaptic tracing. Another possible model that I would like to sort of investigate and think there might be something to it is that there's a difference in the inhibitory control of these 2 regions, where the laryngeal motor cortex has sort of the central and sort of canonical M1 has a lot of inhibition that we're having to surpass compared to the OFC. Therefore, you have sort of a slower latency as the circuits try to build up enough activity to discharge. Alternatively, both regions, again, for technical reasons, OFC would not have been captured, but also has direct projections. LMC it's not necessarily a difference of inhibition, but rather the LMC needs to raise the subthreshold voltage of the ambiguous motor neurons for then the reticular formation to provide firing, whereas the OFC could have more direct control through stronger synapses. These are just kind of proposals that I'm throwing out. Obviously, we need to investigate these. And for the OFC, I'm just going to throw out that there is this really cool early study from 2005 where they showed that in the anterior motor cortex for whisker control, rats and mice later have been shown to also have this. There's a direct projection to the facial nucleus, which is the motor nucleus for whisking. So this would not be the first time that we have an anterior direct projecting circuit. So I'd like to thank people from our lab, particularly rotation student we had, Elena Wademan, who helped collect some of this data, and Michael and Arco, who are collaborators and have been very helpful in sort of conceptualizing a lot of this study. And for the Americans, please go vote.
Thank you very much for the talk.
Sure.
And let's look at if there are questions. Please post your questions to the Q&A box. And while we're waiting, I'm going to ask you something. So what is the functional outcome? What exactly is the latency is representing in a functional sense?
Right. So that's actually a really cool question where, as I've gotten sort of thrust into this literature, it gets more and more confusing. Presumably, you could say that the sort of vocalizations in particular have very quick dynamics. As Arco described even, right? Like exchanges can happen in less than 500 milliseconds. You want very quick control of that. Mouse vocalizations can happen in less, or sort of these modulations are within 100 milliseconds. So you need very tight control of the vocal production and vocal output. And again, you're coordinating a lot of muscles at the same time. You don't want things to trip on each other. So that would sort of be my guess. But then where I say it's kind of interesting is in human studies, for example, stimulating awake patients seems to have a slower latency than anesthetized patients. So why that happens, it's kind of hard to tell. Maybe there's some sort of cognitive top-down. Obviously, the top-down, it's always sort of what we use as an excuse for things not going the way we think, but that would be my proposal for that.
Nice. There is one question from Dari. Is there any spatial organization of the neurons controlling the larynx? I'm sorry, in either OFC or the area you found? Are there cell bodies relatively close together within these areas?
Yeah, it's really hard to tell right now, mostly because, as I said, for the anterior region, we had not traced these. So we're going to have to come up with other, and obviously there's like delta G and some other cool tracing techniques that we can use. We use pseudorabies virus, just to kind of clarify that technical point. Yeah, we have not yet, not that we haven't been able to, we haven't yet done any of the spatial mapping to see sort of where along the ambiguous, for example, or where in the cortex. What is interesting though, that when I showed some of the anatomy to somebody who works in mouse forelimb, he immediately thought that where we traced the LMC was actually going to be forelimbs until he read my poster and was like, wait, that's not what I thought it was. So there seems to be sort of a very tight packing within the motor cortex across different muscle systems.
All right, thank you so much. I'm going to introduce our next speaker while she set up her slides. And so our next speaker is Catherine Paradin. She's a Wellcome Fellow at University College London. Catherine received a bachelor and master's degree in biomedical engineering from EPFL in Switzerland. Her doctoral research at Max Planck Institute for Biological Cybernetics focused on neurosubstrate of vocal processing in the primate brain. Catherine, I'm really looking forward to your talk. Take it away.
Thank you very much. I hope you can all see my slides. So after we've had a few really interesting talks on the vocal production, I would now like to tell you about my work on vocal perception. So it's the other side of the coin. And so, as you know, we humans use complex vocal sequences to communicate. We rely on speech as well as nonverbal vocalizations to exchange information with each other. And at this point, it should not come as a surprise that mice also rely on vocal sequences to communicate with each other. And so mouse vocal sequences or mouse songs are composed of these discrete frequency and amplitude modulated elements that you see above the picture here that are all happening at frequencies that are way outside of the human hearing range here in red. So that means they're ultrasounds. And so an extensive body of work has shown that these vocal signals indeed carry, fulfill a communicative function and carry information that listeners are able to use. And so, for example, in the case of songs produced by males in response to female cues, we know that these songs are attractive to female listeners and that they actually increase the mating success of the singers. And we also know that listeners are able to extract information about the singer from these songs. However, what we didn't know for sure is which of the many acoustic features in these complex vocal sound sequences are listeners actually extracting and using in the context of natural behavior. And so that's what my work aimed at answering. And so the first step to doing this is to actually replicate, as many other people have done before, replicate this natural mouse approach response to intact or to the playback of these male courtship songs inside the lab. And so this is what you see here. So you see here a female mouse in this 2-compartment box that has openings for 2 independent sound sources. And so in an experiment like this, that as I'll talk to you in this talk, the behavior starts by putting the mouse in this silent and dark box. And so she'd have to explore a little bit in this baseline period. After this, the sound playback starts. And you'll see that we're exploiting these independent sound sources. So in this particular example, the speaker on the left remains silent while the speaker on the left plays back normal male songs. And then we track the position of the mouse during the playback. We can measure how much time the mouse spends close to each of the 2 sound sources, indicated here. We then, after brief periods of silence, we repeat the presentation of the same sounds in the second trial. However, the key difference between the trials is that we swap the playback sides. So in this case, you'll see that songs this time are being played back through the left speaker and the right speaker is silent. And so you'll see here that the mouse is starting to spend more time close to the speaker that plays back sounds compared to the silent speaker. And in the third trial, the mouse continues, in this case, to track this song playback speaker, even though it changed to a different part of the box. And so we do this for several trials, take these measures as time and transform these into a place preference index. And so basically a positive value of this index means the mouse displayed a preference for song over silence in this session. And so we did this with many different mouse listeners, and we found that the majority of mouse displayed a preference for song over silence in these sessions, and that the distribution of preference indices was positively biased towards a significantly positive value, as you can see here, suggesting that mice preferentially approach male song playback over silence over the course of one session, but also over the course of individual playback trials. And that it took, on average for this preference to emerge, it took about 20 seconds of sound playback. And so from here, we can now extend this paradigm to ask specific questions about the behavioral relevance of specific acoustic features for mouse vocal perception. And so what we did here is we kept the same behavior and one side playing back intact male songs, but this time we played back a competing set of acoustically manipulated male songs. And so basically this, this experiment can have 2 main possible outcomes. So the first possible outcome is that female mice continue to approach the male songs. Having the playback, the manipulated songs doesn't change their behavior. And this would be evidenced by preference indices that, remain different from 0 positive. And so such a difference in how the mice approach intact and manipulated songs would be interpreted as the female behavior being sensitive to changes along the tested acoustic dimension. Alternatively, we could have a different scenario where mice equally approach both of the sound types, and that would lead to preference indices centered around 0. And so observing no difference in the approach to intact and manipulated songs would be interpreted as mice showing a behavioral invariance to changes along the tested acoustic dimension. And so the first question we asked was, does the global structure of the song play a role in the female's behavior. And so one of the ways we addressed this was to look at sequential organization of syllables in the song. And so what we did here is we manipulated the order of the syllables in the song. And the way we did this was simply to take intact songs and then randomize the order of the syllable in the sequence. And so I'll play some snippets of these examples, of course, brought into your hearing range. So here's the intact song. Hope you could hear this. Obviously, they're lowered in frequency. And this is another portion of the song that obviously you have different syllables happening. And so when we when we ran this test on a bunch of different mice, we found that, as you can see here, there was no consistent preference for either the intact song or this randomized song, and that the distribution of preference indices were centered around 0. We also found no difference in the behavior over trial. And this suggests that, the order of syllables in the songs is not something that is really important for females' approach behavior, at least in this context. The next thing we tried was to manipulate the local structure of individual syllables. And we did this in a couple of different ways, one of which was to just remove the complexity of individual syllables. And so the way we did this was to replace each of the syllables in the song by beeps of equal duration. And so the beeps, the pitch or the frequency of the beeps was chosen to be at the most likely frequency found in these songs. And so you can think of it as a sort of Morse version of these songs. Here's an example again of the intact song and these pure tone sequences. And so when we played this to the mice, again, we found that while there was some variability in how individual animals behaved, again, these preference indices were still mainly centered around 0, suggesting again that, removing syllable frequency modulation is not something that strongly impacted how the mice responded to these songs. And I'm happy to talk more about why this is surprising. And so a third axis we manipulated the songs on was to the temporal structure across the songs. And so as you might have noticed, if you have a clever eye or primed by the earlier talks, and if you saw in the examples I showed you before, when we measure the time difference between the onsets of successive syllables, these values are not uniformly distributed across the songs we recorded or the songs in our sets. Instead, these intervals are actually centered quite tightly around a dominant intersyllable interval. And so this actually means that the syllables in the song happen at quite regular intervals. So it's suggesting that they're actually, the songs are quite temporally regular or have a rhythmic structure. And so we decided here to disrupt this temporal regularity in the songs. And so the way we did this was to adjust only the silent breaks between the syllables in order to make this peak, to attenuate this peak, or hopefully make it disappear. So we stretched or narrowed only the silent breaks in the song to create something that resembles more uniform distribution. And the result of this was a song that was temporally irregular, maybe as if a stuttering mouse has produced a song. And here's an example of this. So the intact song again, and that stuttering song. And so then we played these two sound types to the mice. And what we found this time is that the vast majority of mice, significantly actually preferred the song over the intact regular song over the temporally irregular song. And as a result, the distribution of preference indices was biased towards positive values. And what was interesting is when we zoomed into individual trials, it seemed to take about 1.5 minutes of these sound playbacks for the mice to actually show a difference in this effect of the level of population. So it seems that song temporal irregularity is something that matters for this behavior, and that females notice disruptions to this song temporal regularity. And so, to summarize, we find that of a few features we tested, song temporal regularity is an important acoustic cue for mouse vocal perception, and mice found regularity quite attractive. And so this is interesting because there's been a number of people who've suggested that these mouse courtship songs might have a function that would help listeners assess this fitness of singers. And we're in the context of mate choice, so it would make sense that females are selectively paying attention to something that would tell them about how good that male could be as a mate. And so regularity might be connected to fitness, because you can imagine that a fit male would be able to sustain longer bouts of vocal production and maybe better maintain regular breathing cycles that would then be reflected in the vocal production. And so another interesting hypothesis that my current and future work is going to test is, whether regularity in these songs actually improves both the perceptual detection of songs in noisy environments, but also, the signal to noise ratio or reliability of neuronal coding in the auditory system. And interestingly, the syllable rate in these mouse songs is quite it falls into a common frequency range that has been shown to occur as well in primates as well as in human speech. And so with this, I'd like to thank you for your attention. Thank Dan Bender, with whom I did this work, as well as Colombine Verza, who helped a lot with these experiments, as well as my funding sources. Thank you.
Thank you very much for the talk. It's very nice. That's why it's explain why musicians get more girls and better ones. So there is one question from Cillian. And do you think mice use spectral cues in other contexts and behavior?
Yeah, that's a really good question. And I think this is really, we know mice here can be trained to detect and report, spectral differences. We know that if you train a mouse to tell two syllables apart, you can do it. We know that if a mouse can tell apart a syllable and the pure tones I showed you. So, with training and in different contexts, the perceptual ability is there. So here we're asking specifically in this context whether that is used, and it doesn't seem likely. I mean, it doesn't exclude that in other types of contexts, these spectral cues would be used. And I think importantly here, all my manipulations on syllable shape, we're still keeping the energy of the sounds in the same frequency bands. So, for various reasons of, perceived loudness and, peripheral sensitivity. But it's very possible that it is helpful, certainly for discriminating, for example, songs from adults that are higher pitched than, surprisingly, songs from pups. So certainly spectral cues, play a role, and perhaps not necessarily in the way we want them to play a role, but they certainly have a role to play there.
Thank you. And the next question from Isaac. Did you test changing the pitch of the songs? What do you think could happen if you change it?
Yes, so I have actually started before we had all these disruptions to the lab work. I had tried some experiments where I changed the pitch of the song, where I tried to change the song so much that we actually fell onto a different frequency range, which would presumably not activate the same neurons. I don't have super conclusive results on this. I think what would happen is at some point, the sounds would almost be perceived as a distinct category, because maybe changing the frequency or the pitch of these songs so dramatically could actually make it sound like either they come from an enormous mouse, or just generally be other sound altogether. And it's a little bit difficult to know what to know what the hypothesis for this would be. But that's a good question.
Thank you. We have a lot of questions. I will try to okay, I will try to ask them as many as possible. Fabian says, thank you. How about syllable density? Did you also took a look at the a potential preference for faster versus shorter songs? Or
yes.
Shorter intervals.
Yes, so I didn't test this formally here because I was really trying to, because of the nature of my behavior, I was really trying to keep the song length constant because otherwise it would be trickier to do the analysis. And, it's from what we know about rate of vocal elements in, certainly in monkeys and, possibly other animals, faster is better. Even so fast, if it's not natural, is still better, because you would have, in some cases, a phenomenon of temporal summation or cloudless summation that would just make a faster rate of songs sound louder and therefore more noticeable. So I think, again, these things, doing a test like this would involve actually balancing out the perceived loudness, and I'm not completely sure how to do that. Yeah, but my guess would be faster is better. Yeah.
And Yasser said, very nice. Did you try to extract the discrimination index across different delays across syllables? And I imagine the fitness of the animal would only slightly vary such delay across syllables.
I'm not sure. Maybe can you come on screen and clarify that a bit for me? I'm not sure completely.
Let me see if I can.
Is the question maybe that regularity would not be an all or nothing thing, but maybe something that would vary over the course of a song, or yeah? I'm sorry, it's hard when I can't see the question written. So. Oh, I can see it written actually.
Yeah, hi. Can you hear me now?
Yes, I can.
Very cool. Now I'm just wondering. So you measure the distance across the syllables, right? When you put I think B together, right? Then there was this preference for A, B, and then you also mentioned about this could be a mechanism for or an advantage to select for higher fitness, and I imagine that the fitness could only slightly vary this distance in time across syllables. I wonder if you measure your discrimination index with different delays between syllables rather than none versus the physiological delay.
Yeah, so what I'm doing here in this manipulation is certainly not, any of the manipulations are not physiological manipulations, if that gets it asking. Yeah, so I'm guessing the question I hear in what you just said is, what is the individual variability? How regular, is there a difference between some singers that are just more regular, more often versus others?
Yeah.
Here, I'm doing something that is not regular. I just shuffle this within some constraints to keep the song the same length, and I have, some forbidden intervals.
True.
So it would be actually interesting to see, if this is what we naturally find. Do we have then subsets of very regular, less regular males, and to what extent would females discriminate between this? I think that might be very subtle differences that might not be picked up by that kind of natural behaviorist assay, which relies on the goodwill of the females. But it's certainly an interesting question that could be answered with neuronal recordings, and I'll certainly try to do that going forward.
Thank you very much. I think we're gonna get booted out of the room very quickly. And so I just want to take the last minute to thank all our speakers, and thank you so much for the fascinating talks, and thanks for the participants to ask so many good questions. I took a screenshot of the question that hasn't been answered. I will send them to Catherine.
And where to find all of us. Thank you very much.
All right, thank you. Have a good one.
I'm gonna let people in yet. We're live on YouTube.
Are we ready?
Are we? Yes. Should I start presenting?
No. Hold on.
I will start. Yeah.
Yes.
I will introduce you. Okay. Let us start. Welcome everybody. This is interactive sessions by three amazing researcher fellows today. I am Ruzbe, postdoc researcher at UPenn, working with Conrad Cording, and it's delightful to have these people today. This is interactive. You can ask a question from speakers. You can use Q&A button or chat, or as we learned today, you can use YouTube also to ask your question if you are live streaming. In any case, if we have time, we definitely go to your question. Officially, our speakers have 5 minutes, so we have plenty of time to ask a question if you have. Thank you for joining today, and let me introduce our first speaker. Our first speaker is John. He's a postbachelorate researcher working with Dr. Ariel in the Department of Psychology at the University of Washington. He works on analysis of diffusion MRI, and is developing software called PyAFQ, which is automatically models the white matter of the brain and extract diffusion measurements along major tracks given diffusion MRI data. The talks today are almost all about the MRI. So if you are working on this stuff, tune in for a bunch of information today. Thank you again, John, and we are looking forward to your talk.
All right, so hello, my name is John Kruper. As you said, I'm a research scientist at the University of Washington, Department of Psychology, and I'm here to talk to you about a software called PyFQ. So why study the white matter? The white matter is composed of myelinated axons that connect different brain regions. It can be used to detect, characterize, and monitor diseases like MS, Alzheimer's disease, and schizophrenia. White matter tissue properties can change dramatically during experiences of intensive learning and could account for variances in cognition between individuals. To study the white matter, we use Diffusion MRI, which provides non-invasive and in vivo measurements. All right, so to compare brains across subjects, you could try to register all the brains to a common template and look at the tractography, but each brain has a different 3D geometry. So we need to use our knowledge of anatomy to segment tractography into known bundles. The idea is summed up in the title of a paper by Schilling et al., quote, brain connections derived from diffusion MRI tractography can be highly anatomically accurate if we know where white matter pathways start, where they end, and where they do not go. All right, so what is PyFQ? PyFQ performs tractography, registration, segmentation, cleaning, and profiling automatically. The first step, called tractography, is shown on the left. In the middle, there's the right cortical spinal tract, which is the result of segmentation. And on the right, we have we extract estimates of the microstructural properties of the tissue along the length of that bundle to create a tract profile. So PyFQ performs all of this automatically. A visualization of the 18 bundles segmented during tractometry is shown on the right. PyFQ is also flexible in which parameters and algorithms it can use. There's an existing version of the software written in MATLAB, but we use Python because Python is open source and free for anyone to use. It's a scripting language, so it's easy to develop in, and it's becoming the lingua franca for scientific computing, used from LIGO to Google's TensorFlow. Here's some example results. We used PyFQ in 44 subjects from the Human Connectome project to segment 18 bundles. The track profiles are organized by their anatomical location, and in the center, a select sample bundles are shown in their anatomical locations. For example, on the bottom center right image, there is a dark purple bundle called the forceps minor, and its corresponding track profile is also shown in dark purple on the bottom of the slide. In the track profile plots, the mean track profile has a large width, and individual track profiles are plotted with a smaller width. So one question we can ask is, how replicable is PyFQ? We use the HCP scan-rescan dataset to test the replicability of the profiles produced by PyFQ. In these plots, we use two different kinds of reliability. One we call subject or intersubject reliability, and that shows individual differences in the means of the track profiles. There's also profile reliability, which shows how reliable the shapes of the profiles are. Both are measured as Pearson's R correlation coefficients. We ran this using 3 different configurations of PyFQ, each using different models of tractography. On the top row, we compare the replicability of diffusion tensor imaging, abbreviated DTI, on single cell data to the replicability of diffusion kurtosis imaging, abbreviated DKI, on multi-cell data. On the bottom row, we compare the replicability of constrained spherical deconvolution, abbreviated CSD, and DKI, both on multi-cell data. And on the left are subject replicabilities, and on the right are profile replicabilities. What you can see here is that all of the methods we tested were very replicable. Another question is, how robust is PyFQ? So on the right, we show all of the different decisions the user can make when running PyFQ through the command line interface. Each diamond in box includes 1 or more decisions, and you're not expected to be able to read all of these decisions right now, because there are just know that there are a lot of them. Because there's so many different ways to do tractometry, we need to consider how robust tractometry is to different choices. To quantify robustness, we calculate reliability across methods instead of across scan-rescan data sets. On the left, we show the subject reliability across different tractography models. On the top, we see high subject robustness when comparing the results of PyFQ run with DTI when compared to DKI. And on the bottom, we compare DKI and CSD and see that the subject robustness is less. This makes sense because DTI and DKI are very similar models, where CSD is different. All right, so how does this integrate with the wider Python ecosystem? Various softwares are used for diffusion MRI preprocessing, shown on the left. PyFQ relies heavily upon DiPy to perform its analysis and can be run on Amazon Web Services using CloudNaught, shown on the bottom. And the results of PyFQ can be input to AFQ Browser or AFQ Insight for further interaction and analysis. That's shown on the right. PyFQ version 0.5 has recently been released. It includes several built-in registration templates and techniques, as well as the ability to provide custom tractography and registration templates. It includes a variety of new tractography methods. We also integrated the full 80 bundle template for use with Rico bundles, as well as other improvements and bug fixes. All right, so here's some discussion questions that I'm interested in. At the bottom, there's also a link to an interactive visualization of the results of tractometry, which can be automatically generated by PyFQ. It may take several seconds to load. Thank you for listening to this talk.
Thank you so much, John. That was a great package. And by the way, the link to GitHub and this tiny URL are in the chat if you want to look at the interactive version. Is there anything else that is useful for working with your package?
Yeah, so you can download this package using a pip install, and it will install all of the dependencies.
So in terms of like the models, it seems that you are backend on another package. Is that true? I saw another slide.
Yeah. So go back and show that.
Yeah, great. So how does it like how does it work? Like how is the workflow works here?
Yeah, that's a great question. So there's many different steps to tractometry, like I showed earlier. And for some of the steps, like tractography, we're sort of a wrapper around this other library called DiPy. We make the user interface a little easier and provide it as a part of this greater pipeline, but mostly we're calling off to them. For other steps like segmentation with the waypoint ROIs, that we do ourselves. And so it's sort of some of it's ourselves, some of it's with DiPy. And sometimes we move things from our software to DiPy, but we're sort of supposed to be this API where even if you don't know Python, you can use our command line interface to sort of run this on your diffusion data without needing to know Python.
Awesome. Yeah. So again, audience, if they have a question, there's a Q&A. You can jump in and ask questions from John. John, did you look at the timing? How is the time run of the package?
That's a great question. So it's very dependent on the data. So and which steps you're using. So on this HCP data, which is very big, it can take up to several hours. And that also depends on what settings you use, how many streamlines you want to do in tractography is a big one. And then for some other data sets, like I don't know if I showed it here, but there's a pre-K data set I've been working with a lot, and that can take less than 2 hours.
Awesome. Also, we have time. If you want, you can also look at the demo. It's up to you. But we have 5 minutes if you want to also show the audience how does it work.
Yeah, let me try to pull that up real quick.
How long you're working on this package?
I've been working on it for, I think, about a year now. Only full-time since the beginning of the summer.
That's awesome. How did it start? Oh, that's great. Okay. Do you want to explain?
Yeah. So let me see if I can expand what I'm sharing to show the legend as well. But yeah, we're very proud of this visualization that we automatically produce at the end. Each of these are different colored bundles. For example, that orange bundle here is the left corticospinal tract. They're shaded by the tissue property, a tissue property metric called FA, fractional anisotropy. So where it's brighter, there's higher FA. And in the background, there is the subject's anatomy in the slices. And if you can double click on a given bundle and then sort of wait a few seconds because there's a lot of data here, it will only show that individual bundle, and you can sort of zoom in and and look at it up close.
I'm amazed with the rendering time. It's so fast.
Thank you. Yeah, we're doing a lot of sort of resampling here to try and make it quick. And this is just an automatic output of the software. It's an HTML file that you can open up in any browser.
Right. Sorry, say again, how long it takes to have these bundles from the raw data?
Yeah, so from the raw data per subject, you can get to this visualization anywhere from a few hours to several hours per subject, depending on the data set. And we typically run these, I do on Amazon Web Services, and there's examples we have where you do that with CloudNaut. And then you can run all these subjects in parallel.
Awesome. Also, I just want to announce that if people want to ask a question, I can also let them to be on a screen to ask directly question. Feel free to join for discussion. So you use Amazon, right? For the
yes,
but it doesn't depend necessarily on Amazon, right?
No, you don't have to run this on the cloud.
Mm-hmm.
But if you want to run things in parallel, we're sort of set up to do that if you want to.
Awesome. So what was the motivation for this package? Seems very big, and right now it's like 1 year, right? 1 year old package. So
yeah, we haven't. We're still sort of developing it, and we're very close to like our 1.0 big release. I think it's very stable now. So I think the motivation was this was originally a package in MATLAB that did some of this, and we wanted to move it to Python because there's so many different tools for diffusion imaging that have been being developed in Python. So now that we have this Python base, we can add in things like in version 0.5, we added in particle filtering tractography and stuff like that. So we're sort of integrated with the wider Python world now.
Contribution to open source. Thank you.
Oh, I don't know if you can hear me.
Yeah, we can hear you.
Okay, I was curious. I wanted to ask what future directions you were thinking of going in with like incorporating new features into this package, or like research that you want to use with this package.
Yeah, that's a great question. So as you can see from some of the slides, I'm very interested in reliability. So I use robustness to talk about how different sort of different tweaks in what methods you're using under the hood might affect the results, and reproducibility and all that. And then in terms of future things we're going to add, we're looking at how we can align the track profile. So I didn't go into the details here, but when you extract a track profile out of a bundle like this one, you have to come up with some representative points to pull the underlying tissue properties out of to make the profile, and sort of how to align that, because there's many streamlines in here, and how to get one profile for this bundle is sort of a tricky thing. And right now we take this mean that's sort of a weighted mean that might not be the best.
Okay, cool.
Thank you again. I think that's Joseph. Thank you, Joseph, for coming here. Thank you again, John, for this great work. I know that there's so much work, coding, visualization, everything, but it works very, very nice and neat. Great, thanks. Let's go to the second talk that we have. We have Makarans. He's a master's student in computer science at the University of Michigan, and he's working with Dr. Jiang to use deep learning to reconstruct brain MRI images. He's interested in reinforcement learning and neuroscience foundation of AI. We are very happy to have him here. He is going to talk about deep learning for brain MRI reconstruction, expanding the unit. Thank you again, and can I start?
Yeah, so my name is Makaran. Thanks for the introduction. Yeah, I'm a first-year master's student in computer science at Michigan. And so I'm talking about some work that we did with using deep learning on accelerating MRIs to essentially make MRI acquisition faster. Using magnetic resonance imaging on the brain, or MRIs, can help with investigating, as we've seen, the structure and function of the brain, and even for neurological diagnosis. But the output of the MRI isn't actually an image, but Fourier domain data, or what's called a K-space. And it can take a very long time to acquire these scans. patients often have to sit for over 30 minutes completely still, and motion can add artifacts. And so it's cost prohibitive and expensive in time. And often modern MRI machines have multiple coils that acquire many different K spaces simultaneously. So how to reconstruct an image from the output of an MRI is we get these multiple K spaces, so C for the number of coils, and then we can inverse Fourier transform them to get C images. And then we can use the root sum of squares, which is essentially like an average, to compute a single image. And that's what's used for the final output. But acquiring these K spaces can take a long time. So what if we could undersample those K spaces and not acquire as much data? Well, if we undersample the K spaces, like as you can see on the left, there's a lot of holes where we're not acquiring data. If we try to inverse Fourier transform them to get images, we get these repeated artifacts and blurred noise, so that when we construct this final output image, it's not really appropriate for any sort of usage here because of all this noise. So the upshot is that we can use methods that rely on prior knowledge about what the reconstruction is, what the image is going to be. Like, for example, if we know that it's going to be a brain, we have some prior knowledge about what the image is going to look like, to try to construct an acceptable image using statistical reasoning. So machine learning, specifically supervised machine learning, we can use to train a statistical model if we have labels or correct answers to our question. So what we can do is create a model that can take in K spaces that are undersampled from all the different coils, output a single image, and then compare the reconstruction that that model does with the reconstruction that we would get if we use the fully sampled K space. So we trained these models on the FAST MRI dataset, which is an open dataset used for brain MRI images. And we can evaluate the quality of the images using the structural similarity index measure, which we can use to compare how similar two images are. Now, this isn't a perfect metric, but it does seem to correlate with what neuroscientists and radiologists seem to look for in terms of the quality of reconstructions. So a very common and popular model is the UNet, which is an image-to-image convolutional neural network, which is kind of put in at the end of this reconstruction pipeline. So the CK spaces turn into C images, which is then compressed into one image, and then is given to the model, and then which removes the noise that we can use for, and then output something appropriate. However, this doesn't really, this may be losing information from the multi-coil nature of the data, because the UNet only gets the input by the time it's already collapsed into a single image. So regardless, it still produces some quality reconstruction. So with 7 million parameters in our model, we can create a 0.92 SSIM approximately reconstruction. So if you look at these images, you can see the structure of the brain is preserved, but some of the finer details are lost. Now, I'm not a radiologist or a neuroscientist, so I can't really comment on how precise these reconstructions are, but I can see that some noise is added in reconstructing these. So we experimented with applying the UNet at different parts of the model of the image reconstruction pipeline. So our K-UNet, or K-space UNet, operates directly on the K-space data. Now, the UNet is intended originally for imaging data, so we wanted to see if it would work for the K-spaces, which is a very abstract domain. So the CK spaces come in, are fed to the model. We get CK spaces out, ideally with some with those gaps filled in, which are then turned into images and then final image. And we can see that the K-UNet produces images that do have artifacts, but achieve an SSIM around 0.86. So you can see that the overall structure of the brain is preserved, but there are some blurring and noise. So this is kind of like a proof of concept for being able to operate directly on the Fourier domain data. Next, we tried the I-UNet, or the images UNet, which operates on the multiple images directly and outputs that many images. So this is intended to hopefully take into account the multi-coil nature of the data before it gets collapsed into a single image, so that it may take advantage of the varying sensitivities of the coil in capturing. So the performance of this was around 0.74 SSIM, but you can see that while the brightness kind of varies in the reconstructions, some of the finer details of the brain are preserved, more so than the K-space unit did. So in combining some of these models, if we combine the K unit and I unit into a larger ensemble model, so where the CK spaces turns into, or goes into the K unit, and then is turned into images, which is then fed to the I unit, and then finally collapse into a single image, we're able to achieve images that have little noise, still don't have quite the precise detail as the targets, but are able to construct the images with the same structure of the brain. And this is around a 0.88 SSIM. Now, if we combine the I unit and the unit, so keep in mind that the original unit has 7 million parameters to it. If we combine the I unit and the unit, then we're able to achieve a performance a little bit better than the 7 million parameter unit with only 2 million parameters. So with 1 million to the I unit and then 1 million to the regular unit, so with like a 2/7 parameter cut, we can achieve an SSIM that's slightly better. And you can see in the images, the detail is preserved more or less, and the structure of the brain, and there aren't really too many artifacts. So this is like a promising direction in reconstructing images. So the kind of the conclusion here is putting the model in different parts of the pipeline may allow the model to take advantage of information that could be lost in, say, collapsing multi-coil data or the transformations involved. So multiple models throughout this pipeline could also help stabilize the training and the image reconstruction quality. So further experiments in this vein may involve varying the combinations of models used in reconstructing and seeing how we can cut parameter sizes to create a model that is lightweight yet is able to reconstruct images of high quality. So thanks to Neuromatch for hosting this.
Thank you so much, Makran. That was great talk. By the way, we are able to allow you to come for the attendees to come here and speak directly to Makrans. So if you want to do that, just ask us to give the permission. That was a great talk. Thank you so much. Do you know how, like you said about the UNet, KNet, and INet, do you know like how we can compare them if we have image, like which one we should choose? I'm pretty sure it's a little bit loose question. You don't know exactly, you need expert, but do you have any idea what's the best way to start?
Right, so what we used as a metric was the SSIM, which is, I think, originally intended for like regular images, not really medical images. So there isn't really a great metric for automatically evaluating reconstructed images, which is why certain, like a lot of these tasks will rely on actual radiologists going and looking at the reconstructed images and evaluating them for diagnostic quality. So there really isn't a great automated metric, but SSIM seems to be, I think, the most prominent right now.
I see. And do you use any trick or something for the implementation that you had for this work, like for this unit?
So this was implemented in Python with PyTorch, and we ran these on GPUs using our like University of Michigan computing clusters.
It's not pre-trained, right?
They're not pre-trained. We had to train them. It would take around several hours to train the models, but once trained, you know, it's using the models is very easy, especially with the lower parameter ones.
That's awesome. We have 4 minutes still. Feel free to ask a question. That's great. Do you have a GitHub or something to work on the images that you generated today?
Yes, I can link that in the chat.
That would be great. Yes.
I have a question as well, if you don't mind.
Sure.
Sure.
Well, first, I mean, I love the overall idea of using kind of anatomically informed priors to help your model. Since you're doing that, do you predict that the model would perform worse for kind of brains with lesions or tumors or something like that?
I think that's the real issue, is that because it's based on these priors, it might like perform worse for I guess outliers, but it's hard to get, I guess, data for that. But I suppose that I guess the thing to investigate would be, are those lesions and anomalies, like would they even show up in the case space that's input at all? Because if it's not, then there's no hope for the model to be able to reconstruct that. So I think if there is a suspect for some sort of an anomaly, then you'd have to maybe sample a little bit more or be a little bit more thorough with that.
Thanks.
But these were from the healthy data set, right?
For which data set?
The images that you showed, these are from the healthy subjects.
Yes.
Okay, I just had a question on the data set. Or do you know if there are data sets that are just more broad in the sense of having data from patients that have neurological disorders or maybe had lesions or things like that?
Yeah. So there really haven't been large-scale data sets for MRI reconstructions. Until very recently, the FAST MRI data set is kind of, I think, the largest MRI, brain MRI data set that's available. But in terms of those like specific things, I'm not sure of any large-scale data set that's able to do that. I think the one of the drawbacks of machine learning is that it does rely on large amounts of data, which is maybe hard to acquire.
Gotcha. Thanks.
Was that the FAST MRI, the data set you used
yes,
here, right?
FAST MRI, yeah.
And yeah, interesting. So it doesn't have the unhealthy data set. Thank you so much. With that, let's wrap up this talk. Thank you so much again for your great talk. And let's move on to the last speaker. That we have Adam. He's a data scientist, a postdoc fellow at the University of Washington, eScience Institute, and he received his PhD in theoretical nuclear physics and is transitioning to career in neuroinformatics. Currently, he works with Dr. Ariel Rackham at the University of Washington to develop statistical learning techniques and software pipeline for analyzing a large neuroimaging data set with diffusion weighted images. He's talking about multidimensional analysis and detection of informative features in diffusion MRI measurements of human white matter. Thank you. We are happy to have you, and can start whenever you want.
Okay, thanks. Well, hi, everyone. Again, my name's Adam Ritchie Helford. Thank you, Ruzbeh, for moderating and for the introductions. I'm a postdoc at the University of Washington, eScience Institute, and today I'm going to talk to you about statistical learning approaches for tractometry data. So I like to put my thank you slide first so I don't forget to thank my collaborators if I run out of time. Ariel Rochem is my advisor. I also work with John Kruper, who you met earlier, and Noah Simon at the University of Washington, as well as Jason Yateman and Manjari Narayan at Stanford University. You met John earlier, and I'm glad we're in the same session because it means I can kind of capitalize upon John's excellent explanation of tractometry. So just to review from John's talk, we're interested in the white matter correlates of certain diseases or behaviors. We know that the white matter isn't just composed of these kind of inert cables. In fact, these connections are dynamic, and individual differences in the white matter can predict differences in behavior. Diffusion MRI is a technique to measure the physical properties of these connections in vivo, meaning in a living human's brain. So the image there on the left is from Theodor Meinert's 1870s classification of white matter tract. So we've known about white matter for a while, but those are all derived from post-mortem dissections. Today, we don't have to wait for people to die to look at their white matter. We can use a diffusion MRI. And one can use these diffusion-weighted images to construct an estimate of white matter tracts in the brain. So there are many ways to do this, and John talked about some of them, but one way is to fit a diffusion tensor in each voxel, and then to simply draw streamlines through the major axes of these diffusion ellipsoids in a way that they kind of line up and give you the white matter tracts. This process is called tractography, and you can do it either deterministically or stochastically to get really large collections of these streamlines, and we call those collections tractograms. So these tractograms are actually really massively dimensional and rather unwieldy to work with, and that's what motivated the whole need for a tractometric pipeline, not a tractographic pipeline, but a tractometric pipeline that John talked about. And the main takeaway there is that tractometry represents this kind of massive dimensionality reduction from the tractograms outputted by tractography into tract profiles output by PyAFQ. And this is actual tidy data, tidy in the sort of Hadley-Wickham sense of the word. You can open these up in Excel or Pandas, and more importantly, you can organize them as a feature matrix that you can do machine learning on, where rows correspond to different subjects and columns correspond to the diffusion metrics along each tract. And that's sort of where the bulk of this talk begins. Our interest here is both on prediction of phenotypic data and also on identifying important features of the white matter. So in the language of our linear model, the phenotypic variables are encapsulated in the target array Y. The tractometric features are represented by the feature matrix X there. Each row of X and Y is a subject, and each column of X is a diffusion matrix at one point in the tract profile. And the relationship between these two is characterized by the coefficient vector beta. But diffusion MRI has this immense group structure too, and we should choose an algorithm that can capitalize upon that information. So here I've represented the group structure using color encoding. So all those orange features, maybe they're all nodes that belong to the corticospinal tract, and they should be grouped together because we know, we have an anatomical prior there. And all the green ones are from, say, the callosum forceps. So we want to use that information as we regularize these features. And we do need to regularize because we have way too many features. Typically, we have something like 10,000 features or so in a study with maybe 50 to 100 subjects. Sort of the paradigmatic regularization approach is to use the lasso, which is up there in the top left. It introduces a sparsity penalty to effectively zero out uninformative features. And then there are variations on the lasso for grouped covariates. So the group lasso in the lower left there can send entire groups of coefficients to zero. And then there's the sparse group lasso, which can enforce both intragroup and intergroup sparsity. And I'm purposely kind of moving quickly through the formalism here because I want to show results, but I'm happy to come back to this later if you want. So to test SGL, or the sparse group lasso, in a classification setting, we replicated results from a study by Alicia Sarica et al. In 2017. This study imaged 24 patients with ALS and 24 demographically matched healthy controls, and it identified which parts of the corticospinal tract are informative for the prediction of an ALS diagnosis. So in that study, they did a priori feature selection. They said, we know that the corticospinal tract is important in a motor dystonia like ALS. We're going to throw away all the other tracts and just look at the corticospinal tract. And then they identified which parts of the corticospinal tract were important. In contrast, we leave all the features for the entire brain in our model, and we let SGL determine the feature importance. Here we obtain a classification accuracy of about 93% with an area under the receiver operating curve of 0.978. And moreover, SGL selects the known white matter correlates of ALS. So here I'm showing those feature importances, the regression coefficients plotted onto the kind of the core fiber of the bundles. And we see that it identified the fractional anisotropy measures in the right corticospinal tract as important, which is kind of what we know about ALS. So it's a good test case there. We also tested SGL in a regression setting by predicting age in a study of 77 subjects aged 6 through 50 from Jason Yateman et al. Here we achieve an R-squared of about 0.3 with a median absolute error of 3.6 years in age prediction. And in contrast to the ALS case, where the important features were really sparse and localized to one part of the brain age features are dense and non-localized. And even though they're non-localized, it can be informative to look at the regression coefficients for certain bundles. So here, I've highlighted the left cingulum cingulate and the anterior callosum forceps. You see on the right y-axis, I'm plotting the fractional anisotropy. So those are actual metrics in our feature matrix. And on the right y-axis, I'm plotting the regression coefficients. That's the dotted line. And you can see the parts where the regression coefficients are away from 0. Those are important features. And they do indeed pick out areas where we can discriminate between the different age quantiles. In conclusion, I'm presenting here a novel method for the analysis of tractometry data. It can both make accurate predictions of phenotypic properties, but then also identify which white matter features were most important for that prediction. Can accommodate both regression and classification problems and describe phenomena that are locally confined to certain areas of the brain and also ones that are widely distributed across the brain. It's available as an open source software called AFQ Insight on GitHub. It's part of that ecosystem that John mentioned earlier. And in the future, we'd like to do multiple extensions. There are other sort of sparsity-inducing penalties that we could add, and also other domains that we could apply this to, domains in neuroscience that also have grouped covariates like fMRI or large-scale multi-electrode recordings. So I'm gonna stop there and see if there are any questions, and if there are none, if you want to know more about what the coefficients actually look like on the brain, I have some visualizations for that, but I wanted to stop for questions.
Sure. Yeah. If you have question, you can unmute yourself to ask question. I have actually one question. I'm waiting. If there isn't any question, I can ask my question.
Sure.
So I personally work with morphologies. They have like a tree structure with branching, all this stuff. And this is very exciting for me because that looks like kind of the tree structure that you're extracting from the whole data set. But seems that you are using the raw data as a for feature analysis. I was wondering if we have some kind of tree structure feature extracted from the data set, like angles, I don't know, whatever, looks like that. Have you tried that?
I haven't tried that. Yeah, that's a really interesting approach. So here our groups are kind of predefined, right? We just know, okay, if you're in the arcuate bundle, for example, you should all be in the same group. But you're describing more of a hierarchical grouping, right? Yeah. So there are other forms of the lasso that can accommodate hierarchical covariates, but I haven't investigated those.
Yeah, so hierarchical, and as well as for example, look at the geometry of the tracks, like say when they branch, and if they branch, in what angle they branch, or what is the length of the tracks. I saw on John's that he look at the length of the tracks as a one factor, but in what extent you can add the geometrical features to the analysis.
I see. You're looking to add those to the feature matrix, basically.
Yes.
Yeah, that could definitely be done. And then you could group. Yeah, I guess I would have to think harder about the grouping, and I'm not sure I want to do that on the fly, but I'm sure we could do that.
Yeah, that's very interesting structure. And I don't know how much varies you see across the subjects when you compare.
You mean in terms of the tract profiles?
Right.
Well, actually, I have. It depends on the problem, right?
Right. Yeah.
For example, here's a visualization of. This is AFQ browser. It's another tool that we're not presenting, but basically here we see kind of the 3D brain, and then you can plot your different tract profiles. You can select different profiles here, but because our algorithm highlighted the right corticospinal tract here, I'm showing the right corticospinal tract. Mm-hmm. And let me just take this opacity down. So now we're only seeing the group means. So in blue, we have the controls, and in red, we have the patients with ALS. And I've actually already gone ahead and picked out certain subjects that were misclassified by our algorithm. So we.
That is very cool. So the red one is for the ALS and the blue one is for the healthy one?
Exactly. Yeah, yeah. And so, I mean, what I showed in one plot in the slides is that our algorithm identified this region here of the corticospinal tract, and you can see it kind of highlighted there in the 3D window as, you know, discriminating between patients with ALS and patients and controls. And indeed, when you just look at it, you think, okay, yeah, you know, that makes sense, right? There's a difference there. But there is a lot of variability, as you hinted at, right? There are, like, these are false. What are these? False negatives, right? These are patients with ALS that we misclassified as controls. And when I look at it, I kind of say, okay, yeah, I think I would make that mistake too if all I had to go on was the tract profile.
Right.
You know, it turns out we do need doctors to make diagnoses. But it's also sort of reassuring that it failed in an interpretable way.
That is very interesting. Sorry, that might be a stupid question. So for this graph, the x-axis is the distance from the fiber. What is FA here?
Oh, sorry. FA is fractional anisotropy. So it's a measure of the sort of the diffusion ellipsoid. If it's perfectly spherical, your FA will be 0, and if it's a straight line, it'll be 1. So it's confined to 0 to 1, and it's kind of a good measure of white matter integrity.
I see. Interesting.
And there are all sorts of weird. I mean, maybe you get fiber crossings, and that affects FA. And John could probably talk more about that. But broadly, you can consider it as a measure of integrity of the white matter.
Yeah, that's interesting. Is that open source, this one that you are working?
Yes. Yeah. So the statistical analysis package itself is written. It's in 2 packages, sort of. One that does SGL, because SGL has nothing to do with neuroscience. So it's a package called Grouper. That's GROUPYR. I can put it in the chat.
Right.
If I can find the chat, sorry.
I can write it down if you want. GR, like a group, like as a group.
Like group and then YR. It's on GitHub under Richford. Here, I'll stop my share and then just put it in here. There we go. I've got it. And then the actual you know, neuroscience part of it is in AFQ Insight. And I have to warn people, I've done a pretty major rewrite recently, so it's still kind of beta, but yeah, it's all open source.
Now we all understand, I guess, the beta version.
Yeah, yeah.
Amazing. That is a great and amazing package that you're working. If there is not any question, we are going to wrap up this session. I don't see any question. Okay, great. Thank you so much again, all the panelists today. And that was a great talk. And I see a great future also for this package and works that you are working on MRI. Thanks again. And hope to see you virtually again in Neuromatch Academy. Have a good rest of the day. Goodbye, everybody.
Yep. Thanks for the talks.
Thank you.
Thanks.
Okay. And we are streaming live right now. As people filter in, we're just going to be doing a session we're in Zoom room 3. And we are going to start in a couple of minutes. So we're just very organized today. And while we're waiting for people to start, we'll start on the hour so that we allow people as much possible time as they can for their talks. But before we do that, let me just get started and get you guys who are listening in organized. My name is Ina Puce. I'm going to be your MC today. And I have the pleasure of having Siddharth Sudesh, who's doing the back end and helping us out there. And so he'll be monitoring for questions that come in from the audience. And so because we can't see questions coming in from you guys on YouTube, we can see some stuff on the chat, but we don't see everything. So he's going to be helping us there. So don't be afraid to ask questions. We're also putting up some papers from the speakers, as we did a bit of homework, and so we'll put some recent papers up, web links, so people can see some of their work. And we're just going to have a very interesting session. This is all on the humans. I just came from a session which was animal models, but this is all human models with some very, very interesting themes. We may as well think about getting going. Our first person who's going to be presenting is Marianne Rouault, and she comes from the École Normale Supérieure in Paris, and she's going to be talking about changing one's mind. And that's actually a very important thing right now, changing minds. And it's not as easy as we'd like it to be. And so she's got MEG data and some computational modeling data to tell us about this. So Marianne, take it away. It's all yours.
Thank you very much. All right, can you see the presentation properly? All right, so I'm going to talk today about changes of mind and information seeking. And this is a line of work that has been developed in the team of Valentin Viard at the INS in Paris. And the original idea behind the project was to compare different families of paradigm that are related to information seeking. So there is a whole literature on perceptual decision-making in which participants are passive observers dealing with sensory noisy information about which they have to make decisions. And this is in standing contrast against value-based decision-making paradigm, that is a whole other stream of literature, in which this time people make decisions based on action outcome contingencies that they learn by trial and error. And even if both types of decisions refer both to uncertain information, they are very different processes in terms of both the temporal horizon against which they unfold on the scale of milliseconds for perceptual decisions, whereas we are talking about the course of seconds or even longer in the case of outcome-based decisions. And critically, in perceptual decisions, we are adapting in response to external cues. We are passive observers dealing with uncertain information from the environment, whereas in value-based decision-making, typically we adapt in response to the outcomes of previous decisions. And so these differences make it difficult to compare the types of decision and information seeking across different paradigms. So we try to come up with a framework in which we could actually compare and ask how do humans learn and decide differently when they are based on the same uncertain information in these two situations. So I'm going to describe the framework that we have used. We are comparing cue-based and outcome-based decisions in tightly matched conditions. So we have two generative processes that refers to two hidden states, orange category and a blue category, that is going to give us uncertain information upon which subjects are going to make decisions. So in a particular trial, people will see a sequence of samples from 2 to 8 samples of evidence that come from a latent source, and they will have to form a belief about which is the generative source, the orange one on the blue one. So they'll see a stream of uncertain information, and they will be asked to make a choice about the category that generated them. But the critical difference that we introduced is that we created two conditions that are based on different levels of controllability. So first, in Q-based conditions, people will be asked to monitor the category from which stimuli were sampled. So they will, at the end of each sequence, they were asked to report a category choice together with a confidence estimate in their response. So just 4 response keys, high or low confidence for either of the 2 categories. So in the Q-based condition, they will be asked to monitor the source from which stimuli were drawn. And in contrast, in the outcome-based condition, participants were asked to select an action that will produce stimuli from either category. So they will be asked to sample from a target source determined by the computer. So this allows us to critically match the amount of uncertain information across conditions. But in one situation, people are in control of the evidence they see for decision-making, whereas in the other, the Q-based condition, which is more alike to perceptual decision-making paradigms, they are presented with evidence. And finally, we had a reversal structure. The target category reversed unpredictably after a number of trials, which requires subjects to adapt through constant monitoring of the category source. And so the key difference is the degree of control that subjects had over the evidence available for their decisions. So we first looked at the selection of category after a reversal. So you have in pink the outcome-based condition, in blue the Q-based condition. So you can see that after a reversal, subjects gradually learned to select more often the category corresponding to the true hidden state, but they were slower to adapt after a category reversal in the outcome-based condition as compared to the Q-based condition. So we have a higher reversal time constant, and they also reached a higher performance at the plateau level in the outcome-based conditions. So I'm pointing out here an original study on the same paradigm in which you have a bit more results, but I'm going to unpack what we found in terms of confidence after a reversal. So you can see that after category reversed, subjects were less confident, as you might expect. But importantly, there was a larger confidence drop in the outcome-based conditions. So subject confidence decreased even more when they were in controllable environments. So another important aspect of behavior that we've looked at is that here, so we are looking at the fraction of repeat choices, the proportion of time I repeat my previous choice, but this time plotted as a function of the evidence available for the decision. So we've recoded the evidence available in each sequence as a function of whether it confirms your previous choice or whether it is inconsistent with your previous choice. So you can see again, as you might expect, that as the evidence in favor of repeating your previous choice, people indeed repeated the category choice more often. But importantly, there was a difference between the 2 conditions in which, in the outcome-based condition, people needed more evidence to switch and to change their mind. So even if the uncertain information was the same, in controllable environment, people needed to switch more evidence to switch to change their mind, basically. And this is then explained, by difference in sensitivity to evidence across conditions. So the slope of these curves were the same. So people integrated the evidence equally well across conditions, but just they needed more evidence to change their mind in the outcome-based condition. And finally, the last piece of behavior that I have for today is again the fraction of high confidence responses plotted as a function of the evidence confirming or disconfirming my previous choice. So you can see that on the right part of the graph, where more often the evidence confirms my previous choice, then more evidence led to more confident choices with a similar sensitivity across conditions, a similar slope. But in contrast, in the left part of the graph, where more often the evidence leads me to change my mind, subjects' confidence were lower in the outcome-based condition as compared to the Q-based condition. And this is also revealed on this plot, which is the fraction of high confidence in all possible conditions, whatever the level of evidence available for my choice. So you can see that on repeat trials, the average confidence was the same, but on switch trials, the confidence was lower when I changed my mind in controllable environment. So we thought to see if we could explain the mechanisms by which these differences across conditions occur. And for that, we needed a shared computational framework. And that's the case in this task because the evidence people see on each choice is the same across conditions. So we have some sensory noise coming from the samples of evidence that participants are presented with that I need to integrate to form a posterior belief about which is the correct category choice. So we have some inference noise here that previous work has been shown to be critical when I integrate samples of evidence in this manner. We have a perceived hazard rate, which is a volatility measure for modeling the reversal between categories. And then this evidence, this posterior belief is taken to a choice process, which is simply a maximum here. So we have no selection noise, as that has been shown again in previous work. And that determines my category choice, and my confidence is simply read out from the posterior belief. So it's related to the strength of my belief about the category. So we fitted this parameter to subject choices and their confidence and simulated the obtained data. And here I just want to show you for today the best fitting parameter that we obtained. So importantly, we found that there was no difference in inference noise across conditions. So people were equally good at accumulating the evidence over samples. However, we found a lower volatility parameter in the outcome-based conditions. So it means that people perceive the environment as being more stable in controllable environments, that reversal were less likely to occur. And finally, we found a larger confidence threshold, indicating less confident responses in controllable environments, again, in line with what we observed in the behavior. And finally, we have looked at what happens after I change my mind. So I need more evidence in the outcome-based condition. I'm also less confident, but I'm also in the end more likely to confirm a change of mind on the next trial in the outcome-based conditions. So people are less likely to go back to their previous decision after a change of mind when they are in control. And to finish with, I would like to present a bit of neurophysiological evidence that we have examined to compare these different changes of mind across conditions. So here we are looking at decoding MEG data on switch versus repeat trials, in which we show a stronger involvement of a dorsal attentional network on changes of mind. So we found that for switch as compared to repeat trials, we have a larger amplitude and also a response that is more extended in time in the outcome-based condition. And you can see also that the effect was stronger. And it reflects, I'm showing here the network, so it reflects a larger involvement of a dorsal attentional network, which means that when there is uncertainty at the time of a change of mind, participants become more strongly oriented or paid more attention towards the evidence samples. And you can see that this difference is really driven by changes of mind. There is no difference between conditions on repeat trials. And the difference between the two conditions is also illustrated in pupil dilation variations on changes of mind. So this is again a contrast of switch versus repeat trials, in which we see an increased pupil dilation time locked after the response for changes of mind, and an effect that was again larger in controllable environments. So I hope that I've showed you today that in controllable environments, more evidence is needed to change your mind. When you do change your mind, you do so with less confidence. You are more likely to confirm a change of mind on the next trial. And we have neurophysiological correlates of increased arousal in this dorsal attentional network for changes of mind. Thank you very much for your attention.
Okay, that's his fascinating work, and I'm not seeing any questions here right now as yet, you know, if the other presenters don't have a question, I've always got questions. So, just a more conceptual question, and you know, you're looking at changes of mind, and I wondered about having a richer measure of behavior, because you know you've got a decision with a button press, And I was wondering whether you could do maybe something, or have you investigated using something like mouse tracking, so that if you've got a paradigm like you're using, instead of pressing a button, the person has to move the mouse on a trajectory to two positions on the screen. And that trajectory can be informative and itself can be interesting if someone's going to change their mind. And I was wondering whether that would be additional, a richer behavioral data set that you could use.
Absolutely, thank you. Yeah, that's a very interesting comment indeed that could give us another dimension to look at. I'm aware of some studies in which people have measured the mouse position as people go through their choices. So you can really see choices in which they change their mind and go to one direction and then to the next. To the extent of my knowledge, this has been only done in non-controlled environments. So as in the Q-based condition in our current paradigm. So I would be very interested to see if we can see different statistical signatures of mouse position when you compare such environments. That would be interesting indeed.
Yeah, that would be really, really cool if you could do that. Great. Okay, well, I think that we will move along to the next talk. Thank you, Marion.
Thank you
that was
much.
very, very interesting. And our next speaker today is also going to be talking about choices and the influence at acts of free choice and how they estimate both choice relevant and irrelevant attributes of the chosen item. And this is going to be a talk by Wojciech Zajkowski. So Wojciech, the floor is yours.
All right, thank you, Aina. All right, so choice-induced bias can be understood as an instance of confirmation bias, which is something that is very prevalent in our day-to-day life. For example, in social media, when we often surround ourselves with like-minded people and hear similar opinions to ours. In law, in evidence processing, where sometimes the verdict can be biased in favor of a pre-existing belief. In finance, investment overconfidence can lead to investment bubbles, or in science, where on all stages, from hypothesis generation to data interpretation. So today I want to talk about the specific type of choice-induced bias and distinguish between a within-domain and a cross-domain bias. And a within-domain bias is related to the domain of your belief. So take an example of a debate. For example, if you agree with policies represented by candidate A, you might also believe her arguments were better simply because of that. So that would be something that would be a within-domain bias. On the other hand, an across-domain bias would happen if because I agree with candidate A's policy, I also think she looks better, she is dressed better, has presented herself better. So this would be a bias in a domain that is only superficially related to the bias origin, or to the belief origin. So going back to decision making, we'll look at these biases in terms of preference and perceptual choices, on how preference choice can induce a change in a preference, or a perceptual choice can induce a change in estimation of perception. And on the other hand, similarly, an across-domain bias will be when a preference choice can induce a change in a perceptual judgment, or a perceptual choice can induce a change in a preference estimation. So the study design has three main parts. In the first part, participants rate 18 food items on 2 scales in terms of the value, in terms of the preference, and in terms of the size. The both scales go from 0 to 100. In terms of preference, you rate your preference, and in terms of size, you rate the item size based on the non-white pixels, basically. In the second part, so we get like a basic estimation of how much the items are worth to participants and how they estimate their size. In the second part, participants make a series of binary choices between all possible pairs of the items immediately followed by a comparative judgment between the two items. And there are three choice conditions, either preference, size, or no choice control. And there are two types of judgments, either preference or size. So you can have a within domain trial where preference choice is followed by preference judgment, or a cross domain trial where, for example, a preference choice is followed by size judgment. And based on this, we can generate some basic hypotheses. So we would assume that in the no choice condition, the estimations, the judgments will be centered at the true value, which is given by the initial ratings. But on the other hand, after, for example, a preference choice, we would assume that the preference distribution will be shifted towards the choice. That would be the within bias, but also the size distribution might be shifted towards the choice. That would be the between effect here, or a cross-domain effect here. And similarly, after a perceptual choice, we would assume that a size choice can induce a large shift for the judgment size, and a smaller one for preference. So and finally, there is one more rating of the items, just to see how the ratings changed from start, and how the task basically influenced the item estimations. So this is the main result of study 1 we did. So basically, as you can see, this is choice-dependent bias conditioned on the side the choice was made. So basically, as you can see, if participants chose the right item, the judgments were shifted towards the right. If participants chose the left item, the judgments were shifted towards the left. And you can see a much larger shift for the within trials compared to between trials. So something in congruence with the hypothesis. You can follow these data and get a summary of the main effect for the within and between in terms of size and preference. We were able to replicate this effect on a much larger sample in experiment 2 with a different set of stimuli and a shorter design as well. And we got the result, which is qualitatively basically identical to the first one. So there is a large within bias and a small across or between bias. We also did a series of additional experiments just to test additional hypotheses, although in the interest of time, I will not go through them right now, but you can have like a small glimpse on this slide of some of them. There are actually 5 in total. Okay, so next we want to model like the generative process, which can give rise to these biases. So the model we came up with is a model of which assumes that values fluctuate as a function of choices. So this is a single instance for a single item throughout the task, where the initial points are the initial values, which then fluctuate in time based on choices. The bottom dots represent the trials where the item was chosen, the green ones, or rejected, the red ones, and the value fluctuates accordingly. So more formally, just to see how this model looks like from the inside, it has 4 main parts. The first part is modeling the choice with the softmax function. So here simply the values are inputted to a softmax function to determine the choice. Then the values are updated based on the choice. So the chosen item gets a proportional bonus, denoted as a parameter alpha here, and the rejected item gets is devaluated proportionally as well with the proportion of beta. And you can have a maximum of 8 such updates based on the parameter and based on which values are updated, whether preference or size, and whether it's updated based on a congruent clue. So within the main choice, a congruent choice, or an incongruent one, so a cross domain one. Then the judgments are modeled as normally distributed, centered at the difference between the items. And finally, there is a decay effect, which is controlled by this parameter k, which basically is about how whether these estimations, whether these updates, how permanent are these updates. So it goes from 0 to 1. If the parameter is 1, it's no memory. So basically, after each choice, the values go back to the constant. If the value is 0, it's a perfect memory system where each update is permanent. And we compared 5 models. So the null model assumes that no values are updated, the values are constant. The within value model assumes that only the preference within, so only the preference is updated after a preference choice. A value model assumes that preference is updated after both types of choices. A within model assumes that only congruent clues are updated, so size after size choice and preference after preference choice. And the full model assumes that all the values can vary here. We did the model comparison. The model comparison looks like this, and the lowest scores here are better. So basically, the full and within model can predict the data equally well, which means we obviously should choose the simpler model, which in this case, the within model is a bit simpler. So the conclusion is here that the across-domain effects don't have an underlying effect on value estimation. They're just influence the immediate judgment, but don't don't influence the underlying estimations of value. These are the parameter estimations. From here, you can see that the devaluation is stronger than overvaluation. So basically, this effect is driven more strongly by devaluating the rejected item than overvaluing the chosen item, which is actually congruent with one of our additional experiments. And the decay is close to 0, which means the updates are close to permanent, at least based on the model. And the model can predict the data pretty well. It can actually reduce the congruent or within bias to 0. So this is basically what we would want a good model to do here. And it can also predict the final ratings more accurately than the initial ratings can. Okay, so that's, I believe that is it. Just a quick summary on what we found. We found strong effects of both within and across domain choice-induced bias in both domains, in perceptual and preference domains. The choices needs to be voluntary for this to occur. This is an insight from experiment 3, which I didn't have really time to go through here. If the choices are forced, this bias does not happen. This effect is driven mostly by devaluation of the rejected option. The within domain value adjustment are permanent with a slow decay, while the across domain adjustments are only relevant to the current judgment, but had no influence on the overall long-term value. Thank you.
Great, thank you, Wojciech. It's interesting that I was sitting here thinking about when you showed your model, the fluctuating value model, and how it was changing with time. It made me think that you could add an extra dimension to the data collection. If you could convince somebody to collect EEG or an MEG data in addition to the behavior, then you could actually maybe be monitoring states in the brain as they change. And I'm thinking about, there's a literature on brain microstates, and I'm wondering to what extent you could actually monitor in real time and look at how whether there is any neural signal that actually mimics that kind of model response that you're showing.
Yes, it's definitely like one of the next steps. I'm thinking of adding a neuroimaging modality to this. I haven't really decided which modality. I haven't actually considered like microstates, which is an interesting idea. But yeah, definitely. And also distinguishing between the within and across signals, I think would be interesting, especially that you also have like these behavioral differences that one are kind of permanent and the second are just very temporary.
Yeah, I mean, Marion, what do you think of this? What should he use?
No, EEG and MEG sounds really interesting indeed. I was wondering as well if you would you think that the choice-induced bias that you identify would influence people's confidence judgments in their choices as well? So I don't know if so you have the rating and the choice and then another rating. Do you think if you collected confidence in my preferences, it would be also influenced?
Mm-hmm. Yeah, definitely. Another thing I was thinking to measure confidence in some way, like there is a limited way to measure confidence, and that is simply by rating variability in a way, you know.
So how uncertain, how variable your ratings would be. Okay. Yeah.
That's one way, although it's very limited because we have only 2 initial ratings of the same item in the same category, so we have only 2 data points for that. But yeah, also, yeah, that's also something I'm thinking about next to designing a procedure which enables to capture this uncertainty or confidence, to see whether not only the distributions shift, but also do they change in the variance of the estimation changes based on the choices, and whether it's maybe a kind of a Bayesian update where the distribution kind of each choice kinds of like change the whole shape of the distribution that way.
That would be very interesting indeed. And then you can make the within domain and across domains comparisons again.
Yeah, exactly.
And just so that you know, in the chat there's a lot of activity by a certain person called Megan Peters, who's was very impressed with your talk, Wojciech, and has been making very positive comments in there. So she's just joined us. So any other questions that anyone else has? I don't see any. So what we'll do is we'll go on to the next presenter. Thank you, Wojciech. That was great. And so we're sticking with human behavior and systematic biases. And we've got this interesting ball and jar betting task that I really want to hear about. And this is a talk by Tara Issa. And so Tara, take it away.
Thank you. So I would just like to start actually by putting us in the mindset of what I mean when we say these rare event inferences. So let's just imagine for a moment that we're a doctor and we have a patient and we're worried about whether or not this patient has epilepsy. And the way we usually diagnose epilepsy is whether or not we observe a seizure. And so we send the patient home, we say, take a log of whether or not you think you've had a seizure. A week goes by, they come back, no seizures. We may think, oh, okay, this patient doesn't have epilepsy. Another week goes by, they come back, and now they've said that they have 1 seizure. So now as the doctor, we have a question. Does this patient have epilepsy? And does that 1 seizure, how much do we weigh that? And so I want to take a moment to really point out 2 caveats to these rare event situations. So the first one is we often have very limited information. As I just showed you with the 1 week's worth of information, there's a good chance that we might not see a rare event. So we might misestimate its occurrence or its frequency. And the second thing I want to point out is that when we do see that seizure, not all of the information we have should be weighed the same. Obviously, seeing 1 seizure is much more informative than all the days that we don't see a seizure. So now the question that we're left with is, well, how do we know how to weigh this? How should our evidence be weighed? So the way that we wanted to approach this question was in the form of a jars task. So in this task, we have a set of jars, and within these jars, there's different ratios of blue and red balls. And what we're doing is we're going to draw with replacement a small number of balls, 2 to 10 balls. And then the observer has to say, do I think that it came from one jar or the other? Now, what we're going to do to vary this and ask about rare events is to actually consider 2 types of jars. Asymmetric jars are going to have 1 color ball, here red, to be rare in both jars. And so this is just like the seizure. If we do observe a red ball, that's going to give us a lot more information about the jar than not observing a red ball. In contrast, if we have symmetric jars, the ratio of balls are going to be reciprocals between the two jars. So if I see a red ball, it gives me as much information in favor of one jar as seeing a blue ball gives me in favor of the other jar. So another way to visualize this would just be to think about, here are our ball draws. And when we see a red ball, we see that this gives us more information. It shifts our belief more so towards a higher jar, what we call our H+ jar, than in the symmetric case. So using this setup, the first question we asked was, how would an ideal Bayesian observer approach this question? And so we can consider this in terms of three main components of our model. So the first thing is we're going to set up our prior and say the ideal observer knows each jar is equally likely on any given trial. So there's no biases. The second part is now we can actually define an evidence weight. Because we've actually said that this H value tells us the probability of picking a particular red ball, we can compute that as saying, well, what is the probability of observing a red ball or a blue ball in either jar? And then the last component is our actual decision rule. So we're going to update this using a log likelihood ratio in which every ball drawn is going to give us a piece of information. And at the end, our belief, which here is labeled as Zn, if it's positive, then that's going to motivate us to report it's the H+ jar, that less rare jar in our asymmetric case. And if it's less than 0, we're going to report H-. So using this model, the first question we asked was, what happens if you give the observer many, many trials? What we would expect is that over time, the number of responses would begin to match the prior, meaning we'd have about half of our responses in favor of the H+ jar, half of our responses in favor of H-. But what we found was actually really interesting, which was that in the symmetric jars, we do get that response fraction that you'd expect. But in the asymmetric jars, there was actually a bias, and you have more responses in favor of that more extreme H- jar. And so this is a pretty surprising finding, given that this is supposed to be an optimal observer. So the question we asked was, well, do humans show the same bias? So in order to answer that question, we expanded this task slightly, and we used Amazon Mechanical Turk, which is a crowdsourcing platform, to poll subjects on these exact same tasks. So what we've done now is set up 5 blocks of jars, and we varied the difficulty and the symmetry. So just a quick rundown, we have a control, hard asymmetric, hard symmetric, easy asymmetric, and an easy symmetric. I'm not sure if you guys can actually see the laser pointer. Can you see this now? Okay. So in this particular case, we asked subjects if they could do the same task. And what we found was that just like we saw with our ideal observer, in these asymmetric blocks, subjects were showing the same sort of a bias. They were also reporting the H- jar more often than they should be. And taking this even one step further, I want to go back to what we said about limited information causing biases. We had given them 3 distinct numbers of balls to observe. They could see 2 balls, 5 balls, or 10 balls. And just in line with what we'd expect, we see that when they only had 2 balls, they see a more extreme bias, more responses in favor of the H- jar, which is in line with what you'd expect, since observing a rare ball in the asymmetric case would be very rare there. So this left us with the question of whether or not the biases that are causing this response fraction were the same in human subjects as in our ideal observer. Was it truly caused by the same mechanism? And one of the motivations for this was because we know that humans also show repetition biases and sequence biases when you're doing trial after trial responses. So there is a clever way to actually distinguish between different types of biases. And so what we did was look at a block of trials, and you can actually go through trial by trial and compare the sequences of responses. So for each pair, here we see there would be an alternation from H plus to H minus, followed by a repetition from H minus to H minus again. And when you actually quantify the frequency of each of these responses, you can compare them to one another. So here, if I plot how often I alternate in either direction, you can get a sense of whether or not you have particular biases. So if you don't alternate in either case very often, then you're more likely to repeat your answers. You have a repetition bias. If you alternate often, an alternation bias. But across on the downward diagonal, we also can consider whether or not I have an item bias. Do I prefer one particular jar? And so we used this and plotted where do our subjects fall in these different categories. And what we see is very reassuring in terms of we see these orange points are our symmetric trials, and they're very close to the center, which is actually where we would have unbiased space. These green points, in contrast, are our asymmetric blocks, and they show very distinctly a preference for the H minus jar, the more extreme jar. So now we've confirmed that this bias is really coming from very similar motivations as our ideal observer. But since we know the ideal observer shows a bias, we're now left with the question of, is this bias the same? Is that all that we're seeing? Are they just doing the optimal thing? So we gave ideal observers the same trials as each subject and normalized to see how similar they really were. And what we saw was that our symmetric trials, again, all of our subjects were very close to being very similar to the ideal observer. But what was quite surprising is there was a large number of subjects that still had an enhanced bias, even beyond what we expected. So this really asks the question then, what's causing that enhanced bias? So in order to get at this, we decided to use model fitting and model comparison. And so the first question is, are even using Bayesian methods? So I'm not going to go through all of the models, but we tested out 4 different Bayesian type models. And the main important point was this noisy Bayesian model, where we said, what is the actual weight you're giving to a rare ball? And then some alternative options. Perhaps you are doing things just like the ideal observer. Perhaps you are applying a prior, and maybe you're not even using all of the information. And then the other set of models that we considered were some heuristics. Maybe you're just guessing. And then these 2 were what we call rare ball models, where if you saw any rare balls at all, then with some probability, you're going to respond for a particular jar. And so using these model comparisons, what we found was most of our subjects fell within these Bayesian models. But really importantly, they were not necessarily using optimal approaches. And so we wanted to take this one step further and now go back to that and say, well, what were those enhanced subjects doing? What best fit model did they have? And so if I just plot those same plots we see before, but with the models that best fit our subjects, we see that there's 2 really distinct groups that are shifted up this diagonal. The first one are those noisy Bayesian subjects. And what's really important about this is that that shift seems to be caused by the rare ball weight. So here, these black lines are actually showing where the ideal observer believes we should set the weight for a rare ball. And we can see that as you have more and more of this enhanced H minus response, you're weighing these rare balls less and less. And then the second one are people who actually have a prior. And so there is also some innate bias towards these more rare options. So in summary, I just want to bring back what our main findings here were. So we saw that both the ideal observer and human subjects showed a bias towards more extreme versions in rare event decision-making, and that subjects did appear to be using a Bayesian, but not necessarily optimal approach to these problems. And this is what was actually causing us to have an enhanced preference for these rare balls or these rare events. And that was often caused by underweighing the rare events themselves or having innate biases. So I'd just like to thank all of my collaborators at the University of Colorado Boulder, Houston, and Pennsylvania. Thank you.
Thank you, Tara. That was great. It was very, very clearly presented. Thank you, and everyone's been presenting beautifully today. Very clear talks, and I would like to ask people in the audience to ask some questions and of these interesting presentations. I was just thinking about, as you were presenting, Tara, I was thinking about why people go out and buy those lottery tickets.
Yes.
And, you were talking about seizures, and that's something that I can relate to because I used to test epilepsy surgery patients, and those are rare events. They are rare events, but lottery tickets is another thing, and lightning strikes as well. Lightning strikes on a golf course is another one where people go out and play golf. But my question to you is, how does the mental affective state of a person affect this kind of decision making? Do you have a sense of that? And let me tell you what I'm saying, what I'm thinking. If somebody is more depressed, is that likely to change their the bias that they've got in this with these kinds of decisions?
That's an excellent question. And our data cannot directly speak to that since it is crowdsourced and we didn't ask any questions about emotional or mental state. But there have been studies that have shown people are much better at assessing negative rare events than positive. And we actually have different biases based on those particular situations. The other component is actually experience versus if you're doing this based in probability. So oftentimes experience can greatly shift these biases. And so in some cases, you actually see the complete opposite bias based solely on how you structure these types of tasks.
That's interesting. And the other thing, as you were answering my question, it made me think of how younger and older people look at things as well, how older people, I'm told, have a more positive, may make more positive outlook on things and maybe make more positive decisions than people who are younger. There's again something to look into, age differences. But we have a question from Joseph de Souza, and who says that was a lovely talk. But his question is, how about one trial learning in children, and can this be modeled?
Ooh, that is a good question. The short answer is I would have to think about it. I think, well, first of all, we were only dealing with adults in this particular case. And so I think that even going back to your comment about age, right? And as you age, you have different experiences. I think it's very difficult to compare this type of a task to something in children only because of the developmental and the experiential portion of it, right? So whether or not we like to admit it, this task comes with some amount of bias built in from people automatically having experiences. So perhaps if we restructured it, then we could maybe get at some of those other questions, but not in its current state, I guess.
Okay, so some remodeling of the task, no pun intended. All right, well, on that note, I think it's time to draw it to a close. And I'd like to thank all of the presenters for doing a presenting beautifully clear slides. It was an absolute pleasure to watch this. And I'd also like to thank Siddharth Suresh, who did an absolutely fabulous job navigating the back end for us. So thank you so much. Enjoy the rest of Neuromatch. We've got one more day tomorrow, so hang in there, and have a great evening.
Thank you so much.
Thank you so much for having us.